guys, welcome to Vedanta Teach English. I am Miss Kopika, your own biology master teacher, and welcome all of you for Biology 360 Club. So I know all of you have taken the membership for Biology 360 Club, and we are going to start our first ever session, that is reproduction unit. And how many marks am I going to get, ma'am, at the end of the session? If I sit from now till the end of the session, how many marks am I going to get? You're going to get approximately 40 marks for your NEET examination, because sexual reproduction in flowering plants and human reproduction is one of the high weightage chapters, which can fetch you at least 5 questions from both the chapters. So you already know that you're going to get 40 marks if you're going to start the session right now and stay till the end. So quickly share the link with all of them and ask them to join for Biology 360 Club. So don't disappoint us, because this is done totally for students who are not able to cross 300, 320, 350, no matter how much they know the concept, or maybe they know the concept but they're not able to solve question. So today we are going to teach you in such a way that you're going to understand everything very simple with our handwritten notes. At the same time, some questions are going to pop up in the middle. Also, there is going to be NCERT lines. So if you look at it, it's like a one-stop solution for all of you. Yes, I hope all of you are here. Quickly like the video. Let's make this Biology 360 Club, you know, the talk of the town, and let everyone talk about it and let everyone get FOMO for those students who did not join. Okay, ready all of you? Yes, the energy is very nice. So I hope all of you are ready for it. Fine, shall we start? You can see me, I am fully ready. I'm fully, you know, energetic for the class. So let's start the class. And let me tell you, students, if you stay till the end, you are going to be mind blown that, oh my god, this is how the concept should be taught. Sexual reproduction in flowering plants is a very long chapter. Human reproduction also. Some students like to learn about human reproduction but do not like about plants, which your partiality is about. But let me tell you, today you're going to learn this chapter with me in such a way that you'll be like, ma'am, even plants are so interesting. Yes, I will make sure that plants are interesting, being a zoology teacher as well. So shall we start? Wait, awesome. Okay. Energy is awesome. Red hearts and fire emojis in the chat box continuously. Whenever you feel like ma'am's energy is going a little down, make sure that you boost. Okay, boost my energy. Fine? Yes. So shall we start? So sexual reproduction in flowering plants. So we are going to understand what exactly these flowers are going to do, or why do we call flowers the site of reproduction. All of this we are going to decode in this class. Okay, now if you look at here, you will see the range of questions. Okay, how many marks have come each year from this chapter, and last year also we got around 4 questions from this chapter, right? So that means you have good chance of getting 16 to 20 marks directly from this chapter, and the questions are not that difficult. So shall we go? Okay, shall we start? Yes, ma'am, definitely. Now, the merits of flower that we enjoy gazing at, the scents and the perfumes that we spoon over, the rich colors that attracts us, are all of these as an aid to sexual reproduction. So they're telling us we love a bouquet of flower, right? If I give you a bouquet of flower, I don't think any student is going to tell me a no. Everyone is going to enjoy that. So we enjoy the beauty of the flower, we enjoy the scents of the flower, we also enjoy the rich colors in the flower. Now just imagine you are an insect. Okay, all of you consider yourself as an insect. If you're an insect and you're flying at a good height, if you look down, you're seeing a very beautiful inflorescence, or you're seeing a good amount of red color flower standing. Automatically you're attracted to it. So aids for sexual reproduction. That means the flowers are produced in such a way so that sexual reproduction can take place. Are you all understanding? That means they are not just making these flowers. It's not for human selfishness that they are making all these beautiful colors, perfumes and scents. They are doing this so that they can reward the abiotic and the biotic factors who are coming for pollination and helping in the ultimate goal of making a fruit or a seed. Clear to all of you? Yes or no? That is important. Is the frame okay? Yes, students, the Joe shouldn't go down. Okay, it should be there till the end. I want all of you to share the link as well and like the video as well. Okay, yes. So you can see that this is very important. Rich colors, scents, aid to the sexual reproduction. Okay, aid to sexual reproduction. Yeah, it works. Okay, now all flowering plants all flowering plants show sexual reproduction. Okay. Okay, 1 second. Just a little. That's why it's getting cut. Yes, I'm going to take the whole lesson, obviously. Yes. Thank you. Yes. So, students, look here. All flowering plants show sexual reproduction. So, that means what? Here, they are talking about your angiosperms. They're talking about your angiosperms, because all of you know that when we speak about gymnosperm, we are talking about the naked seeds. Right? That is what we have learned. We have learned about algae, we have learned about pteridophyte, we have learned about gymnosperm, angiosperm. All the flowering plants show sexual reproduction. That means flowers are the site for reproduction. Okay. Now, end products of sexual reproduction is fruits and seeds. That means the ovary develops into fruit and the ovule develops into seed. Clear or not? Ovary develops into fruit and seed. Ovule develops into seed. This is the end product. So, it is not made for your selfishness. It is not for you to give it to Chintu, Bunty or anyone. It is for you that the plants are making these flowers so that they can do sexual reproduction. They can ultimately form a fruit. They can ultimately form a seed. Clear? I hope this is super duper clear to all of you. Now, let us also understand what are we going to learn in this chapter. That is also important for you to know, no? Let us also understand the morphology, structure and the processes of sexual reproduction. That means, students, when we are learning this chapter, you're going to learn about the morphology, you're going to learn about the anatomy, not exactly the anatomy, but you have studied most of it in your morphology in flowering plants in your class 11. Now, to a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. A very important line, and they can ask you an assertion reason from it. They can ask you a statement based question from it. That means, to a biologist, what is flowers? It is morphological and embryological marvels, right? So, this is what you study in your morphology chapter. That flowers, let's say you're talking about the shoot, root, everything, no? So, let's say this is what you have learned. This is the soil, okay? You learned that there is root, okay? Root is developed from your radicle. All of you answer, your energy is going to decide this entire session, right? Radicle is going to do the root. Then, on the top of that, we have learned that there is shoot, okay? There is shoot. So, there is root apical, shoot apical. So, this root apical, shoot apical plays an important role. That means what? This shoot, students, the shoot apical meristems are going to get modified. Okay, some of them are going to get modified to form the floral meristem. And this floral meristem is going to ultimately form my flowers. Right? Ultimately form the flowers. So, everything has a role. Everything has a role in this, no? So, look here. Some of the, do you see any flower in the root? Has any students digged, and seen something in the root? Yes, tell me. What is this, ma'am? Is this botany or zoology? That much you people can't understand. Huh? Sexual reproduction in flowering plants means what? No one has ever seen a flower, right? No one has seen a flower in the root, but everyone has seen it in the shoot itself. So, shoot apical meristem, some of the cells are going to get modified to floral meristem and form the flowers. Okay, clear or not? Got it? Yes. So, students, please understand, we are going to do your entire reproduction unit. So, please stop asking, is it zoology or botany? Entire thing, that is sexual reproduction in flowering plants as well as human reproduction, we are going to do it. Clear? Clear or not? So, don't be like, ma'am, is it botany, is it zoology? I am a zoology teacher. I have taught you all the zoology chapter. That does not mean that I do not know, botany. So, stop asking this unnecessary question and try to focus. This is a very important class. Okay. So, we are going to talk about this. So, shoot apical meristem, some of them got modified to floral meristem and they are ultimately forming the flowers. So, I can say that flowers right? I can say flowers are modified shoot. Yes or no? Agree with me? Agree with me on this basis? Tell me. Yes. Right? Okay. So, moving forward. Now, what are the events? Ma'am, what am I going to learn in this chapter? Exactly, I want to know what is the flow of the class, right? So, I'm going to tell you that exactly what you're going to study, students. We are totally focused on reproduction. All these notes will be available on Vedantu NEET English Telegram as well as Biology by Gopika Telegram. You can join both of the Telegram. For my classes, definitely you can join this Telegram. And for all the updates related to physics, chemistry, biology, you're going to get it in Vedantu NEET English Telegram. Okay. So, reproduction when I talk about, mainly this chapter is focusing on 3 events. Events are pre-fertilization, okay, fertilization, and what? Post-fertilization, ma'am. Post-fertilization. So, students, here, this entire chapter revolves around these 3 topics only. That is, pre-fertilization, fertilization, post-fertilization. Now, when I'm talking about pre-fertilization, what am I exactly talking about? I am telling that in pre-fertilization, the first job that I have is to form gamete. So, gamete formation. After forming the gamete, what am I supposed to do with the gamete? Am I supposed to keep it in the fridge and just look at it? No, I'm supposed to make sure that the gamete gets transferred so fertilization can happen. Now, what am I talking about? Pollen grains have to go and land on the stigma because the male gamete is inside that. So, my thing is that, right? My thing is that. So, after the gamete formation, I have to focus on gamete transfer. Yes or no? I have to focus on gamete transfer. So, these are my pre-fertilization events, okay? Now, in this gamete formation, students, what am I talking about? When I am trying to make microspores, I will call that process as microsporogenesis. When I am going to make megaspores, I am going to call the process as megasporogenesis, right? I am making male gamete and female gamete. Then only fertilization is going to happen. So, I am here talking about how I can form the male gamete and how I can form the female gamete. So, here when I talk about gamete formation, I am talking about 2 things. That is microsporogenesis and megasporogenesis. Clear to all of you? This is what you're going to study in the starting of this class. Next thing that we are going to study is gamete transfer. Now, I made the pollen. Now, all of you tell me, how am I supposed to transfer this? Am I supposed to take it or call some other person to go and transport it, or are there people to do it? So, this gamete transfer, can I simply call it pollination? Yes, ma'am, you can call it pollination. So, here I am focusing on different kinds of pollination and the agents who are going to do pollination, okay? Now, the second event, fertilization. Now, when I talk about fertilization, here you are going to study one and only double fertilization. And this double fertilization is going to lead in 2 things. What are they? One is a zygote formation. Right, a zygote I will be able to get. Another thing that I will be getting is an endosperm, or I can call it the pen, primary endosperm nucleus. These are the 2 things that I'm going to get at the end of my double fertilization, right or wrong. Now, when I talk about post-fertilization, you people tell me, what is the ultimate of all the process that we are going to do? That is, we are going to make sure that the plant is going to form ovule into seed, right? And the ovary into fruit, right? Ovary into fruit. These are the post-fertilization events. So, is this concept clear to you? What you are exactly learning in this chapter? Okay, you should know what you are looking forward in this chapter. All these notes will be available on the Telegram channel, so don't worry. And during the break time also, you people will be practicing the questions, okay? For those students who's asking, ma'am, will you do questions, will you do PYQs? I will be doing some PYQs. Other than that, in between the break, you are going to do some PYQs as well from the Vedantu's end, okay? That will be proper quiz pattern. So you will be going through that quiz when we go for a break, okay? So when I take a break, you will be solving the test and your marks you can tell me after the break. Clear? Okay, this is the whole thing that you're going to study. That is pre-fertilization, fertilization and post-fertilization. What is going to happen before that actual fertilization, what is going to happen during the fertilization and what is going to happen after fertilization. This is all about it. This is what the chapter is. So when I talk about reproduction, please try to understand that I am talking fully focused on angiosperms. Okay, I am fully focused on angiosperms. Because here I am not going to have naked seeds, I am going to have fruit and seed. Ovary is going to develop, ovule is going to develop, right? And when I am talking about angiosperms also, I am talking about the flower which is going to be the site of reproduction. Site of reproduction. I hope no doubts on this. Easy peasy, no? Simple things this is, okay? And those who haven't liked the video, quickly like the video. I can see some of you haven't liked the video, okay? Yes. So this is the events. Now moving forward, we are going to understand the structure of a flower. Flower is very important. Yes, ma'am, definitely. Without flower, how am I supposed to study? So in morphology, you have already studied the structure of flower. But here, it is important for you to equally know the structure, how to draw, how to identify. It's very important. Okay. So let's say, students, we start from the bottom portion. Okay, we start from the bottom portion. What am I drawing? So as I'm drawing, you people tell me the labeling. Okay, so I'm going to draw a stalk kind of structure. You will only need these notes for sexual reproduction and flowering plants. You will not have to make your own notes. You can make the lecture notes, but you do not have to go search for another note. This notes will be more than enough because I will write everything for you people. Now here I have drawn a pedicel, and after that I am drawing a swollen part which is called the thalamus, right? Which is called the thalamus. See, this is the swollen part called the thalamus. Now on the thalamus I am going to draw some important structures like your female and male part, right? So here we go. I am drawing a landing place where the pollen lands, a complete bigger stalk, and then we come down to a swollen part called the ovary. Stigma, style and ovary. After the ovary, inside the ovary, okay, I will be drawing a small ovule, okay, which will be attached to a placenta, okay, in an ovarian follicle. Now, after that, ma'am, you are supposed to draw a bilobed structure which is called the anther, right? Anther, stigma, style, done. Now to attract the insects, I need some beautifully colored structures. What are they? My petals, right? I'm going to draw some petals which are going to attract the insects. Flower done. Okay. Now there are one more whorl in this which is called the calyx. Okay. Calyx, or we simply call it sepals. Flower structure is easily done, students. Any doubt in this? Now shall we label it? All of you help me label it. Okay, starting from the starting. Okay, yes. So this is the place where we hold and give flower to someone. Okay, that was a very nice dance move, no? So this is called the pedicel. This is called the pedicel. Okay, then the swollen portion is called the thalamus. Then this portion that you're seeing, the sepal, which is a calyx, okay, that has to be what? That will be protecting the bud. Okay, that will be protecting the bud. So this is the sepal. I would like to write the whorl, that is the calyx, which you represent with your C, K, okay. Then here comes the corolla, which has units called petals. So these are all whorls, and in that there are units, okay. And then you come to this portion which is your androecium, okay, which is the male part, okay. and this is your female part, which is called your gynoecium. Okay, gynoecium. This is what your flower has. Now, this is what your flower has. Now, let me come into more depth of this. That is, they are telling that everything is not essential. Do I need to, think of everything? No, I do not feel that everything in my life is essential. No, some will be essential, some are very necessary, like our family, very necessary for us. But there will be some friends who are like, okay, if they are there, it's good. If they are not there also, it's fine. Some things are very essential for us during our life. Some things are not essential, but they do play an important role in our life. No. So here also, students, when I talk about this, understand that your androecium, okay, androecium and gynoecium, androecium and gynoecium is going to be called as the essential world. What are they called? Essential world. Okay, essential world. Why, ma'am, they are called the essential world? Why are you not calling corolla and calyx? I am calling these people the essential world because they are directly involved in the fertilization. I'm talking about fertilization. I'm talking about how the flowers are going to be benefited. So they are essential world. Okay. Then when I go further, and talk about your calyx and corolla, I cannot tell them that you people are useless. Definitely not. If petals are not there, insects are not going to get attracted. If sepals are not there at the bud stage, the flower is not going to be protected. So they are not unnecessary, but they are accessory, right? They are accessory, or we can call them the non-essential world or the accessory world. Okay. Students, such simple questions also can come. Don't always expect, like, next high level questions. Sometimes simple questions also students will not know. Like, they will not know which is, essential and non-essential word, because they will be reading NCERT in a hurry. So they will not focus on it. So such kind of questions also can come. I hope this is clear. Okay, this is clear. Now, come back to NCERT. So I have tried as much as possible to put every single line that we are not supposed to miss from NCERT, because when I'm teaching, obviously I will be telling it in my language, but NCERT line will be little different, and when you read it, you might get confused. So that's why I have made sure that wherever required, NCERT snips are also added. Now look here, several important hormonal and structural changes are initiated which lead to differentiation and further development of the floral primordium, okay, or primordium. So this is what I told you, that some shoot apical meristem is going to get modified, undergo differentiation, undergo development to become the floral meristem. That ultimately is going to make the flower. So when the plant is blooming itself, they already know that the decision to form the flower is already decided. Now, the moment you are born, your parents are not going to be like, you will become a doctor. Some parents do that, but majority of the parents will not be like, as soon as you are in the hospital, they'll be like, you are a doctor. They will not do that. No, they will take some time. They will see what you're good at. Maybe some kids are good at something else, some kind of art or some music or, games. So athletic wise, they might go. Some of them will be proper education wise. So different things, no? But here, the plant has already decided that I am going to make the flower, and everything has changed according to that. Morphologically, structurally, development wise, everything is already decided. So here, look here, inflorescence are formed which bear the floral buds and then the flowers. Why inflorescence are formed? Inflorescence are formed when the flowers are too small and they are not so attractive. So when the flowers are too small and not, attractive, suddenly inflorescence will be formed. Everyone will come together and they'll be like, we look beautiful together, let's be together, right? You have racemose and cymose inflorescence. That and all you will study in your morphology. Racemose and cymose. Clear to all of you? Any doubts so far? Any doubts so far here? So I'm just going to write that as well. Okay, shoot apical. Are you all enjoying? Come on, tell me in the chat box. Floral meristem forming the flowers. Yes. Whole story is done. No doubts? Perfect? Yes. Fine? Okay. Perfect. Perfect. This is the whole story. So, flower structure is clear. Okay. Ovule is clear. Now, again, ovule, anatropous ovule, we have to study in detail, right? So, that's why we don't have to focus right now on ovule, but just understand this is a structure, landing, anther, that is what you're going to study in the further. So, this also is very important for you to know what are the changes that happen, because they can give you missing, they can ask you to fill. So that time you should know several hormonal as well as structural changes have happened in the body of the plant to make sure that the flower is happening. Okay, flower is going to happen. Perfect. Now moving forward, structure of an anther, very, very important. I think every year questions also, you know, based on this only it will keep coming, because it's a very important topic. Now students, when I talk about anther, first we should draw an anther. So lot of drawings are there in this diagram, in this chapter. So I will draw all the diagrams. I just want you people to write the most important concepts, okay, in your lecture notes. Okay, I want all of you to make lecture notes, and if possible share it with me on my Instagram page. So the best lecture notes, I will share it on my story as well. Okay, same name itself, biology, Gopika biology. Okay, yes, look at this students, you might look like it's some, mom, what have you drawn? But this is your anther. Okay, now look here, this whole stalk-like structure which is holding the anther is called as your filament. What is it called? Filament. And if you look here, this is your anther. This is your anther. Ma'am, what's so special about the anther? Yes, ask me about it. Anther, what you're seeing here, how many lobes it has, students? So let's say L1 and L2. That means what? Anther is bilobed. Okay, very important. Anther is bilobed. Important. Now, when you look at this bilobed anther, there are these cavities. Okay, there are this structures which are going to be called as theca. Okay, they are going to be called as theca, inside which a microsporangia is going to be there. So let's just imagine, okay, I do not have a cover. So let's say this is a cover. Okay, this is the cover. Now you tell me inside this, or let's say this is a glass. Inside this juice is there. What will you call this? You will tell, ma'am, please take your juice glass. Let's say inside this water is there. You will be like, ma'am, please take your water. Let's say inside this tea is there. Ma'am, you will be like, ma'am, please take your tea. That means depending on what is inside this glass, the glass name is going to keep changing. Right, same thing here students. When microspore is inside this, I will call this a microsporangia. When pollen grains are inside this, I will call it a pollen sac. Understood? So, when microspore is inside this, this is a microsporangia. When pollen grains are inside this, this is a pollen sac. That means microsporangia matures to form your pollen sac, and inside pollen grains are going to be there. Clear to all of you? Yes or no? So this is the simplest thing. Okay, this is the simplest thing. That is, anther is there, bilobed is there, thecas are there. So I will also call it, dithecus. Dithecus. That means one lobe has two thecas. So totally 4 thecas are there. So we are talking about one lobe. So it is dithecus. Okay. It's like a small cavity where your microsporangia is going to be there. Now, when you look here, you know that this is bilobed and dithecus. Now we have to see the magnified image. Before that, this filament is there, no students. Let's assume this is the thalamus. Let's assume this is the thalamus. Now, one line is given in your NCERT, which is very important. Okay, I am not going to miss one line of NCERT also. So, blindly just listen to the class. Look here, this is the thalamus. Now, filament is, now I am here. Okay, camera is there. Now, I am at distal end with the camera. But as I walk closer, don't tell me, ma'am, I'm getting scared. Okay, as I walk closer, now I'm going to be at the proximal end. As I move further, I'm going to be at the distal end, right? I'm going to be at the distal end. So, compared to the thalamus and the filament is going to be at the proximal end. And your anther is going to be at what end? Ma'am, anther is going to be at the distal end. Clear to all of you? Two ends are clear? Okay, two ends are clear? Yes, very good. So, Anther, bilobed, dithecous, no doubt in this, okay? And filament is at proximal end to the thalamus, and your anther is at distal end. Any doubts so far on this? No doubts? Perfect? Yes, okay, very nice. Okay, so theca is like a cavity. Inside this, your microsporangia is going to be there, okay? Are you all liking the video? Is someone putting the target? Who's going to put the target today? Meghna, put the target. I think Tanuja is missing. So, Meghna, put the target. Quickly. So, Meghna is going to put the target and you people have to achieve the target, okay? Chalo, let's go forward. So, this is clear. Now, let's say I want to draw a bigger structure of the anther, okay? I'm going to draw a bigger structure of the anther. So, it is going to look something like this. I think all of you would have studied this structure in your... Okay, does this look a little different, students? Yes, look here. So the bigger structure, okay, bigger structure. Now inside this, what I'm going to draw, ma'am, you're definitely going to draw the theca. See, I drew a big theca. Okay, here, 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 and here. Let's say I draw a structure like this. Okay, done. Now 4 thecas I have drawn. Okay, 4 thecas I have drawn. Now remember that if we run a groove like this, we are going to get the 2 lobes and the 2 thecas, right? See, I'm going to run a groove like this. You notice that, yes ma'am, 2 lobes are there, 2 thecas are there. Perfect ma'am. What you told, I understood. Dithecous and it is bilobed. Okay, fine. Now students, if I want to see the structure of these people, they are telling that these people are tetragonal. That means what? So, let's say from here to here, I make a connection. Okay. And here to here, I make a connection. Okay. And from here to here, I make a connection. And from here to here. Yes ma'am, they are tetragonal also. Right? They are tetragonal also. Correct or not? So, they are tetragonal, and if you go through a groove, okay, you cut it longitudinally also, or you cut transverse section, you will notice that there are 2 lobes and thecas. Now, moving further into the depth of what is microsporogenesis, understand that this microsporangia, okay, I'm just going to remove the tetragonal structure, students, so that I can just fill it up there, okay. Yes, so I hope that is clear, okay. Now, let's go to the microsporangia. Now, I told you if microspores are inside it, you call it microsporangia. If pollen grains are inside it, you're going to call it as your pollen sac. Now, this has 4 layers, which is given in your NCERT. So, let's say the first layer that I've drawn here is your epidermis. First layer is epidermis. Inside that, there is going to be one more layer. Okay, inside this, there is going to be one more layer. That is called as what? Endothecium. So, these people are going to protect your microspore mother cell so that they can do meiosis and finally form what is required. Okay, is the colors not very different? One second, students. If the colors are not different, then you will not understand. That's why I am specifically asking. Okay, let's draw it with this color. Okay, next layer, one single layer called the endothecium. Okay, after this, there is going to be a multi-layered structure. Okay, multi-layered structure, which I'm going to draw it with blue, which is called the middle layer. It is made up of 2 to 3 layers. That's why I told it multi-layered. Okay, a little more thicker layer I'm drawing. This is your middle layer. Okay, middle layer. Now, inside this, okay, inside this, there is going to be tapetum. What is going to be there? Tapetum is going to be there, okay. This is your tapetum. Okay, tapetum. The green color one is your tapetum. Layer is done. Layer is done. Yes. Yes, it is enough. Mithula, more than enough. But you have to listen carefully. Okay. Yes. So, students, here, one second. Now, this layers, lot of questions come based on this layer. So, I want all of you to not talk to each other, ask about reproductive health. Okay, we know exactly what we are going to teach you people. So, don't worry, just have the trust. Okay, Don't keep asking about another chapter when we are doing in this chapter. In this only you will waste your half time. So tapetum is there. Now between the students there's a very, very important layer, very important layer. That is not layer, they are tissues called as the sporogenous tissues. Now you are going to understand microsporogenesis also with this structure. Okay. Now the diagram is clear to all of you. Can you all see the diagram very carefully? Okay, chalo. If you can see the diagram, we will go into the labeling. So I told you first layer is epidermis. Yellow color layer is going to be endothelium. Okay, endothelium. Then the blue color layer, which is a multi-layered, is going to be middle layer. And one of the most important layers is going to be the green one, which is called the tapetum. Because this tapetum is going to help with the nourishment. And in the between, the tissues that you are seeing is called as your sporogenous tissue. Sporogenous tissue. So to remember this trick, there is one small, okay, to remember this people in order, you can just remember like this. Elephant, okay, elephant. So here P is there, you're going to remember E and P is going to be there. So it is epidermis, okay, epidermis. So elephant eats my tomato, okay. So you are a farmer, elephant eats your tomato. So that means what? E for EP means epidermis. Okay, EP means epidermis. So epidermis, endothelium, middle layer and your tapetum. Now let's go each layer one by one. Are you all ready? Now they are telling that epidermis, endothelium, epidermis, endothelium and middle layer is going to help. It's not like they are useless students. They are going to help. Okay, they are going to help. They are like, ma'am, like we will do the job, but we are not going to do the nourishment job. What we are going to do is, we are going to help with protection. They are going to help in protection. These are lines of NCERT. Make a note. Protection and dehiscence. What is dehiscence? Imagine I have in my hand, okay, tightly I'm holding, let's say, some pearls. I'm holding some pearls in my hand, okay. Let's say those pearls are your pollen grains. If this anther does not break open, how will the pollen grains get transferred? So dehiscence means what? That breaking open of the anther so that the pollen grains can get scattered, okay. Pollen grains can get scattered. So epidermis, endothelium and the middle layer is going to help the protection and dehiscence. But on the other end, you have your hero, that is your tapetum. Tapetum is the hero. Why? Because this fellow is going to help in the nourishment of the growing pollen grains. Okay, it is going to help with the nourishment. So it is going to help in the nourishment of the sporogenous tissue to become the pollen mother cell. Okay. Now, nourishment, it is going to, at the same time, it is also going to help in the secretion of, okay, secretion of Calais enzyme, whose job I will tell you in some time. Okay, Calais enzyme. Just remember this, Calais enzyme. Okay, yes, very good. So they are going to help with the nourishment, but these three people are going to help with the protection. Done. No doubts on this. Now let's go to sporogenous tissue. What do you people know about sporogenous tissue? Ma'am, I know that these tissues are present in the center. Okay, so this sporogenous tissue, students, it is going to be present exactly in the center of your microsporangia. Okay, this is your microsporangia. All these are going to be your microsporangia, right? Theca is just like a cavity. Inside that your microsporangia is going to be there. And let's say, since I drew your microsporangia in pink, I'm just drawing some dots in the center. So microsporangias are going to be there. Okay, you cannot see it definitely, but you will be able to see it in the, in the notes. So we will draw it with a different color. Okay. Microsporogenous tissues will be there in the center. Okay. After that, they mature. Pollen grains will be inside that. Got it? So this is clear. Now, let's also label this. Okay, microsporangia. Now, to make sure that these two lobes are connected to each other, students, you have a connective. Okay, you have a connective in the center to make sure these two lobes are connected to each other. So whole thing is done. Nothing is missed, I hope, right? Everything is there. This is a connective. Okay, this is a connective. I'm just going to erase this and write it a little on the top. Connective. And this is going to be microsporangia. Okay, done. That whole thing is clear. Now, coming back to sporogenous tissue, I told you it is going to be present in the center of the microsporangia. Yes, ma'am, understood. These sporogenous tissue, students, these are also called as your PMC. That means these people are going to undergo some development, and these people are going to become your pollen mother cell. These people are going to become your pollen mother cell or microspore mother cell. Microspore mother cell. Clear to all of you? So sporogenous tissue is very, very important. Okay. Yes. 35 likes to go. My God. All of you drink some water. What happened, Joey? So distracted you are. Yes, clear? This is done. Pollen mother cell or your microspore mother cell. Okay. Keeping all this in mind, let me tell you, keeping every single point in your mind, you are going to understand microsporogenesis, because everyone are going to play one or the other role. So it is important for you to remember all of it. Clear? Sukanya, you can just write lecture notes fast, because see, you cannot do the coloring and all of this like this, no? Anyway, I will send you the notes. So you make sure that you write the lecture notes in such a way that only you understand. No one else have to understand, students. It is just for you people. Okay, I don't think I'll be able to do this. I've just put the trick in the box. Otherwise, you'll not know which is the trick, what is what. Okay, done. Thank you, Mohammed. Yes. Baswa sir will take after my class. You want Baswa sir also to take in between this, like one side, one board human reproduction, one board sexual reproduction. After this class, it will take, students. It's a long marathon. We have already told you it's a long marathon. This is going to be the marathon to get you 360. Okay. Thank you. Thank you, Joey. Okay. Now, microsporogenesis. Very, very important. Very, very important. Right? Microsporogenesis. Chalo, let's start. Yes, after this class, on the same live, on the same video, you do not have to go anywhere. Baswa sir will come here. Okay, Baswa sir will come and start the live. Understood? Okay, half we can't do, students. Please try to understand. If you want 360, this is not the shortcut to make teacher, like, you know, people. I know that a lot of you will be searching sexual reproduction under 1 hour. If you read NCERT also, it will take more than 1 hour. Then how do you expect a teacher to finish it in 1 hour? Okay. Yes, definitely, Mithula. Okay, now coming forward, microsporogenesis. What is microsporogenesis? Ma'am, microsporogenesis is where, with the help of the microspore mother cell, we are going to form the male gamete. Right? That means with the help of, we are going to form microspores with the help of microspore mother cell. That also I can tell. Right? That is the definition given in your NCERT. Now, how did this MMC come, PMC come? For that, we start again from sporogenous. Okay? So, students, you have sporogenous tissue, which is going to develop and form your microspore mother cell. Microspore mother cell. Okay, microspore mother cell. Now, from here, your main process starts. So, I want all of you to pay attention here. Your microspore mother cell or your PMC, or we can write or PMC. Pollen mother cell, okay? Pollen mother cell with the help of meiosis. With the help of meiosis. That means these people are going to be diploid, 2N, okay? Because meiosis is going to happen, we are going to end up getting haploid, okay? So, with the help of meiosis, we are going to happen, what is going to happen? Here, we are going to form microspore tetrad, okay? Microspore tetrad. Now, listen to me carefully. listen to me carefully. This is your microspore. Okay, this is another microspore. Tetrad we are forming. Okay, why this is called tetrad also I'll tell you now. Oh, everything is going missing. Okay, I hope it doesn't come back somewhere. Yes, now students, look here. This whole thing is going to have a coating. Okay, like one whole layer to keep them together. This is why we call this tetrad. What is this called? Microspore tetrad. Okay, microspore tetrad. I'm so sorry, the board is dancing. Microspore tetrad. Now, you can ask me a question, ma'am, why are these people together? Why are they sitting like, there's no more space? Why? Because these outer layer is made up of callose. Okay, one layer called as callose, which is stopping them from separating. If they don't get separated, I'm not going to get individual pollen grains, but these people are together. Then what happens? Tapetum will tell, don't worry. Tapetum will tell, don't worry. Don't you take any tension. Okay. Praveen, you can ask your mother, dear. I think she'll be the right person for this. Yeah. So, what is going to happen, students, here? Here, the callose is going to make sure to keep all this microspore together in an attached pattern. So, these people will not be able to go anywhere. They'll be like stuck, full stuck. Okay. Then, tapetum will be like, don't you worry. Tapetum will release an enzyme called as callase enzyme. Okay, callase enzyme which will degrade, okay, which will degrade this callose, okay, which will degrade this callose and make sure this microspores are separated. Now I am going to get the separated version of it. Okay, I am going to get individual microspores. Clear or not? Clear? Any doubts in this? Individual microspores I got. Yes, ma'am, individual microspores you got. So these are all N, okay, N, haploid, because obviously meiosis happened, reductional division, haploid will be formed. Now I got this. Now each of the microspore is going to become what? Each of the microspore is going to become ultimately the pollen grain. So students, I'm just going to write this on the top, so we have more space to, write the rest of it. So I'm writing it here, microspore tetrad. Okay, tetrad, all are N, 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 N. Okay, I'm writing haploid. Now, this individually one microspore is going to ultimately form what, students? The beautiful pollen grain that you were waiting for so far. Okay, oh. Pollen grain. Now, pollen grain outside, what do we have? We have the exine. Okay, we have the exine. Okay, exine done, intine is there. Now what is going to happen? Two cells are going to be formed. Yes, pollen grain. All of this is going to form pollen grain. Okay, all of this is going to form pollen grain, but I'm not going to draw. Okay, but I'm not going to draw. So during this process, okay, here a mitosis is going to take place. Here a mitosis is going to take place to form a two-celled stage. What is two-cell stage? One vegetative cell, one generative cell. Clear or not? So now let's label this. Are you all ready to label this? Yes, very good. So the outer layer is going to be called exine. Inner layer is going to be called intine. And in between, ma'am, did you forget to continue, draw the full thing? No, students, because exine is a discontinuous layer. Why? Because it should allow the germination of the pollen tube. Exine is made up of sporopollen, which is one of the most resistant material. Imagine no space only is there. Pollen tube will not be able to emerge and come. It's no Bahubali, no? So pollen tube germination will not happen. So that exine is a discontinuous layer, and that space is called as the germ pore. Because here germination is going to happen of the pollen tube. That is the germ pore. Now if you look here, the bigger cell is going to be called the vegetative cell. I will just write VC, is that okay? Okay, VC, vegetative cell, and the smaller cell is going to be called the generative cell, which will have the male gamete. Clear? Exine discontinuous. Yes, perfect. Discontinuous. Okay. I'm going to check your attendance now. Let me see. Wait. Let me see your attendance and scold you people. Yes, quickly. Some confused people in the class. What, students? What am I asking? It's like I'm asking your people's kidney, liver, heart. Very painful, ma'am, to put that one like. If you're enjoying the class, put the like. If you're not enjoying the class, it's okay. Okay, if you're enjoying the class, you should definitely put. I know all of you are enjoying. So, is this clear? This microspore tetrad, they got separated because we made sure that the dehydration of this callose happened with the help of callase enzyme, which was secreted by the tapetum. Right, that is why I told you this purpose. I am going to tell here secretion of the callase enzyme. So, tapetum did the job and made sure that this callose is gone. So, now these people are all separated and happy. Okay, separated and happy. Clear or not? Okay, now this mitosis will happen, exine, intine, generative cell, vegetative cell. So, 2 cell stage form happened, mitosis happened. Now, going forward, remember, one more mitosis can happen when this should be a 3 cell stage. So, now this is a 2 cell stage. Yes or no? You see only 2 cell. Okay, 2 cell stage. But further also, one more mitosis can happen leading to a 3 celled stage. Okay, 3 celled stage, which you're going to study in your double fertilization. No, syngamy plus triple fusion, how you're going to get your, how did this triple fusion happen? Because there were 2 male gametes. So, that 2 male gamete formation is going to lead to the 3 cell stage. That time, one more mitosis will happen. Done, done, done? Yes or no? Okay, very good. Very good. Now, let's look at the diagram from the NCERT. So far good. Any student is facing any doubt, first time attending my class, speed is okay, everything is okay? Laugh, laugh. You people laugh. You will not like, but you laugh. That you people will do very nicely. Okay, look here. This is your anther filament. Clear? Anther is there. Now, when we cut this, okay, these are longitudinal grooves. Okay, you can see, small. Now, that is your line of dehiscence. Through that only, the anther is going to open and the pollen grains is going to get scattered. Okay, so you can see the thecas like this. See, the thecas are there. Like this. Inside that, you can see the pollen grains. Okay, this is the matured one. Now, this one's enlarged image, you will notice that there is a connective, epidermis, endothelium, sporogenous tissue, tapetum and middle layer. Everything you learned. Now, if you look at the larger picture, you will notice that epidermis is a single layer, endothelium is also a single layer, but middle layer is going to be having 2 to 3 layers. That means they are multi-layered. Okay. And students, you will see the microspore mother cell. Now, here tapetum, remember these people are going to have dense cytoplasm. They are going to have dense cytoplasm and most of the time they are multinucleated. They are what? They will have more than one nucleus. So, they are multinucleated. This question had come one of the years as a PYQ. Okay, it has come as a PYQ. So, just make a note on that in the diagram itself that tapetum will have dense cytoplasm and they are multinucleated. Okay, and now if you look at this, these are the pollen grains. Can you see? They are all happily running around that side, this side. Okay, space and all is there. They are happy because this pollen sac is formed and then pollen grain is formed. Now, remember one thing, when I am always stressing on pollen grain, you should understand that the pollen grain is a male gametophyte. What is it? It's a male gametophyte. It's not the male gamete. Okay. Pollen grain is a male gametophyte. That means male gamete is going to be inside this. Okay. It's a male gametophyte. That means the generative cell will have the male gamete. This, if you understand, you will be able to answer some questions. No, that pollen grain is the male gametophyte. Clear to all of you? I hope this is clear. Now, microsporogenesis is clear. No doubt in this. Let's go through 1, 2, 3 lines of NCERT as well. See, sporogenous tissue, okay, is capable of giving rise to the microspore tetrad. Microspore tetrad. Each one is a potential pollen or a microspore mother cell. That means a pollen mother cell or a microspore mother cell. The sporogenous tissue can, again, you call it as PMC or MMC. The process of formation, the definition, the process of formation of microspores from the pollen mother cell is called as microsporogenesis. Now, the microspores as they are formed are arranged in cluster of 4 cells. That means what? They are attached to each other, and that is why these people will be called as the microspore tetrad. Clear? Same thing only we learn, no? Yes. Now, as the anther matures and dehydrates, the microspore dissociates from each other. That means they were attached together. Callose when they started moving that side and developing into pollen grains. So here, remember students, here we need to do more microspores. We need to form more microspores, right? So why? Because imagine I am the anther, and if I am making pollen grains, let's say I am trying to put this pollen grains on that. Okay, stigma is there. I'm trying to put the pollen grains. Wind is coming, taking some pollen grains and going. One man is coming, he's dusting some pollens and going. Insect is coming, that fellow is taking some pollens and going. So it is not necessary that how many pollens I'm trying to focus on that to fall on the stigma, it will fall. So I need to make multiple, I need to make many so that there is chances of pollination or fertilization to happen. Remember that way. But when we learn in megasporogenesis, that we do not think about, because we know that, okay, pollen will fall, stigma is there, ovule is there, done. Here, pollen has to be formed in multiple combis. Same goes for human reproduction. Sperm is formed in millions. Okay, but how many ovums are formed? Only one ovum. So that is the difference. Understood? Yes. Yes, gametophyte obviously carries the gamete, right? Yes. Now, pollen grains, inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. So, if the anther does not dehiscent, then the pollen grains will be stuck inside only, right? So, this is very, very important. And for dehiscence, who is going to help? Your epidermis, endothelium and the middle layer is going to help. Okay, understood that? Yes. Clear? Yes, very good. Now, the vegetative cell. We are talking about the pollen grain, okay? The vegetative cell and generative cell was there. This vegetative cell, when you looked also, all of you noticed, no? Vegetative cell is bigger. Ma'am, why is this vegetative cell bigger? But you're telling that generative cell has the male gamete. That means what? Vegetative cell is nourishing, providing the nourishment. So look here, students. Vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. This can come as a question. They can give you different types of cells and they can ask you which is the one that has a large irregularly shaped nucleus, right? So it's a possible MCQ. It's a possible MCQ. Now, the generative cell, but on the other side is small and it floats in the cytoplasm of the vegetative cell. It has a spindle shaped with dense cytoplasm and a nucleus. So they are spindle shaped. When you saw me draw also, all of you noticed that I drew a structure like this. I drew something like this and I drew a spindle shaped nucleus. And here I drew something circular. So that means here they are telling that the spindle shaped nucleus is present in the generative cell. And it is smaller because vegetative cell is going to be the food reserve. Okay, vegetative cell is the food reserve. Done? Okay, yes, very good. Now, very, very important line. In over 60% of the angiosperm, pollen grains are shed at a 2-cell stage. Here, what are they talking about? Ma'am, they are telling that here in 60% of the angiosperm, 2-cell stage. That means 1 GC, 1 generative cell, 1 VC, that is 1 vegetative cell stage. 60% of the angiosperms will dehiscent. Okay, dehiscent. But rest of the 40%, let's not take 40% students because that's a very straightforward answer. So let's say approximately. The rest of them are going for a, let's approximate, okay, I'm putting approximately 40%, 30 to 40% of them are going to dehiscent in a 3-celled stage. 3-celled stage. What is 3-celled stage? Here we are going to have 1 VC, 2 GC. Okay, we are going to have 1 vice president, vice principal, vice counselor, and we are going to have 2 GC. That means we are talking about 2 male gametes. This question can come, so remember carefully. In the remaining species, the generator cell divides mitotically to give rise to 2 male gametes before pollen grains are shared, which will form a 3-celled stage. So generator cell is going to undergo division, and here it is going to lead to the formation of 2 male gametes. Clear? Yes, very good. 2 male gametes. These are all important lines of NCERT, so I have taken it as such and I have put it here. Okay. Now, pollen grain is clear, students? No doubt. But then I have some more to teach. So, pollen grain allergy and pollen grain, both we will do it in the same page. Okay. I think the next page is viability. Chick, 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 chick. Yes. So, we will do here only pollen grain. We have to write about the layers. Okay. We have to write about the layers. Shall we draw a very perfect pollen? Okay, yes. So this is your big pollen. Around the pollen, ma'am, we are going to draw beautiful exine, and students, exine will decide the pattern and the design on the pollen. All of you can open your NCERT and see they have given that different designs are there in the pollen. Okay, different designs are there in the pollen. No, who is going to decide? Exine is going to decide. So exine, when I talk about, you have to remember one thing. Exine is made up of sporopollenin, which is one of the hardest substance found. What do you know about sporopollenin? Tell me. Ma'am, sporopollenin is highly resistant, right? It is not going to, you know, by any ways, we have not found a way to, you know, break this fellow down. That is, you put alkali, okay, you do acid, you put alkali, you put heat, temperature. None of this can degrade this sporopollenin, which is one of the most resistant, okay, substance. Now, when I come into the inside, and this exine is discontinuous, no? Discontinuous, having a germ pore. Discontinuous, and this is the one and only germ pore, allowing the pollen grain to do germination, okay, germ pore. Now, students, inside layer is going to be called the intine, and intine is made up of what? It is made up of cellulose and pectin. Cellulose and pectin. This is not that hard, and this is continuous. Okay, pavam this fellow is. Okay, clear? High temperature, alkali, yes, resistant to everything. Okay, it is resistant to every single thing. So sad, Pradeep. Don't worry. Okay. Yes. Now, this is clear, no? Exine, intine, cellulose, pectin, sporopollenin. These are the questions that usually come. They'll ask you what material is intine made up of. So, you should write cellulose and pectin. They'll ask you what material exine is made up of. You should write sporopollenin. Now, coming back to pollen allergy. Students, I think we all know about pollen allergy. You know, what is this? So, pollen allergy is because when you are allergic to pollen and the pollen grains, okay, when pollen grains act as an allergen, when pollen grains act as an allergen, that means this is going to be the allergen and you are going to get allergy, okay, you are going to get allergy. Clear or not? What am I eating? I am drinking water. What and all you people need to know. Okay, so people usually have allergy to pollen dust, okay, to pollen dust, and because of that these person can suffer from asthma, okay, or any bronchi related issues. We have studied it in breathing and exchange as well. No, if bronchi is getting blocked, bronchioles are getting blocked, we will definitely get issues, okay. So, bronchial issues, all this can come, and as well as wheezing. Asthma is related to wheezing only. So here only you can write wheezing. Some people will have continuously sneezing, okay, if they are exposed to pollen grains. So before you give flower to someone, please ask them if they have pollen allergy. Otherwise, if you go with flowers and give, first thing they will do is they'll before taking the flowers only, they will start sneezing and they will run back home. So before you give flower to someone, always ask if maybe we are giving it out of consideration, or you want to enjoy, or you want to congratulate someone, but there are a lot of people who are allergic to pollen, okay? So you will go running with a bouquet of flower, telling that take the bouquet of flower, I bought it with all my pocket money. First what they will do, they will sneeze on you. Okay, so make sure that you ask a person before giving a bouquet that do you have any kind of allergy. Okay, clear? So that is what is called sneezing and wheezing. Yes. Now this is done. This is done. So pollen allergy is related to allergy towards the pollen dust, and many people are allergic, and because of it you will end up having asthma or your bronchial issues. Now one of the most common allergen is your parthenium. Parthenium, which is also called as carrot grass. Okay, carrot grass. And students, this is very common. You would have seen it everywhere around your home and all. How does it look like? It will look like a bouquet. Okay, it will look like a bouquet like this. Like this it will be. Wait. It will look something like this. Okay. No, I do not have allergy. You can give me a bouquet of flower. Okay, once you people crack need, you can come and give me a bouquet of flower. I do not have allergy. Okay, so parthenium, which is also called as the carrot grass, which is commonly seen everywhere. Okay, you just walk around today itself after class, Chuma, just go that's it, this and see, you will see parthenium, a carrot, grass. That is very, very allergic, and they also have a fragrance. Okay, they also have a fragrance. Okay, now students, pollen is considered to be rich in nutrients. Okay, pollen is rich in nutrients. Rich in nutrients. Since it is rich in nutrients, lot of people have started using pollen, okay, in the form of supplements. In the form of supplements. Like who, ma'am? Especially the ones who are athletes. Okay. So athletic people, okay, people who do this racing, all these people have started taking pollen as a supplement because these people are rich in nutrients. You can just imagine, insects are very attracted to them. We know that nectar is given, scent is given, all these are rewards. At the same time, pollen is also a reward given to an insect who is coming to collect the pollen. No, it's like a reward for it. So this pollen is rich in nutrients, and now people have started using it as supplements. It's not very common here, okay, right now, but then it is very common in other places, okay, like abroad. Here only you can see from England. So it is very common in England, as well as you can see pollen products are like your pollen, bee, then you get creams, you get jellies, all these are very available because these people are rich in nutrients. Okay, just that much you remember. Yeah, racehorse, athletes, okay, all these people, all the words have got jumbled up. Okay, these people are going to use it. Now, very, very important, viability. Viability question can come, okay. Yes. Okay. Now, viability. What is viability, ma'am? Imagine you are trying to log in, okay, to some app, and they will send you an OTP. That OTP will be valid for 5 minutes. With that time, you will immediately type the OTP, okay. But what do our parents do? When we call them to ask the OTP, they will ask you 100 questions why you need the OTP. By then, the 5 minutes time would have gone, right? So, OTP is valid only for 5 minutes. Same thing, students, if the pollen grain is coming. Okay, Anther has released the pollen grains and the pollen grains are coming. But how long is their expiration day? That means they are going to get expired in 30 minutes. They are going to be expired in 1 month. Or if I preserve it and store it, they might stay it for longer time. Like that, like just like how OTP has an expiry date, same way this pollen grains also has a viability. That means how long these people are going to be viable, how long these people are going to help in germination. That is a question. So viability means how long these people are going to help or be active for germination. Yes, very good. So it is their ability, okay, ability of pollen grains Okay, all these notes you'll get it on the Telegram channel. So quickly go and join the Telegram channel, Vedantu NEET English Telegram channel and Biology by Gopika. Okay, ability of the pollen grain to germinate on the stigma. Okay, on the stigma. Clear? This is the viability. Now, different people will have different viability, right? So here they have given that some cereals, okay, like your rice and wheat have a viability time of around 30 minutes. 30 minutes. So this can come as a question for you, right? A direct MCQ can be asked, right? Direct MCQ can be asked. Yes, thank you so much. Thank you for your compliments. Okay. Yes. So I'm going to write that. Please wait. But still, students, why haven't we reached the target? Yeah. People have become so conjuice. Huh? So conjuice. My students. Okay. Okay. Very good. Now, cereals, rice and wheat for 30 minutes. Then there are some members. Okay. Members of some families like I'm going to give you a trick for this. Rosole. What is it? Rosole. Rosole. Okay. RO is for Rosaceae family. SO is for Solanaceae family. Solanaceae family. LE for your legumes, no? Leguminaceae family. Okay, Leguminaceae family. So, Rosole, okay, Rosole is going to be Rosaceae, Solanaceae and Leguminaceae. These people have a viability for months. These people have viability for months. But please do understand, the viability also depends on factors like your temperature. It depends, viability also depends on temperature and humidity. Temperature and humidity. Artha aita, understood, arthama. I don't know in Telugu, students. You people should teach me Telugu. Yes. Clear? Okay. Cereals like rice and wheat has up to 30 minutes. Members like Rosole. I think it's a Hindi word. Okay. Rosole for months. Okay. Rosaceae, Solanaceae and Leguminaceae family will have it for months. Now, I am telling that this is too less. Yeah. 30 minutes. Seriously, 30 minutes is the time. What if this pollen grains come to me and I do not, transfer it, and what if, time we get waste. So, we are going to do something called as storing. Okay, we are going to store the pollen grains just like how you have your blood bank. Okay, store the pollen grains in what? In liquid nitrogen in minus 196 degrees Celsius in liquid nitrogen. Clear? Liquid nitrogen. Very oftenly asked question also. So, make a note of it. This is your entire story. This is your entire story. Microsporogenesis clear? Microsporogenesis clear? Everything so far is clear to all of you? I hope there's no doubt. Yeah, it's here. Okay, tell me. So we have done microsporogenesis. This is the entire microsporogenesis for you. Meiosis, mitosis. Again, one more mitosis will happen here. This is the structure. Okay, bilobe, dithecous and tetrasporangiate and tetragonal. Okay, I will add that also here for you, all of you. So here we learnt it. Okay, tetragonal. Sporangiate. Okay, this is all the things we have learned. Okay, learned. Now, question. First question for all of you. How many meiotic and mitotic divisions occur during the development of male gametophyte from the mother cells? Mother cell. Lucky, thank you so much. Tell me. Dileep, the class will go till 12 o'clock. Both the classes. Students, what do you expect? Yeah, the chapter is so big. How can we not do justice for the chapter? Answer this question, all of you, quickly in the chat box. No, it's okay. Dear, this class will go, I think my class will go up to 8:30 and then sir's class will start. Please answer this quickly now. Yes, what's the answer? Students, it's only about microsporogenesis. Don't tell for, sorry, it's only for microsporogenesis, not for megasporogenesis. Okay, got it. Yes, very good. Some of you are getting the right answer. Yes, perfect. Janani, good explanation. Okay, now some of you, very good answer. Okay, so let's decode this for those students who did not know. We know that one meiosis definitely happens. Ma'am, when does this one meiosis happen? If you ask me, it is from MMC or PMC to your microspore tetrad. To your microspore tetrad. Then they are telling that two mitosis happens. How? From your microspore tetrad, you will, let's say microspore to your pollen grain, that is your 2-cell stage. 2-cell stage, one mitosis happen. Okay, one mitosis happen. So if it is a 2-cell stage, then we have one meiosis and one mitosis. Okay, if you have one more, that is the 3-cell stage. Okay, you go for one more 3-celled stage, then the generative cell will undergo one more mitosis to form 2 male gametes. Alright, second mitosis. So here it is going to be one meiosis and 2 mitosis. Clear to all of you? This is up to 3-celled stage. Okay, up to 3-cell stage. Question is clear? What happened, Kanmani? Oh, okay. Yes. Done. Answer is clear to all of you. Any confusion? One meiosis, 2 mitosis if it is up to 3-cell stage. One meiosis, 1 mitosis up to 2-cell stage. No? Because here just now you read in NCERT that your generative cell will undergo one more mitosis to form the 2 male gametes. Okay, good question. Now we are starting the next topic, that is going to be your pistil, carpel and megasporogenesis. After megasporogenesis, we have the post-fertilization events, then the chapter is over. Right, chapter is very going to be tuck. So all of you listen carefully, don't lose your motivation. We are going to make sure that we are going to answer all the questions as well. Okay, chalo, let's do it. Okay, I know that a lot of NEET Express students wanted to attend this class, but it is very sad because your classes are happening there, but still you're not wasting time, so it is fine. Okay, and this class will be always there online. You can come and check it out anytime. Okay, going forward, students, pistil and carpel, these two people are who? Ma'am, pistil and carpel are the units of gynoecium or gynoecium. There, who are the units? Ma'am, filament anthers together called as stamen were the units of your androecium. Okay, units of your androecium. That is why we studied the structure of a stamen and structure of anther separately. Here we are telling that the units of gynoecium is going to be pistil or carpel. Now remember, what is there in your entire pistil or carpel? We have stigma, which is the landing place for the pollen grain, a long stalk-like structure called the style, which is going to connect the stigma to the ovary. Okay, and we have a big swollen portion called the ovary. Inside the ovary, what do you have? You have a small placenta. Okay, like this, one small placenta will be there. On this placenta, there will be one ovule. Okay, on this placenta, there will be one ovule. One or many ovules depending on the structure, okay, depending on the plant. Now, this portion is called the stigma. This is where your pollen grains are going to land. This is the long structure which is connecting the stigma to the ovary style, and this is your ovary. And inside this, what you're seeing is the ovarian locule. Okay, again like a small cavity, and this is going to be your ovule, and this is going to be placenta, and that's how you study placentation, no? Marginal placentation, axial placentation, depending on the placenta and the ovary, how they are attached. Okay, how they are attached. Done, done, done. This is your entire carpel or pistil. Students, there we did not write stamen anywhere, no? We forgot our pavam stamen in one word. Okay, so we will just write here, okay? Okay, stamen is the unit of androecium. Yes. Okay, clear? Now, go here, it is carpel and pistil. Now, this carpel, students, this carpel can be fused or separate, okay, depending on the carpel. So, if the carpel, okay, if the carpel is many carpels are there. Many carpels are there. We will call it what? All of you tell me in the chat box. Ma'am, we will call it multicarpellary. Multicarpellary. Yes, ma'am, definitely we will call it multicarpellary. If they are multicarpellary, then it is understood that they can neither be fused or they can be separate. Yes or no? They neither can be fused. So, if they are fused, what is this term for it? Syncarpus. Syncarpus. Okay, fused. Syncarpus. If they are free, apocarpus. Apocarpus. Clear? So, if there are many carpels, they are called as multicarpellary, and if they are fused, they are called as syncarpus. If they are free, we call it apocarpus. Okay, apocarpus. So, like that only, fused and single. Okay, now fused and free. So, many carpels, multicarpellary, otherwise there will be single, no? Only one. Now, depending on which is syncarpus and apocarpus, you have two examples to study. All of you tell me what is the trick to remember it. Let me see if my students remember. Tell me, there is a trick to remember. I will not tell the trick, my students will tell the trick. Quickly tell me in the chat box, what is the trick? Yes, lot of students have joined now. Quickly like the video so that you know Biology 360 Club will be a very successful club. Okay, I'm planning to open one club after that. Okay, Biology 360 Club. All of you, I'll take tuitions there. Okay, me and Baswa sir. With one stick we will stand and we will be like, come on, learn, learn, learn. Okay, yes. Ma'am, MSP. Okay, so here, M-A-M. Basically, I did not use any trick, students. I just added the words together. Okay, M-S-P. Now, this is, the examples are important. M for multi. Okay, multi. A for apocarpus. That means free. M for Michelia. Okay, example. This is the EG. That is example. Okay. Again, M, multicarpellary. S, syncarpus. P, Papaver somniferum. This is your example. Ma'am, MSP. Okay, that's the example. How will you remember? Ma'am, MSP. So, ma'am stands for apocarpus. Ma'am is free. Full time free. That's why, you people keep telling, ma'am, take revision of that, ma'am, take revision of this. But we will not attend the class. You do revision. I will sit here. No, so ma'am is always free. MSP is syncarpus. Okay, they are fused. That is going to be Papaver somniferum. Clear or not? Target reach. Wow, wow, wow. God's grace. Okay, God's grace. Papaver somniferum we will study in your drug abuse. Okay, drug abuse. Okay, done. This is clear. This is the general idea of it. Now we have to study megasporogenesis, ovule and embryo sac. That also we will study in very like aram se we'll study. Okay, aram se. Yes. Let's go. So this is the structure given. So if the structure is given, you should be in a position to identify it. They can ask you image based question. Okay, they can give you an image and they can ask you questions. Okay, so look here. This is your ovary, thalamus. Again, style and stigma. Now, you would have seen this in hibiscus, no? Stigma, you would have seen the pink, reddish pink color. Now, here you see stigma is fused. Okay, stigma here, all of the carpels are coming together, no? So, here you can see that these are syncarpus. Here you see these people are free. Okay, these people are free. This is apocarpus. Okay, apocarpus. So, here you can see example, Michelia, and here it is Papaver. Okay, Papaver. So, syncarpus, pistil of Papaver, and apocarpus, gynoecium of Michelia. And this is your ovule, which we are going to study now. Okay, which we are going to study now. So, diagram is clear. What is this? Fused. Look at it. Doesn't it look fused? Yes, ma'am, it looks fused. This people looks free, ma'am. Okay, so this is free, this is fused. So when you're identifying it, they will expect the example from you or they will expect the condition. Is it syn or apocarpus? Okay, is it syn or apocarpus? Chalo, let's do megasporogenesis. Okay, what is megasporogenesis? Microsporogenesis is formation of microspores from the microspore mother cell or the pollen mother cell. Okay, ma'am, understood. Megasporogenesis. Okay, I will just replace microspore with megaspore. So, it is the formation of megaspores from megaspore mother cell. Over. Right? Over. So, now, look here. This is going to be a megasporogenesis. So, megasporogenesis is going to be formation of megaspore from megaspore mother cell, right? Megaspore mother cell, that is MMC. Okay, MMC. Yes. Who is Santa? On May 5th, who gives you question paper? That is going to be your Santa. Okay, if the question paper is easy, you will be like, oh, thank God. After that, question paper is difficult, you will scold the Santa telling that this is not the gift I expected. Okay, your Santa is going to be your question paper this year. Right? It's students over. This year is over. 24th. Tomorrow is Christmas. 5 days New Year. This year is over. All this while what were you telling? Ma'am, 2024 is still there. Ma'am, 2025 I will write the exam. Exam is now near. Neck to neck it is going to come. Okay, so start studying now at least. Formation of megaspore from megaspore mother cell is called as megasporogenesis. Now, before I go to megasporogenesis, we have to draw our entire stigma and style and everything once again. Okay, yes. So, let's draw. Let's draw, no? I will draw. You people will chumma sit and laugh and smile and giggle. Okay, this is the structure. Okay, yes. Placenta I am drawing. Okay, inside this what is going to be there? One ovule is going to be there. Okay, one ovule is going to be there, right? Thank you, Madhavati. Yes. Now, okay, look here. Now here, this is done. Now, why I drew this, you will understand. Now, I am going to only magnify this. Okay, I am going to just magnify this and draw it here. Okay, draw it here. Let's say this is your placenta. Okay, this is your placenta. For the students to understand how is the placement of your ovule. And now when we draw the ovule, students, in the middle, okay, this ovule, no, in the middle there is going to be a set of cells which are called as a nucellus. What is it called? Nucellus. Okay, clear? So these cells are called the nucellus cells. Yeah, nucellus. Now to protect this nucellus, there is going to be two people who are going to be like, one is the younger brother, one is the elder brother. Who are they? One integument, inner integument, okay, inner integument and outer integument. Okay, outer integument. See, inner integument and outer integument is there, okay. Now from here, there is going to be one attachment which is going to be called your funicle. Okay, funicle, which is going to attach to this. Okay, which is going to attach to this. I will just draw a placenta a little more big because my funicle is outside the light. Okay, yes. So this is your ovule for you. This is called as an anatropous ovule. What ovule? Anatropous ovule. Yes. Look here. Now look here, all of you. Yes. Now this structure is going to teach you the entire megasporogenesis for you. Now what is going to happen, students? This first we will label this. Okay, what is this? Inner integument. Okay, inner integument. These people are going to later modify to form your testa and tegmen. Okay, testa and tegmen, your seed coats. Okay, this is going to be the outer integument. Outer integument. Now, here, this portion. Okay, there's one stalk-like structure. Now, what is that going to be called? Funicle. This is going to be called the funicle. And the portion where the ovary attaches to the funicle is going to be called the hilum. So, let's draw this portion. Okay, this whole portion is the hilum. Okay, this is going to be the hilum where the funicle and the ovary attach. Okay, that is the hilum. Now, this portion is going to be called the chalazal end. Chalazal end. And the portion where you see a small opening. Okay, where you see a small opening, this is going to be called, God, one second. This is going to be called the micropylar end. Micropylar end. Now, exactly what happens during megasporogenesis? Okay, what happens exactly during your megasporogenesis? That is, these cells are called as your nucellus cells. What cells? Nucellus cells. So, one of the nucellus cell will be like, don't worry, I'm going to take up this big responsibility of becoming the megaspore mother cell. So, this nucellus cell, because here students, we do not have the need for making millions of copies. We do not have the need for making multiple copies. Why? Because here, we know that only one megaspore is going to become functional, that is going to get fused. But in pollen grains, that was not the situation. Many sporogenous tissue came telling that we are going to become the megaspore mother cell, we are going to become the pollen mother cell and undergo meiosis. So, here, only one person, one nucellus is going to be like, I am coming. Okay, I will take up the responsibility of becoming the MMC. So, this nucellus one cell is going to become the megaspore mother cell. Megaspore mother cell. Clear to all of you? Clear to all of you? Now, are you ready to study megasporogenesis? So, this entire thing, okay, this entire thing only we have drawn here. Okay, we have drawn here. This entire thing we have drawn here. The entire ovule. Fine. Now, let's study megasporogenesis. Now, what exactly is going to happen? Nucellus was like, I am ready to come. So, the nucellus is going to become the MMC. MMC. MMC is going to undergo meiosis, obviously. And MMC is going to form what? It is going to form megaspore tetrad. It is going to form megaspore tetrad. Megaspore tetrad. Correct? Megaspore tetrad is going to form. Now, Okay. Thank you, Rasna. Now, this megaspore tetrad, no students, what is going to happen? 3 of them are going to degenerate. 3 of them are going to degenerate, and the megaspore near the chalazal end, the megaspore near the chalazal end is going to do the development. So, this is called as monosporic development. This is called as monosporic development. That means mono means what? One person, only one fellow decided to develop. So, this kind of development is called monosporic development. 3 of them are going to degenerate, only one is going to become active and continue the process. Okay, and the one who is going to be active is going to be near the chalazal end. So, let's say, okay, ma'am, understood. Now, this fellow, this monosporic development is going to happen. So, we got a single nucleated. This fellow I am taking, okay, out. So, this the one we got was a 1 nucleated stage. Okay, again it will undergo division. It will form 2 nucleated stage. Okay, 2 nucleated stage. From 2, it will become 4. Okay, 4. From 4 to 8. Okay, 4 to 8. Finally, we will get a 8 nucleated stage. Correct? This is your entire megasporogenesis for you. That means, first we got a single nucleated stage. Okay, this is 1. Okay, this we got. This is the 3rd nucleated stage we got. So, I will write 4 cell stage. Okay, this is 4 cell, and finally we got 8 nucleated stage. Okay, 8 nucleated stage. Now, this 8 nucleated stage is going to finally form your embryo sac. Now, students, remember, 1 meiosis is going to happen, but 3 mitosis is going to happen. From this to this, this to this, this to this. So, this is going to lead to 3 mitotic division until it forms an 8 nucleated stage. Clear to all of you? Yes or no? Very good. Very good. Okay? Yes. So, MMC, that is your megaspore mother cell, underwent meiosis, formed a megaspore tetrad, out of which 3 got degenerated. Okay? Out of which 3 degenerated. Only 1 became functional, and that kind of development is called as monosporic development. Okay? Where only 1 got functional. Okay, 1 became functional. Monosporic development. From that single nucleated stage, we made double nucleated, 2 nucleated stage, 4 nucleated cell, and 8 nucleated stage. Okay, 4 nucleated. Okay, 4 nucleated to 8 nucleated. So, during this 3 mitotic division happens. Okay, so 1 meiosis, 3 mitosis happens in megasporogenesis. If this is clear, can we proceed? Okay, now we have to form the embryo sac. So, now students, what do we have? What do we have? Ma'am, we have an 8 nucleated stage. This is what we have. Correct? Now, from this, what exactly is going to happen? Everything is going to move towards the pole. Cell formation will happen near them, and they are going to form the embryo sac. So, we will draw the embryo sac. Okay? We will draw the embryo sac. See, this is the embryo sac. Now, look here. You have three antipodal cells towards the chalazal end, okay, towards the chalazal end. Towards the chalazal end, you have what? You have the antipodals. These people are not going to come for any disturbance. They're just going to be there. Okay, they're just going to be there. Okay, now towards the this pole. Okay, towards this pole, what is going to happen, students? You will have two synergids. Okay, you will have two synergids, and you will have one egg cell, and together we can call this the egg apparatus. We can call this people together the Egg apparatus. Very, very good. All of you are answering. Very nice. So these people are going to be called the synergids, and this is going to be called the egg, and together these people are going to be called the egg apparatus. Okay, egg apparatus. Clear? Now, two people will go to the center, saying that I want to dance in the center, I want to be in the center. So, two people will go to the center, and these are going to be called as the polar nuclei. Polar nuclei. So, during this time, remember students, there is going to be one more cell formation, that is going to be the center cell. Okay, like this. Okay, one center cell formation will happen. Perfectly clear to all of you? Center cell. This is your entire embryo sac for you. This is your entire embryo sac for you. Now, one more important thing is, students, the synergids, no, they're not chuma going to sit. They will have a finger-like structure called the filiform apparatus. What will they have? They will have a finger-like projections called the filiform apparatus, who are going to help in leading the pollen tube. They are going to help in leading the pollen tube. Later when pollen tube is coming, no, that time these people will be like, come this way, please come. Okay, so they are going to lead the pollen tube, show the way for the pollen tube to reach the egg. Filiform apparatus. Clear or not? Thank you, Pradeep. Yes, filiform apparatus. Very good. Guide the pollen tube. So, is this clear? All this is clear? This is going to lead to. So, students, all of this is going to be 1, 2, 3, 4, 5, 6. This is going to be considered as 1. So, this will give us, this embryo sac is going to be 7-celled, 8 nucleated. 7-celled, 8 nucleated embryo sac we got. 7-celled, 8 nucleated embryo sac is done. Fine. Embryo sac formation also is done. Anatropous ovule is done. Why is it called anatropous ovule? Because the funicle, the chalazal end and the micropylar end does not come in one line. Okay, one line. Clear or not? Nitrosonium, don't worry. Okay, so the funicle, micropylar and chalazal end is not coming in a straight line. That's why this is called as an anatropous ovule. Okay, now embryo sac also is done. Now we are going further to make sure that fertilization happens, because now our job is done. Egg is formed, their male gamete is formed. Now we have to do fertilization, right? Now we have to do fertilization, right? So here you see the diagram, students, what I just drew, no? Same thing here. Megaspore mother cell is there that will undergo meiosis and it will form megaspore dyad, megaspore tetrad. Okay, and going further, you will see 2 nucleated stage, 4, 8 nucleated stage, finally arranging themselves to forming the embryo sac. Okay, you can see antipodals towards the chalazal. You can remember it as AC. Okay, and here you have polar nuclei, central cell, egg, synergids, this together called egg apparatus, and you have filiform apparatus. Clear? Filiform apparatus job you will understand. Okay, you will understand later when we are studying about double fertilization. Ready? Now we are going to do what? Fertilization. Okay, we have to make them meet each other. Only forming will not help, students. Meeting is important. Forming, say what you will get. Forming and keeping them in the fridge, it will work. No, ma'am. We have to make sure that fertilization happens, right? Yes, so the functional megaspore is going to be from the chalazal end, okay, because functional one is going to be from the micropylar end. So consider chalazal end not very active person, okay. Chalazal end is just going to have your antipodals and going to be happy. That's all. Clear or not? Likes have to increase quickly, like yes Janani, yes Janani. How many times you will ask? I'm answering. Okay, now let's do pollination. So students, pollination definition is the same that what you are studying from EVS, no? That is transfer of pollen of pollen from anther, which is the male part, to the stigma, which is the female part. Now, pollination basically is two types. Pollination is basically two types. Self-pollination and cross-pollination. Right? This is what we have studied in EVS. Self-pollination and cross-pollination. But as you go in the further classes, you will learn that advance happy Christmas children's. Whose children? Who told you you can become a father of this many children's? Came off Santa Claus from nowhere. You cannot be. Okay? First you sit in class, change your name and come back to the class. I know who you are. Children. My children. I wish you. Okay? Yes, Supriya. Depending on you people. Okay, so fast he came off. Reindeer, no one was there. He came by himself. He's like, okay, class is happening. I will go make this many, okay, 70, 80 students, my children, and I will wish them. Come tomorrow for Christmas. Okay, Diksha ma'am's class is there. Come tomorrow and you wish. But first of all, who made you Santa Claus? I want to ask. Show the certificate, then you go forward. Okay, now jokes apart, self-pollination. Students, as you went further in your class, no, information started adding on, no? So, same way here. Self-pollination is going to be called autogamy and geitonogamy. Okay? Geitonogamy, everyone pronounces it little different. Okay? Little different. Okay. Now, cross-pollination, you have only xenogamy. Okay? Xenogamy. Now, with the help of a diagram, I'll teach you the whole concept. You do not have to worry anything. Okay. Now, let's start autogamy. Students, from the word auto means what? Now, we put auto mode. Okay, auto mode in our phone. No, that means what? Light will be arranged, contrast will be arranged, exposure will be arranged. Everything will be arranged by the phone. You do not have to adjust the light, bring down the light. That means auto mode, done everything by themselves. So, autogamy means if this is the, okay, this is the stigma. Okay. This is the anther. The pollen has gone from this anther to this stigma. This is called as self-pollination. Same plant, self-pollination. Okay, clear? Okay. Now, what happens? This is autogamy. That means same stigma, same anther. From the same flower, anther and stigma has gone and done it. Okay. Now, what is geitonogamy, ma'am? I have not heard about geitonogamy. Let's say these are two flowers. Okay. Okay, this is a plant. This is one flower. Students, diagrams are the best way to study biology. Don't think that, Ayo, why ma'am is drawing diagrams, I can study without diagrams. Diagrams will let you remember the concept for a longer time. Okay, these are the flowers on the same plant. Can you see? These are the flowers on the same plant. Now, let's say this is a male, okay, this is a female, or let's say from this anther, this is the anther. From this anther, the pollen grains has come on this stigma. Okay, pollen grains has come on this stigma. This is called as geitonogamy. That means flower is the same. The plant is the same, but different flowers. This is called as geitonogamy. That means different flowers, but same plant. Here also, students, don't expect a lot of genetic variation. In these two situations also, genetic variation is going to be very invariable. Okay, genetic variation is going to be almost invariable. That means it's not going to make a big difference. Okay, even in geitonogamy. Clear or not? Yes. Now, moving forward, what is xenogamy? Ma'am, xenogamy, I know that, let's say this is one flower. Okay, this is one. And another flower is there. Let's say from here to here, transfer has happened with the help of an agent. With the help of an agent. This is called as cross-pollination or xenogamy. Is this clear to all of you? Yes? Clear to all of you? Enough. Stop going behind the Santa and learn something. Okay? Otherwise, on May 5th, you have to pray for Santa only to come down and write the exam. Okay? This is different. This is different. Same species. Okay? You cannot expect rose and some other sunflower to have a thing. The stigma will reject it. Stigma knows the species. Okay? What happened? Oh, sorry. Same plant. So sorry. Same plant. Got it? Same, different flower, same plant. Here it is. Different flowers, different plant. Okay. Yes. Done? May 4th this time. May 4th, first Sunday. The whole pollination is clear. Autogamy, geitonogamy, xenogamy. Now, we have to learn kinds of pollination. That means, who are going to, your autogamy, xenogamy is done. Okay, now we are going to learn under kinds. Here we are going to learn agents, students. Okay, agents as plus kinds. Okay, little about kinds. Okay, so first we will start with autogamies. First, autogamy. We understood what is autogamy, ma'am, simply called as self-pollination. But under autogamy, they have mentioned about clistogamous flowers. All of you tell me what is clistogamous flowers? Clistogamous flower. What is it? Clistogamous flowers are the flowers who will not open. Okay, they were not going to open. They are always going to be closed. Like this. They are always going to be closed. Like this. On the same plant, there could be clistogamous as well as chasmogamous flower, but this clistogamous flowers are the ones who are not going to open. Okay? Not open. Okay? So, because they are not going to open, you only tell me what is the chance? What is the chance for cross-pollination to happen? Almost 0 or nil. They are never ready to open. They are not going to open. So, they are going to not open, and because of it, these people are going to always go for autogamy. They are always going to go for autogamy, and because of which, students, inbreeding depression is going to happen. Okay, what is going to happen? Inbreeding depression. Inbreeding depression is going to happen. Now, what is chasmogamous? Okay, what is chasmogamous? So, in autogamy itself, you have one more type, okay, which is called as chasmogamous flowers. These flowers are going to be normal flowers with exposed anthers. They are going to be open, okay, and they'll have exposed stigma as well as anthers. Okay, anthers, no problem here. So, these people will be ready for cross-pollination. Okay, these people will be ready for cross-pollination. Now, you have to remember the examples. That is going to be COVID, no? COVID, you remember cove. Okay, remember cove. C for Commelina, O for oxalis, V for viola. Clear? These are the examples. Cove, cove. Really? Was that Shreyas sir? I don't think so. Shreyas sir will become Santa Claus, right? Yeah. Yes. Shreyas sir will give you people some gift soon. Yes or no? Shreyas sir will give you people some good. Yeah, exactly. Correct. Antipodals. Okay. So, Commelina, oxalis and viola. Yes, clistogamous are, okay, clistogamous and chasmogamous will be present in the same plant. Okay, same plant it will be present. If this is the plant, you will notice that there are some flowers which are open. Okay, which are open, and you will notice that there are some flowers which are closed like this. Okay, both will be present on the plant. Clear or not? Cove is the example, Comedina, Oxalis and Voila. That will not open, so these people will lead to autogamy and this will lead to inbreeding depression. That means depression from self-pollination, continuously doing self-pollination, and this continuous self-pollination will lead to what? Problems. That is, inbreeding depression will happen. No variation, so obviously inbreeding depression will happen. Okay, and chasmogamous flowers are normal flowers which will have open exposed anthers or feathery stigma. No problem, normal flowers they will have. Okay, clear? Clear or not? Okay, now students, will finish this pollination, then I'll give you a cutie break, because right now it's in a flow. I do not want to break the flow. Okay, I do not want to break the flow. Yes, even me, 100 and 200% it's not, sir, but I'm telling you, okay, I'm telling you that sir will give you a gift. Okay, now students, here itself, we will write two more topics, okay, two more things. I'll write it here only. The agents of pollination, okay, agents of pollination. Who are going to help? There should be some people, no? Now, cross-pollination has to happen, agents should be there. Agents can be biotic and abiotic. Biotic and abiotic. Under biotic, you are only going to study insect. Okay, you are only going to study insect. Abiotic, you are going to study wind and you are going to study water. Okay, in that wind is the most common. Okay, wind is most common and biotic also is most common. So, after biotic only abiotic will come. Okay, abiotic will come. Clear or not? I hope no doubts in this. Biotic, abiotic. After biotic only abiotic, that too wind will come. Okay. Yes, very good. So, we call it entomophily. Okay. Insect is called entomophily. Then you have hydrophily and anemophily. Okay. Wind is anemophily, hydrophily, entomophily. If you cannot remember, please don't break your head, but just understand. Okay. Yes. Yes. Nandu, just skip it. Okay, if ads are coming, if you tell me what will I do, you should only skip it, no dear, you skip it. Okay, yes, biotic, abiotic and wind, done, done, done. So, these are the agents. Now, insect, remember that insect pollinating flowers are going to be different, okay, are going to be different. That means the flowers have to attract the insect. Imagine you are an insect, okay. How many ever colors are used on the board, you people will still tell, ma'am, can you draw this diagram also? You people are not attracted, so you just understand, okay. So, here, what is going to happen? If insect is coming from a higher distance, it's flying and coming, it will be like, butterfly, butterfly, where are you going? It has to go to an attractive insect. It has to go to an attractive flower. So, for that, insect pollinating flowers will be different. Wind and water pollinating flowers will be different. Correct? Wind and water pollinating flowers will be different. So, let's see. Students, after we finish pollination, I'll give you a small break. Okay? Small break. So, till that, please be patient. I'm also standing here and teaching. So, this is going in a flow. Okay, now if we break the flow and come back, you will also not feel interested in the topic. So, that's why I don't want to give a break. So, let's continue. Okay. Yes. Pollination by wind is more common amongst abiotic pollination. Okay. Wind pollination also requires that pollen grains are light and non-sticky. Imagine I want to take this tissue. Okay. I am taking this tissue and I am doing, okay, I am doing this. Now, the tissue is lightweight and it is not sticking to my hand. So, it will fly. It flew. Okay, it flew and I caught. But imagine that, imagine that we are having something which is sticky and heavy. It will not be able to fly. So, we expect that if you are going to go for wind pollination, pollen grains have to be light and non-sticky so that they can be transported in the wind currents. They often possess well-exposed stamen. Why well-exposed stamen? So that the pollen grains can be easily taken by the wind. So, they will have a well-exposed stamen. Whatever words I have highlighted and I have understood, sorry, I have underlined, you are supposed to underline in your textbook. Okay? Clear or not? Okay. Now, so that pollens are easily, you can go to Shreyas sir's live later. First you attend class now. Okay, class is important. Finish the class, then you go to the live. Okay, live no, later you can watch the recorded lecture as well. It is not a lecture, it is to inform you some things. Okay, so that the pollens are easily dispersed into the wind currents and large, often feathery stigma also is going to be there. Okay, why? so that they can easily trap the pollen that is coming in the air. Okay, pollen all will be dancing and coming. The stigma should be feathery so that they can hold it, right? That is also done. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed in an inflorescence, because wind is not going to see that is it attractive or not. Okay, is it attractive or not? But wind is going to make sure the pollen grains which are going to be easily transported and the stigma who is going to easily attract it. Pollination will happen easily. Okay, so here there will be inflorescence. A familiar example is corn cob, that is the tassels that you see is nothing but the stigma and the style which wave in the wind to trap the pollen grains. One question has come, what is the use of tassels? You people tell me what is the answer. What is the use of tassels? Tell me. Yes, tell me the answer. It's okay, Nandu, you don't need the book. You just study from me. Okay, if you don't have, just write it in your lecture notes. Students, here the question is going to be, what is the tassels of corn cob going to be used to trap the pollen grains? See, here it is very clearly given. No, many students have written to attract the insects. How can it attract the insects? Wind pollination does not have to attract the insect. So here the answer is going to be to trap the pollen grains. Wind pollination is quite common in monocots. That means mainly your grasses. So this is how your wind pollinated. You can see feathery stigma is going to be there so that it can easily attract the pollen grains. Clear or not? Points are clear. So they will be light and feathery. The stigma is going to be feathery. Pollen grains are going to be light and non-sticky, and the anther is going to be exposed. And the wind pollination, you can write the tassels of corn cob, which is going to be a mix of style and stigma together, which is going to trap the pollen grains. Clear? Okay. Yes, clear. Now, pollination by water. It is already quite rare in flowering plants and it's limited to 30 genera. Okay, only 30 of them, that's all. Why? Because most of the water pollinate, like water living, you know, the plants which are living in water also, they also prefer wind and insect pollination. That means what they will do when they are ready for pollination, no, they will just come above the water and they will tell that, okay, take the pollen and go put the stigma. They will expect the wind or insect to pollinate them. That means most of the plants which are underwater, which are in water, also does not do water pollination. Clear or not? Clear or not? Now moving forward, mostly monocots. As against this, you would recall that water is a regular mode of transport for the male gametes among the lower plants, such as your algae, bryophytes, pteridophytes. Students, that is why we tell, no, to complete their life cycle, these people will need water, and that is also one of the reasons why bryophyte is called amphibians of the plant kingdom. Why? Because it needs both land and water to complete its life cycle. So even though these people are dependent on water, still not much plants are going to do water pollination. Okay. Now, it is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transport of male gametes for fertilization. Clear? Yes. Now, Vallisneria and Hydrilla is an example, and Zostera, which is a seagrass. Okay. Vallisneria, I think many of you would have seen many questions while practicing, and even sea grasses such as Zostera. Students, you people are not going to get break. That means, like, you people have to do, you have to solve some quiz. Okay, you have to solve some quiz. Yes or no? Okay, moving forward. Now, let's see what is exactly wind pollination. Water pollination. Not all aquatic plants use water for pollination. In majority of the aquatic plants like water hyacinth and water lily, the flower emerges above the level of water and are pollinated by insects or wind. That's what I told you. Even though they are underwater, even though you people are getting all the resources on Vedantu NEET English, still you people don't study, you know, same way. Even though they are in water, they still want insect and wind pollination. Okay, some people are not studying, and I know that. Okay, I got so many messages on Instagram, ma'am, 3 months can I crack NEET? Students, you think that easy NEET is? Then every nook and corner doctor will be sitting. No, there also one doctor, here also one doctor, I will be a doctor, you, every person in this chat box will be a doctor. No. So it's not that easy. Please understand. Stop searching that 3 months can I crack NEET, 2 months can I crack NEET, 1 month can I crack NEET, 1 week can I crack NEET. 1 week to definitely you can crack NEET. You take the NEET, you crack it. Okay, write it on a brick of piece, crack it and you make it into 2 pieces. More than that you cannot do anything. Students, please don't take the exam very lightly. It has become one of the competitive exams where, you know, lakhs of students are writing and lakhs of students are preparing. And you know what is happening in Kota, you know what is happening in different places, okay? Everyone are like, you know, under so much pressure. So when you are in a medium like this, you might not feel that much of pressure. But you should understand that when we are giving you resources, this Biology 360 Club also was done only on that basis. Otherwise, we could have just done normal classes. No one is going to ask us. But we wanted to do something different. Like make sure that students sit with us for 5 to 6 hours, write notes. We also write notes. It should be like they should feel like they're sitting in an offline class and teacher is, you know, continuously giving them, you know, lecturing and giving them knowledge. That is why we started this club. So don't disappoint us after one class. Next class it'll be like, oh, later I'll watch it in 2x. Okay, I cannot watch it live. Don't do all that. It disappoints us and then we will stop. And then you will again ask, ma'am, why did you stop? Okay, yes. Clear or not? Many students have asked, I don't, like their names also I don't know. Okay, they have asked me, ma'am, please make a video how I can crack NEET in 1 month. Okay. How? Even I don't know. Yes, now look here. The flowers emerge above the level of water and it is pollinated by insect or wind. This is the reason why water pollination is not very common. Now, in Vallisneria, very important, the female flower will reach to the surface of water. Look here. Here what is going to happen, students? This is the stigma. You can see the female flower will reach to the surface of water by the long stalk and the male flower or the pollen grains will be released in the surface of water. Released in the surface of water. My Instagram ID is Gopika Biology. Okay, Gopika Biology. My Telegram is Biology by Gopika. Okay, yes. They are carried passively by the water current. Some of them eventually reach the female flower. It is not even like, you know, we cannot even tell 100% will happen. Now imagine I am the female flower. I have a long stalk. I came above the water. Why did I come above the water? So that the pollen grain which is coming with the help of water current, I can fuse, like fertilization can happen. But students, it's not necessary. You know, the water currents can be at any direction. The pollens can be lost. Okay. So they are carried passively by the water current. Some of them will eventually reach the female flower and the stigma. In other group of water pollinated plants like your sea grasses, okay, sea grasses, female flower remains submerged in the water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most water pollination, what happens? The pollen grain will have a mucilaginous covering so that it is protected from wetting. Because if it is wet, it will go down. Right? Yes. Clear? Yes. Everything will be like we are trying to do as much as, you know, combined classes we will be doing, because in 11 there are very less units which are connected where Basar sir and I can take. So that's why that will be individual. So students, here you will remember that the stigma, here it will come up. So the pollen grain will be here. It has to reach the stigma. In second case, like your Zostera, what is going to happen? Stigma is going to be underwater. This is the water. Okay, pollen grain also is going to be underwater. Okay, here it is going to come just above the water level. Is this clear? So this is going to be in Vallisneria. Okay, Vallisneria. This is going to be in sea grasses. So this you just remember the example. Now, majority of the flowering plants use a range of animals as pollinating agents like bees, butterflies, flies, beetle, wasps, ants, moths, birds. Okay, which birds also they have written. Bats are common pollinating agents. Among the animals, insects, particularly the bees, are the dominant biotic pollinating agents. Okay. Even larger animals such as some primates like your lemurs, arboreal tree-dwelling rodents, reptiles, okay, lizards have also been reported as pollinators in some species. That means it's not necessary that only the bees are going to be pollinating, like the Sony bee. It's not necessary only Sony bee does pollination, okay. Big rodents, lemurs, lizards, garden lizards, these people also sometimes carry pollen with them. But in this insect pollination, no, the pollen grains will be sticky. The pollen grains are going to be sticky. Why? Because if they only stick to the body of the animals, the pollen grains will reach the location, no? Like that, destination. Same thing here. Look here. Majority of the insect pollinated flowers are large, colorful and fragrant and rich in nectar. So these are all rewards for the insect, no? For coming and pollinating, these are actually the rewards. Okay. Now, when the flowers are small, what happened? We will try to make an inflorescence because the insect will not be able to see one small flower, or there will be modification of the brad. Okay, like we have shown you also, no? It could be modification of the brad into the colorful petals. So such things also happen, like in bougainvillea you would have seen. In different plants you will see modifications happening. Okay, now flowers are small, a number of flowers are clustered into an inflorescence. Animals are attracted by the flowers by the color or by the fragrance. Even the bad fragrance, that means the foul smell, also attracts, like your houseflies. Okay, houseflies. Flowers pollinated by flies, okay, and beetles secrete foul odors to attract these animals. Okay, your Rafflesia is there, no? That makes a foul odor, like it's like a dead meat, that kind of a smell that attracts bees and flies, or the house flies and the bees. Okay, beetles. To sustain animal visit, flowers have provided rewards to the animals. What are the rewards? Nectar and pollen grains are usually the floral rewards. Why, students? Because understand, nectar is a reward, pollen grains are a reward because they are rich in nutrients. Rich in nutrients, and it becomes like an addiction for the insect that yes, every time I'm going, okay, every time I'm going, I'm going to get the pollen grain. Okay, every time I'm going to get some nutrients, I'm going to get some nectar. So this becomes an addiction, and that is why insect always goes back to the flowers. Now, for harvesting the reward from the flower, the animal visitor comes in contact with the anther and the stigma, and the body of the animal gets coating of the pollen grains, which are generally sticky in animal pollinated flowers. Then, students, there are cases where there is robbery also. Okay, there are insects who come, take the nectar, take the pollen grains, but they do not do pollination. So those are called as pollen robbers. Okay, nectar robbers, we tell, no, like that. Yeah. So when the animals carrying pollen on its body come in contact with the stigma, it brings about pollination. Easy, no? These are all small things. Okay, small thing. Clear? Okay. Now, in some species, one mutualism you will get to see here. Okay, can you see? I don't know if you, yeah, can you see this diagram, all of you? Look at this diagram, all of you. Can you see this? This is an insect, Prunuba moth. What is this? This is the Prunuba moth, and these you can see there. Okay, the small larvae is here. Okay, small larvae are there. The flower that you are seeing is yucca. So what has happened? Okay, what has happened? These people, there is mutualism, telling that you do one thing, you protect me. You protect me, I will do pollination for you and give. So that kind of mutualism also happens. The first one is going to happen in your Amorphophallus, which is a 6 feet. Can you see how tall it is? Okay, taller than us. We will look like this small in under that, no? Like that it looks. So it's a 6 feet in height one, amorphous. Here also what happened, this flower will tell that, okay, I will protect you, you just do pollination for me. Same thing goes here, plant yucca, where both species of moth and the plant cannot complete their life cycle without each other. And the moth used here is, the moth is Prunuba moth. Prunuba moth and the yucca plant, students. This is the image. Okay, this is the image. Can you see here? I think on the PDF you will be able to see better. Okay, this is a mutualism. That is, see, the moth deposits its egg. The moth deposits its egg in the locule of the ovary and the flowers. In return, it gets pollinated by the moth. The larvae of the moth come out of the eggs as the seeds start developing. Can you see? They have come out of the egg as the seed will start developing. Clear? Yes. Prunuba moth and yucca. Okay, and Amorphophallus. So these are mutualisms. These are mutualisms. Mutualisms. Clear? Any doubts? Okay. Okay, so we'll do a quick revision. Okay. I think Nandu, someone else will share you the link. Okay. Someone can share the link with her. Okay, listen. First of all, first we will start with your pollination, autogamy, geitonogamy, xenogamy. Autogamy means self-pollination, okay. Geitonogamy means different flowers but the same plant, so there is no much genetic variation. Third thing, xenogamy, cross-pollination, genetic variation is going to be there. Now, when you go for autogamy, you have clistogamous and chasmogamous flower. One is a closed flower, one is an open flower. One will go through cross-pollination or even geitonogamy, not necessarily xenogamy. Geitonogamy can happen there. Again, a part of autogamy only. The other person will only go through autogamy because it never opens. Now, when you go to the agents of pollination, you have abiotic and biotic. Biotic is going to be the most common, that is by insects, and abiotic you have wind and water, and that wind is going to be the most common. Now, when you go to wind pollinated flowers, remember that wind pollinated flowers will have exposed anthers and well exposed stigma as well, feathery stigma, which will attract the pollen grains. They are not going to be large, colorful or as such, because they are not going to attract the wind. No, they just want to make sure that the pollen grains are non-sticky and lightweight. Now, in wind pollination, you are going to remember your corn cob tassels, okay, which is going to help in trapping the pollen grains. When you come to water pollinated flowers, only 30 genera of species are going to do water pollination. The ones who are underwater also will prefer insect as well as wind pollination. Now, if you go further, you will understand that in Vallisneria and in Hydrilla, there is going to be pollination where the flower will come above the water level and it will wait for the pollen grain to come through with the help of water currents. Now, these pollen grains will have mucilaginous covering, so wetting can be avoided. Now, in Zostera or sea grasses, what is going to happen? The pollen grain as well as the stigma is going to be underwater, and they will wait for the pollen grain to come and fall on the stigma with the help of water currents. Okay. Now, here again the flowers are not going to be colorful or anything, but they are going to be large and long. They will have long stalk, long stigmas, anthers will be there. Even the pollen grains will have a mucilaginous covering. When you come to insect pollinated, flowers are going to be large. They will have nectar, they will have fragrance, and nectar and pollen grains are given as a reward for the insect who are doing pollination. Most common pollinating agents are going to be your bees. There are also flowers which make foul odors and foul smells to attract the flies and the beetles. Clear to all of you? Then you also have mutualism, where you have Amorphophallus, Yucca and moth, who are going to help in, one is going to do protection, the other one is going to promise for pollination. Clear for all of you? This is the whole thing. Okay, this is the whole thing. Clear? Okay, we'll take a small break. I don't want to give a bigger break. If no break is required, I'll do of outbreeding devices also, because everything is a continuation. I think let's do outbreeding devices and then we will I'll give you a break. Okay, yes. Wow, Joey, I understood. Okay, yes, now come here. Okay, let's continue. So, outbreeding devices, students. First of all, I told you, when autogamy continuously happen, okay, when self-pollination continuously happens, okay. Self-pollination continues. What is going to happen? Ma'am, depression is going to come, ma'am. If you see your family members every day, every single day, what do you tell? I feel depressed. I need to go out. I need to meet my friends. That means what? You are seeing the same people every day. You are getting, okay, you are feeling difficult. Students, I do not have water, so I'll just bring some water. Please don't go anywhere. Okay? Yeah, questions I'll tell you, Rani. Don't go anywhere. I'll just get some water. People who are going. Did anyone leave? Yes. Okay, now look here. Coming back, inbreeding depression. That is, when continuously self-pollination happens, the plant goes through inbreeding depression. But plant also knows how to get rid of it. That is, what they are going to tell, they are going to be like, we are going to make some outbreeding devices. That is, I know that I am going through depression. So what will I do? I'll go for therapy, I will try to listen to music, I will try to talk to a friend, or I'll do something that I like. Same way, plant also is like, I do not want to depend on anyone. I know what to do exactly. So it will do outbreeding devices. So outbreeding devices are mainly done to avoid inbreeding depressions. Okay, inbreeding depressions. So let's see which are the outbreeding devices. 4 you have to study in your, according to your textbook. Okay, so to avoid inbreeding depression, we go for outbreeding devices. Okay, outbreeding devices. Where have we reached on the target? Yes, tell me. So first outbreeding device is going to be, students, let's do one situation. Okay, this is your stigma. Okay, this is your stigma. Okay, this is one more stigma. This is the anther. Okay, anther. This was situation 1. Okay, this was situation 1. Too many ants are coming up. I don't know what is wrong here. Now, I don't know how this many ants are coming. Usually ads used to not come. Okay. Okay. Now, let's say this was the situation 1. Okay, this was the situation 1. Now, what they have done, the plant is like, I will do one thing. Now, anther was above. Anther was above, no? So, it will do a situation where the anther stigma's height is increased and the anther height is decreased, so that what? See, from here, pollen grains falling was very easier. Now, this way, the pollen grains cannot fall. So, first they did was asynchronization. That is, no, first they did was the change of the position. Okay? Change position of anther and stigma. Students, all this have terminologies, okay? But then, like, it's not given in your, textbook, so we don't teach actually, okay? Heterostyle, progandry, all that with terminologies are there, so that we don't teach, okay? Yes. Now, let's see. So, here, change position, okay? Change position of anther as well as stigma. Now, the next thing that they are going to do is they are going to form unisexual flowers. They are going to form unisexual flowers. Next idea. Okay, the plant will be like, I will form dioecious situation. I will not go for monoecious situation. That is, I will not form both the sexes in the same flower. I will form unisexual flowers. Third thing that they are going to do is, students, that they are going to make asynchronization, or let's say in other words, the stigma is going to mature faster than the anther, or the anther will mature faster than the stigma. So, different time of maturation, different time of maturation of stigma and anther. Okay, stigma and anther. This way, what will happen? Obviously, before the pollen grain happens, the egg will not happen. When the egg is happening, pollen grains will not happen. So there is no sync that is going to be there. Okay. Last one is going to be what? They are going to make sure that compatibility is not there. So self-incompatibility. Self-incompatibility. That is, if the self-pollen, let's say this is the same flower. If the pollen grains from this flower falls on the stigma, stigma will reject immediately, telling, hey, you're from our house only, no, go. Okay, so it will tell like that. So that means self-incompatibility. It is not going to accept the pollens who are from the same flower, same plant. So that is called as self-incompatibility. So these are the 4 things, 4 outbreeding devices used by the plant. Clear or not? Easy peasy lemon squeezy? Easy, no? Change the position of anther and stigma. Like before, anther was above. Now the stigma is above. Just increase the length of the style. Unisexual flowers. Different time of maturation of the stigma and anther. Self-incompatibility. Done, 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 done. Yes or no? This is your outbreeding devices. This way we can avoid inbreeding depression. Okay, this way we can avoid inbreeding depression. Now students, to avoid this inbreeding depression, we can also do something called as artificial hybridization. What is artificial hybridization? That is, okay, this is your anther. Okay, sorry, this is your stamen. Okay, this stamen I can emasculation I can do. What I can do? Emasculation. Emasculation I can do. That means I remove the anther. Okay, I remove the anther and I will make sure that the pollen grains from this anther does not fall on the stigma. So I will do emasculation and after that I will do bagging. So let's say this is the stigma. Okay, this is the stigma. So I want to make sure that the pollen grains from this anther does not fall on the stigma. So we can cut off the anther. Okay, we can remove it with the help of a forceps. We will remove the anther. After that, we will make sure that nothing falls on the stigma. We can do bagging. Okay, we can use a butter paper, something and do a bagging. Okay, bag the stigma and keep so that unwanted pollen grains also doesn't fall. After bagging, what we can do? We can dust the necessary pollen. Okay, whatever pollen that I want on the stigma. Okay, my desired pollen, whichever I am expecting to fall. Okay, I can dust that on the stigma. And again, I will do bagging. Why? Because so that unwanted pollen grains do not fall or no other contamination happens. Again, I will do bagging. This is called as rebagging. Okay, rebagging. So the two methods of artificial hybridization. First of all, artificial hybridization is done for what? Imagine Avengers. Okay, one guy called Avengers is in our class. He is telling that I want to make a flower. Okay, or I want this pollen grain only to fall on this. So he is actually selecting it. Artificial hybridization is doing. So what he will do, he noticed that this flower is not unisexual. Anther and stigma is there. He knows that pakka the anther is going to release the pollen grain. So he will, with using a forceps, he will remove the anther. Okay, he will remove the anther and he will bag the stigma and keep so that no other unwanted pollen grains fall on this. Then he will get the desired pollen grains. He will get the desired pollen grain. So he will bring the desired pollen grain, dust it, okay, slowly with a brush or something. You can just tap on it, the pollen grains will fall. Dust the pollen grains on the stigma. Then again bag it. Okay, bag it and keep it. This is rebagging until your desired plant is going to happen. So this two process is called as emasculation and bagging. This is going to come under your artificial hybridization. Okay, where you get an emasculated anther, that is your pollen grains, only the desired pollen will be allowed to land on the stigma. Clear? Clear? This is what is called as artificial hybridization. So we remove this. Okay, we remove this. Inside this definitely pollens are going to be there. We will make sure that this pollens will not be taken and we will use a forceps to remove this. Okay, we will take a forceps to remove this. Done? Easy. This is called as artificial hybridization. Desired pollen grains will be fertilized. Okay, fertilized. Yes, now we have pollen pistil interaction, double fertilization and post fertilization events. So yes, you can go for a 10 minutes break, but I don't want any of you to leave. I want all the 65 of you to come back. If you promise that, then there is a break, otherwise no break. Tell fast. Okay, tell fast. Will you all come back or else I will continue the class? I have energy. Okay, I have energy. You people don't have energy. But I am the one who is talking and writing. Pakka? Okay. You people will confuse me. No break, ma'am. Break, ma'am. Okay, some of you want break, some of you don't want break. It's a mix. So those who want break, take a break. Those who do not want a break, I have a task for you. Okay, those who don't want a break. Okay, those who don't want a break, this is the video that you're watching. Those who don't want a break, click on this first link, sexual reproduction in flowering plants. Type your phone number, you will automatically deviate it to a test. Once the break is over, I want all of you to come back. All of you to come back and tell me your marks. Okay, you'll have around 50-60 questions. So go solve that question during that time and come back. If you're not able to solve all the question, obviously you can't solve all the question in 6 to 7 minutes. So what you will do, solve half, don't close the tab, come back to the class, then go back and solve it. Okay, what you're going to do, this is the video that you're watching. Okay, first of all, like the video, click on the first link, sexual reproduction in flowering plants. Click, enter phone number. If you have already logged in, just use your email and log in. Okay, and finish the class and then come back. Only 5 minutes break. Okay, don't go anywhere, just keep the live open. Finish your work, drink some coffee, water, and come back here only. I'll be here only. Go. I want all 70 of them back. Okay, that's the promise. If it is reducing, then I'm not going to give any more breaks. I gave such a small bit that I couldn't even make a lemon tea, green tea in that time. So yeah. Yes, all of you are back or all of them ran? See, I told you, all of them ran. Now you people who assured me, bring them back. Quickly. Okay. Good. You'll watch it more from today. Okay. So let's continue. Pole and pistil interaction. Pole and pistil interaction. So students, as I told you, stigma knows what it should do. Stigma knows whom to accept, whom not to accept. See, when we are also in, trying to do something like selecting a class. Now, example, my life. Okay, my life, one student is there in the chat box. He's telling that, ma'am, first time I'm watching Vedantu, but he's going to enjoy this, okay? 91 marks. Very good, very good. Some of you solved the quiz, I believe. After this, you have to, those who did not solve the quiz, go solve the quiz. Then only join your human reproduction class. I'll give you a break for that. Okay, 15 minutes, I'll give you time for that. Okay, chalo. Pole and pistil interaction, as I told you. Now, let's say this is the stigma. Okay, I think today I've drawn stigma like one imposition, no? Okay, this is the stigma. Now, this is the stigma. Now, on the stigma, students, there are many chances, okay, many chances that many different types of pollen grains fall. Okay, let's say this is one pollen. Okay, different designs of pollens will be there. Now, this is one pollen. Different pollens have fallen. Now, the stigma knows that what is the receptivity. Receptivity, right? Very good. 100 marks, only 15 questions done from the quiz. Okay, it's okay. You can go back and do it later. Now, the stigma knows the receptivity or how, it has to, continue the process. Does it have to allow for pollen tube germination? The stigma knows. Now, the first thing that is going to happen during a pollen pistil interaction is going to be continuous chemical dialogues. Continuous chemical dialogues. Now, let's say we meet a person. First thing, what do? We talk to them. We try to understand them, right? Like, is this person really nice? How do they talk? Do they have knowledge? So we talk to a person and we understand, is this then, is this person nice for me or not? Same way here also, first thing that is going to happen between a stigma and pollen is going to be chemical dialogues, telling that what is going to be happening. I'm a person like this. I'm going to give you this. I'm going to do this. That thing is going to happen, okay? Second thing is, if the chemical dialogues go well, the stigma would have decided which pollen is compatible for me and which pollen is not compatible for me. Okay, let's say the stigma rejected this and this pollen is allowed to come inside. So, second thing that is going to happen is the germination of the pollen tube, or it is going to allow, okay, it is going to allow germination of pollen tube. Germination of pollen tube. Telling that, okay, you are compatible. That means I am going to let you form the pollen tube. Pollen tube formation happened. I think in PU, you people had this experiment, no? Pollen tube germination. So, pollen tube germination happened. Now, the third thing that is going to happen is the growth of the pollen tube, okay? Growth of pollen tube. That means the pollen tube is going to extend to reach up the embryo sac, okay? So, let's draw one embryo sac, students, here. Ready? One big embryo sac I have drawn. Okay, in that we will draw synergids, okay, and the egg apparatus. Okay, we will draw antipodals, okay, and the polar nuclei. See, done. Embryo sac done, ma'am. Very good. Now, the pollen tube is slowly emerging and coming, okay. It is coming. Growth is happening. Ooh. Stigma broke, students. Okay, yes. So, slowly pollen tube is growing, and it is now going to enter inside. Okay, now it is going to enter inside. So, slowly we are drawing the pollen tube, okay, all the way till here. Now in 60% of the scenario, what is going to happen? Very good, all of you are back. Good job. Good job. I'll give you more breaks. Okay, otherwise no breaks for you people. Yes, so listen here. In 60% of angiosperm, in 60% of angiosperm, what is going to happen? Ma'am, you told that in 60% of angiosperm, in the 2-cell stage only dehiscence is going to happen. 2-cell stage dehiscence is going to happen. That means when the pollen tube formation happens, your generative cell, your vegetative cell, everything is going to be there. That is your tube nucleus. It is going to come fuse with the egg. Zygote formation is going to happen. But in scenarios where there is a 3-celled stage, generative cell is going to mitotically divide and form 2 male gametes. What is going to happen during that stage? So, pollen pistil interaction is clear to you, all of you. Simple thing. Stigma is like compatible. You are compatible, I am compatible. Just like how it happens in marriages, no? These people will talk, then they will go and see astrology. They'll be like, yes, these two people are compatible. Whatever happens, hurricane come also, these two will not separate. So that means what? They're very compatible. So even stigma and pollen will be like, yeah, you're compatible. Chemical dialogues will happen. After chemical dialogues happen, stigma will be like, yes, you are the right one for me. Now you can form pollen tube. He'll start forming pollen tube. As and when pollen tube growth will happen, it will reach all the way up to the embryo sac. Now in the embryo sac, your synergids with the help of filiform apparatus, will guide your pollen grain to go inside and fuse with the egg. This is what is going to happen in 60% of the angiosperm. But in scenarios where there is a 3-celled stage, more eventful matters will happen. That is double fertilization. So students, in the embryo sac, okay, so we are drawing a bigger image of that, okay. This is the ovary. Became like one pot. Okay, this is the ovary. This is the embryo sac. Okay, same thing. Okay, same thing we are going to draw. Synergids, egg. Okay, antipodals, polar nuclei. Okay, polar nuclei. Done. Embryo sac done. Now look carefully and try to understand. Pollen tubers coming? Yes, ma'am. Pollen tubers coming. Okay, tube nucleus is going to help. Yes, ma'am. Tube nucleus is going to be there on the starting only. Now here, vegetative cell was there, generative cell was there. This was 2-cell stage. But here, generative cell, that is your GC, underwent mitotic division, and it formed 2 male gametes. Okay, 1 mg, 1 more mg. Okay. Now, vegetative cell, during this formation of 2 male gametes, it will give all the nourishment. It will give all the nourishment and it will get finished, like it will be over. Okay, it will be over. That means it will vanish. This 2 generative cell, what is going to happen? That is, this 2 male gametes, students, they will slowly go forward. The filiform apparatus will guide the pollen tube, saying that, hey, come this way, man. Okay, so it will, pollen tube will enter. Now what is going to happen? Very, very important for all of you. Remember here, stop talking to each other, listen to the class. It is going to enter and one male gamete will fuse with the egg, okay, from the egg apparatus and end up forming the zygote. Okay, this is the main heroine. This is the main process that we were waiting for. This is the main heroine. Okay, then the second male gamete will be like, no, even I need to fuse, even I came this much far, even I need to fuse. Then they'll be like, okay, you do one thing, you go and fuse with the polar nuclei. So this fellow will go and fuse with the polar nuclei and form the primary endosperm nucleus, or else you can simply write endosperm. Actually, primary endosperm nucleus again. Okay, endosperm forms the endosperm. Is this clear? Is this clear? This is what double fertilization is about. 1 second, students. Place is not there. Okay, one more male gamete will fuse with the polar nuclei to form endosperm or PEN, primary endosperm nucleus. This is called as triple fusion. Why? Because polar nuclei is considered to be a single celled but two nucleus. No, two nucleus they are. So it is called, it is going to lead to the formation of 3N. It is going to lead to the formation of 3N. This is going to lead to the formation of 2N. This is regular scenario 2N, but this is going to form 3N. So it is going to be called triple fusion. Okay, so this process, this is going to be called triple fusion, and this is going to be called syngamy. So double fertilization is the syngamy plus triple fusion. Any doubt so far? Any student has a doubt who is studying it for first time, second time? So double fertilization formula, students. Double fertilization is equal to triple fusion. No, first I write syngamy. Syngamy plus triple fusion. Syngamy plus triple fusion. Clear or not? Yes, very good. Very good. Clear? Who are the three people who haven't liked the video? Quickly like the video, and then we will be doing the class faster. This is called as double fertilization. So is this clear to all of you? Pollen pistil interaction and double fertilization, we usually teach it one together, okay? That is here 60%, then 3 cell stage, they will enter. One will fuse with the egg, one will fuse with the polar nuclei, okay? So here we can learn this together, but just understand what is this pollen pistil interaction, because here everything is going to be based on the compatibility, okay? Okay, compatibility of stigma and pollen. Yes, done. See, this is the diagram. You can see two male gametes are there, vegetative nucleus will be there, or they will almost degenerate because giving all the nourishment for the male gametes. Okay, once, see, with the help of filiform apparatus, it will be ready to go and fuse with the egg, and it will form the zygote, and then it will enter slowly. Again, fusing will happen. The male gamete will move. One will go and fuse with the polar nuclei, one will go and fuse with the egg. Clear or not? So this is going to lead to, this one is going to lead to syngamy. Okay, syngamy, and this is going to lead to triple fusion. Triple fusion. Yes, done. We are going to reach post fertilization events. Pre done, fertilization done, now going to post. Okay, yes, very good. Antipodals and the synergids will degenerate. Polar, they will continue. One will form zygote, one will form endosperm, and this endosperm is going to give the nourishment for the growing embryo. Okay, now post fertilization events. Any doubts so far? Any student has any doubts? Questions will come later. Okay, questions will come in the middle. Yes, done. Okay, good job. Now post fertilization events. Post fertilization events is going to be what, students? Zygote is going to mature and form embryo. Okay, embryo. This embryo will have plumule, radicle, root, shoot, that will continue. Okay, the pen will undergo differentiation to form your endosperm, and this endosperm is going to be the food reserve for the growing embryo. Food reserve for the growing embryo. Right? Then we have what main task is that ovule will become seed and ovary will become fruit. Ovary, fruit. This is our post fertilization events. Okay, post fertilization events. Yes, let's go. First question. Oh, question came, students. Ready? Double fertilization is what? NEET 2018 question. I told you, I don't know where the question will be placed. I told you, no. See, double fertilization, trick your brain. Double fertilization is fusion of You people read, please. Okay, and the dioecious flowering plants prevent. So tell me this, then tell this. Quickly. Yes, all NEET warriors, NEET battle people, everyone tell the answer for this. So bitter. Fast answer should come. Yes, very good. Answer is going to be D. Okay, all of you got plus there, plus here 4. Okay. Now, dioecious flowering plants prevents both. NEET 2017 question. NEET 2017 question. Gaietnogamy and xenogamy, cleistogamy and xenogamy, autogamy and xenogamy, autogamy and gaietnogamy. Yes, very good. This is also going to be D because here unisexual flowers are going to avoid any kind of self-pollination. They will go for cross-pollination, right? Yes, good job. 8. Oh, given below are two statements. Cleistogamous flowers are invariably autogamous. Cleistogamy is disadvantageous as there is no chance for the cross-pollination. Statement 1, statement 2. In light of the above, keep a memory of your marks. I'll ask you your marks at the end. Okay, and you have a particular comment to put today. That comment you cannot miss. Cleistogamous flowers are invariably autogamous. Cleistogamy is disadvantageous as there is no chance for cross-pollination. Students, in case we do not have time to do quiz because even Baswa sir should start the class, then I am going to give you the snip of the quiz on my Telegram on Vedantu NEET English. You solve the questions, okay, and let us know. Solve the questions and let us know in the comments. That's all, okay. Practicing questions is important even if it's not with me. Yes, very good. That is both statement 1 and 2 are correct, okay. Okay. In some plants, the female gamete develop into embryo without fertilization. This phenomena is known as, we will keep this on a hold because I have not taught you the concept. I know you people will know the answer. But still I want to teach you. Okay, now we are going to further, okay, what is going to happen. Now look here. The cells of this tissue, that is the endosperms, are filled with reserve food materials and are used for the nutrition of the developing embryo. So this is what I told you about the endosperm. Endosperm, okay, I'll tell you, comments means in Telegram you have an option to comment, no, how many marks you got, that you can comment. Look here students, embryo is going to be reserve food material, and they are going to provide the nutrition for the growing or developing embryo. In most common type of endosperm development, the pen undergoes, that is PEN, undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm is called as free nuclear endosperm. So there is free nuclear endosperm as well as cellular endosperm. I will teach you that now. Subsequently, there will be cell wall formation occurring and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from the tender coconut that you drink is actually familiar, right? Are you people familiar? If not, tell me, okay? So the coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm, okay? And the white kernel, the one that you eat, okay, the white kernel is the cellular endosperm, okay, cellular endosperm. Who is asking something? Nothing, no? Okay, now look here, what did they say? They are telling that this is going to be the cell. They will have, let's say, they will start forming free nuclear division. That means only nucleus is going to continuously divide, okay. Nucleus is going to, this is going to be free nuclear. Free nuclear. This free nuclear can go through, okay, it is not necessary that all the cells will get, wall formation. This free nuclear slowly you will start noticing that there will be wall formation, okay, like this wall formation will be there, Okay, and students, it's not necessary that all of them will get the wall. So this will lead to cellular endosperm. Okay, cellular endosperm. So this is free nuclear. This free nuclear is the water, coconut water that you're drinking. Okay, coconut water that you drink, and cellular endosperm is the white kernel that you eat. Done, done, done. Is this clear to all of you? So endosperm will undergo two types, no, two categories. One is free nuclear division, that is free nuclear endosperm, we can say. Nuclear endosperm and cellular endosperm. In cellular endosperm, what is happening? Wall formation you will start seeing. Okay, here you will notice only nucleus dividing. Got it? Clear or not? Yes. Okay, so here free nuclear division, here wall formation. Okay, wall formation. Good, clear. Moving forward. Now, embryo develops at the micropylar end of the embryo sacs where the zygote is situated. Most zygote divide only after a certain amount of endosperm is formed. This is an adaptation to provide. This question was asked. Okay, will endosperm precede will the zygote development precede the endosperm development? So if that is going to be, okay, if that is going to be, you know, the question, you should always remember that after, like, you're going home after work, first you will check if you have food or not. No, then only you will go home, or you will buy something from the shop and you will go. That means always remember the zygote formation, the embryo formation will only happen after some amount of endosperm is stored. Why? Because endosperm is the food reserve for them. Okay, the question has come, that's why remember this. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, that is embryogeny, are similar in both monocot as well as dicot. Even though later they are going to divide into two cotyledon, single cotyledon, starting stage everything is going to be same. Plumule is going to be there, radicle is going to be there, root shoot formation is going to be there, epicotyle is going to be there, hypocotyle is going to be there, scutellum is going to be there. All this is going to be there. Later that partitioning is going to happen. Okay, the zygote gives rise to the proembryony and subsequently to globular, heart-shaped as well as mature embryo. They are just depending on the shape. Okay, one is going to be globular. Okay, globular like this. Okay, one is going to be heart-shaped like this. Okay, and one is going to finally form the mature embryo. Finally going to form the mature embryo. Now, a typical dicotyledon embryo consists of an embryonal axis and two cotyledons. Here, look at this, students. So here, what are they talking about? These are your two cotyledons. Okay. Okay, this is your embryonal axis. See, this is your embryonal axis and these are the two cotyledons. So the one which is on the top, okay, one which is on the top is going to give rise to your shoot. The one which is in the bottom is going to give rise to the root. And these are going to be the two cotyledons. These are going to be the two cotyledons. Clear or not? So here they are trying to say, since you have studied this in morphology, I am going to be a little more fast. Okay? Yes. Embryonal axis and two cotyledons. So the center one which I have drawn is the embryonal axis. The portion of the embryonal axis above the level of the cotyledon is the epicotyle. That means this one is going to be the epicotyle. Okay, epicotyle. And the one below is going to be the hypocotyle. Okay, the one below is going to be the hypocotyle. Any doubts so far? Now, look here, which terminates with the plumule or the stem tip. The cylindrical portion below the level of the cotyledon is called as hypocotyle, and that terminates at its lower end of the radicle, or there is going to be a root tip. There is going to be a root tip, all of you know there is going to be a root tip so that there is no damage to the root, because it has to pass through hard soil and layers. Okay, hard soil and layers. The root tip is covered with a root cap. Now the cap is going to be present again to make sure that the root tip does not get damaged. Clear? Any doubts so far? Okay, moving forward. Now look here students, this is the diagrams given in your NCERT. So they can ask you the diagrams and ask you to identify. So let's learn the diagram as well. Look here, this is the root cap which is going to protect the root tip, because it has to go through the hard surface. This is the radicle which is going to further develop into the root. This is the hypocotyl, the portion that is going to extend downwards of the cotyledons. Okay, the embryonal axis down portion is going to be called as the, down portion is going to be called as the hypocotyl. These are the two cotyledons, that means it's the dicot. And then you have plumule, that is the epicotyl is going to end in the plumule. Now, when you go here, you will notice there is chorioryza, which is a portion that is going to protect your radicle, root cap, radicle, epiblast, shoot apex, coleoptile, scutellum. Scutellum is going to be a portion that is going to show us the partitioning or the difference, or like like a boundary, like how we have septum, no, like a septum, it is going to separate the endosperm and the embryo. Okay, the endosperm and the embryo. Clear? Are you able to recall whatever you studied in morphology? If you have anywhere problem, just ask me to slow down. I will stop. Okay, I will stop and teach you. Now, embryos of the monocot possesses only one cotyledon. In the grass family, the cotyledon is called as scutellum. That is, okay, cotyledon is going to be called as scutellum, that is situated towards one side of the embryonal axis. At the lower end, the embryonal axis has a radicle and a root cap enclosed in an undifferentiated sheath called as the chorioryza. Again, for protection only, okay, for like a protecting cover. The portion of the embryonal axis above the level attached to the scutellum, okay, scutellum is going to be called the epicotyl. Epicotyl has a shoot apex and a few leaf primordia enclosed in a hollow folia structure called as the coleoptile. So remember, epicotyl, coleoptile, chorioryza, hypocotyl. Chorioryza, hypocotyl. This is monocots, all the grasses. Okay, all the grasses. So what did we just study? Just an overview. Okay, just an overview. Okay, this is your two cotyledon. Okay. This is an embryonal axis. Okay, this is an embryonal axis. Okay, now look here. Okay, embryonal axis. The portion that goes above, okay, is going to end in a plumule. Remember that it's going to have plumule. Okay, and this portion is going to be called the epicotyl. Okay, epicotyl. This is all what you have learned. So I'm just doing an overview. Okay, and this portion is going to end, okay, in a radicle, and this portion is going to be called the hypocotyl. Okay, hypocotyl, and these are the two cotyledons. These are the two cotyledons. This much done. Okay, now remember, in the hypocotyl area, you're going to have chorioryza. Chorioryza. Okay, chorioryza, which is going to protect here. And in this region, epicotyl region, you're going to have coleoptile with some leaf primordia. Any doubts on this? Yes, very good. Okay, done. Done. Now students, seed. What do we know about seed? So there'll be two types of seed. Okay, X-albuminous and albuminous. That is actually depending on the endosperm. Okay, depending on the endosperm. Okay, yes. So Depending on the endosperm, we have two categories. Okay, two categories. Albuminous, exalbuminous. Okay, tell me, all of you have studied this in your morphology. What is albuminous? Tell quickly. Yes. Presence and absence is correct. Yes. So students here, no, just remember your coleoptile and coleorhiza. I think who asked? I think Rani asked, no? Coleoptile and coleorhiza is going to be present in monocots. So I'm erasing from here. Okay, they are going to be present in monocots. So just remember that, okay? Dicot, they will not be present. So here I'm just erasing this. Just remember that, epicotyl, hypocotyl. Yeah. Albuminous, non-albuminous, tell me. Yes, very good. They retain some amount of endosperm, okay? Retain some amount of endosperm. They retain some amount of endosperm. So this, they are called as albuminous. Here they do not retain, okay? No endosperm. No endosperm. This is called as exalbuminous. Now, do you know any albuminous example? Wow, some of your NEET warrior has already told. Very good. What is it? Cafe Coffee Day. Okay. To remember albuminous, you will remember Cafe Coffee Day. Okay. Castor and coconut. Castor and coconut. Yes, castor, coconut, maize, wheat, barley, you can remember here. Here you can remember, trick. Was there a trick for this? I don't remember any trick. Yes, wheat, maize, barley, castor. Exalbuminous seed. I think something related to PUBG I had told you people, no? Yeah, PUBG. PUBG, but then U doesn't have any role. This is P, B for beans, okay, and G for groundnut, okay, groundnut. Yeah, U doesn't have any role, okay, PUBG. Yes, PUBG, okay, PUBG is your albuminous and exalbuminous, okay. Oh, here it was there, students. Okay, now in angiosperm, the seed is the final product of sexual reproduction. It is often described as fertilized ovule. Okay, important, fertilized ovule. Seeds are found inside the fruit. A seed typically consists of a seed coat. Your integuments are going to develop into testa and tegmen. Okay, outer and inner layer. Cotyledons and embryonal axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to the storage of food reserves. Mature seeds may be non-albuminous or exalbuminous. Non-albuminous seeds have no residual endosperm, okay, as it is completely consumed during the embryo formation. Example is going to be P, groundnut, that is, you can remember PUBG, U you forget of, okay, U you forget of. Now albuminous seed retain a part of the endosperm, that is, they will not utilize the entire thing, they will have some amount of endosperm. You can remember wheat, maize, barley, castor, coconut. In coconut, please remember white kernel and the tender, the liquid part is going to be nuclear endosperm as well as cellular endosperm, okay. Now, very important, students, in some of the seeds, nucellus will be persistent. Look here, in some seeds, like black pepper and beet, remnants of nucellus are also persistent. So these are called as perisperm. What are these people called? Perisperm. That is, in scenarios where, okay, where the nucellus is persistent. Okay, we call it as perisperm. Okay, remember B square, or you remember BB house, Big Boss house, no? BB house, you remember? That is going to be black pepper. Okay, black pepper and beet. Okay, beet. Clear? Nucellus is persistent. This is your two things that you have to very consciously remember. Now students, when the seed, okay, is not having a favorable condition, they go to something called as dormancy. That means like how your plants go for summer sleep and winter sleep, no, estivation and hibernation, like that this seeds also go for dormancy when the conditions are unfavorable. When the condition get favorable, they will start their germination. So look here, the micropyle here, integuments of the ovules harden as tough protective seed coat. This I told you. So every person in your embryo sac, in your ovule has a job. Here these people, the testa and tegument is going to be formed by the integuments. Okay, testa teguments is going to be formed by the integuments. The micropylar remains important. The micropylar remains as a small pore in the seed coat. Why? So that it can facilitate entry of oxygen and water into the seed during germination. Because micropylar was already open. Both the integuments were there. Micropylar was open like my face, like this. Okay, two integuments were there. Micropylar was open. So that opening will remain open so that oxygen and water can come in and go out during germination. As the seed matures, its water content will get reduced so that they will shrink, okay, and the seed becomes relatively dry. That is why when you go to shop and when you buy seed from seed bank and all of that, you will notice all the seeds are very dry. But when we, buy it, when we see it grow, you'll notice that, oh my god, the seed is actually, has the water content, it looks more fresh. That is because the seed becomes relatively smaller, it shrinks and the water also will be dried. So it becomes dry, that is 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down and the embryo may enter a stage of inactiveness called as dormancy. Or if favorable conditions are available, adequate moisture, oxygen and suitable temperature, they will germinate. As ovules mature into seeds, the ovary develops into fruit, that is transformation of ovules into seeds. This is what the ultimate goal was. Transformation of ovules into seed and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of the fruit called the pericarp. So the students, wall of the ovary will become what? Pericarp. Wall of the ovary will become pericarp. The fruit may be fleshy as in guava, orange or mango. Any doubts? Any doubts? Yes. He's walking, he's Santa, he's nitrosonium, he's everything. Okay, he has 200 names. I don't know how he has this many accounts. I think YouTube will ban you one day. YouTube will be like, what is this fellow? He's using everyone's account. So students, fruits. Okay, so fruits. Wow, we are coming to the end of the chapter. Are you all excited? Okay. Fruits are of two types. True fruits and false fruits. Oh, everything is dancing. False fruits. True fruits. Yeah. And false fruits. What is true fruit, students? Ma'am, if something has to develop this way only, okay, if something has to develop this way only, then we will call it true, no? Like true silo. That means what? Here true fruits means we know that the ultimate goal is ovary will develop to become the fruit. Okay? And the ovary wall will become the fruit wall. This we know, pericarp, just now we studied. In cases where the ovary, the ovary does not become fruit. The ovary does not become the fruit, but thalamus, but other parts of the flower, but other parts of the flower I think this board will make me forget the spelling also. Other parts of the flower become the fruit, like your thalamus. So every time you open the apple, you cut an apple, next time you should remember that you are eating the thalamus. Okay, you are eating the thalamus. So like in example, apple. Okay, apple. In apple, what is happening? The thalamus is becoming the fruit. Okay, true fruits, you can remember your mango. Many other fruits you can write, students, whichever you remember that time. Yes, strawberry. Yes, strawberry also you can write here. Okay, strawberry. Yes. Now, parthenocarpic fruit. Someone was asking me now about banana. Banana is going to be a parthenocarpic fruit. What is parthenocarpic fruit? That means fruit formed without fertilization. Fruit formed without fertilization. Ma'am, what are you saying? Yes, what I am telling is correct. Fruit without, okay, fruit formed without fertilization is called as parthenocarpic fruit. Okay, parthenocarpic fruit. Now, example is going to be banana. Okay, banana is going to be the example for parthenocarpic fruit. Clear? Yes. That is, yes, banana. Fruit without fertilization. Very, very good. Wow, the likes are good. Looks nice. Every class should be like this. Next class is going to be on Friday. Okay, so don't miss it. Clear? Okay, very good. So parthenocarpy is banana, even grapes also. Grapes also, parthenocarpic fruit. Students, people induce now parthenocarpy because fertilization does not happen, no? So they will induce parthenocarpy. Next most important topic is apomixis. See, look here. Although seeds in general are products of fertilization, a few flowering plants such as some species of Asteraceae and grasses, okay, grasses have evolved a special mechanism to produce seeds without fertilization, which is called as apomixis. So here what is happening? There it was fruit without fertilization, here it is seed without fertilization. Okay, fruit without fertilization, and here it is seed without fertilization. What is fruit production without fertilization called? Parthenocarpy. Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomixic fruit. In some species, the diploid egg cell, no, is formed without a reduction division and develops into the embryo without fertilization. That means the egg will not undergo any meiotic division and directly it will form into the embryo. There are about nucellus you will study now, polyembryony. So more things are there, right? Okay. Yes, oxen. Very good. More often, as in many citrus and mangoes, you will notice apomixis happening. That is where seed is formed without fertilization. Seed is formed without fertilization. Okay. Yes. Moving forward. Now, look here, students. Polyembryony. What is happening here? Some of the nuclear cells surrounding the embryo sac starts dividing and protruding into the embryo sac and develop into embryos. So, normally you have one embryo sac. Okay, ma'am, one embryo sac is the egg will become zygote, zygote to embryo. But sometimes what happens, nuclear cells are there, no, around the embryo. They will start dividing and they will start coming inside and they will also become like an embryo, and that will lead to something called as polyembryony, which you see normally in citrus fruits. Okay, occurrence of more than one embryo in a seed. More than one embryo in a seed is referred to as polyembryony. This is mostly seen in citrus fruits. Okay, some seeds of orange, squeeze them and see, you will get the answer. Okay, that means mostly in citrus fruits. Any doubts in this? Any doubts in this? Student, this is totally about, you know, why hybrid varieties of several fruits are available in India. Okay, all that stories. But here I would like to make you all write few things. Okay, we have come to an end of the chapter. So first thing, first fruit. Okay, first you write Parthenocarpy. Second, you write apomixis. This is very important, okay? Third, you will write polyembryony. Polyembryony. Right? All of you tell me one by one. Yes, Parthenocarpy is fruit. This is just for your reference, okay? Last minute revision. Fruit without fertilization. Example, according to NCERT, banana. Okay? This is seed without fertilization. Seed without fertilization. Example, what is seed without fertilization? Citrus, mango. Okay, citrus and mango. And this is happening because chances are there that the egg, okay, diploid egg, does not undergo meiosis. Okay, meiosis does not happen and it directly forms the seed. Okay, directly forms the seed. This could happen. Okay, yes. Now here, occurrence of more than one embryo is called as polyembryony, and this is happening because of the nuclear cells. Okay, this is happening because of the nuclear cells dividing and protruding. Okay, so this is the example. Is going to be citrus. Okay, citrus fruits like your orange, lemon. Yes, orange. Okay, these three topics you remember very clearly. Yes. Now, chapter done. Okay, do few questions. We have 5 more minutes. Okay, 5 more minutes. I should give the class to Baswa. Okay, so quickly do this question. So students, don't worry. You also have questions here. So don't be like, you ma'am did not make us do questions. So this was the plan to make you do proper tests. So you know the marks. See, many of them did it during the break time. So here, this is the video you're watching. Okay, I'm just going to refresh the video so I can see the likes as well. See, I'm refreshing the video. This is the video you're watching. Okay, and here, first link. Can you see this first link clearly? Okay, the first link, click on this first link after this class. Oh, right. Yes, this first link you click and put your phone number. In case it is not logging in, just go to that incognito mode. No, just click on that, try to open with the link. Okay, sometimes some glitch can happen when everyone is trying to open. So yeah, so do the quiz here. Now answer this. Yes, very good. The egg apparatus is made up of 2 dash, 1 dash and the dash end of the embryo sac. Egg apparatus is made up of 2 synergids and 1 egg cell at the micropylar end. Okay, very good. Option number 4. Okay, so plus 12. Plus 12. Consider the following features seen in a plant and you have to tell which kind of pollination is it. Okay, faster. After that, I'll give you a break. Male and female reproductive organs are generally found in separate flowers. Male flowers having a number of long filaments terminating in exposed anthers. Female flowers having long feather-like stigmas. The flowers of the plant would most likely be pollinated by. Yes. Easy question. Feathery stigma, only one person will have. Who is that? Yes, very good. Wind pollinated, plus 16. Okay, plus 16. The cells of which of the following layers of the anther wall are multinucleated due to the endomitosis? You know this answer as well. From my notes, every answer you should be able to know. Yes, quickly. Very good, very good. Answer is going to be tapetum. It's multinucleated. Okay, multinucleated plus. Yes, the body of the ovule is fused within the funicle at NEET 2020 question. So I told you, no, don't ignore simple questions. Simple questions also come. Okay. Okay, people have added that first question also, mitosis and meiosis. No, that also plus 24. Body of the ovule is fused within the funicle at what? Oh, this is not the answer. Okay, I just put 2020 tick. Yes, very good. C, hilum, plus 28. Okay, I've added that 4 marks also. Plus 28. What is the fate of the male gamete discharged in the synergids? 2019 question. Fast you do and then you go for a break. Yeah, it's 8:35. Okay, sir should be able to start by sometime. What is the fate of male gametes? Male gametes discharged in synergids. Madhumati fan of Ammu ma'am. Wow. Wow, wow, wow. What a name. All these things are small things that makes, teachers smile. Yeah. What's the answer? Jaldi bolo. All fuse with the egg. One fuses with the egg. The other fuses with the synergids. One fuses with the egg. The other fuses with the center nuclei. Yes. Yeah, even I am hungry. Only you're hungry. I'm not human or what? I'm also hungry. What is the fate of the male gamete discharged in the synergids? One will fuse with the egg, the other one will fuse with the central nuclei. Double fertilization, right? Plus 32. Okay, plus 32. Okay, faster. Pollen grains can be stored for several years in liquid nitrogen having a temperature of. Easy question, 2018 questions. Who told you questions will be difficult from this chapter? Sukhmeet, I can see your messages now. Try to put letters in case numbers are not coming. Try to put numbers in case letters are not coming. Okay, try things like that, students. Sometimes I don't know why they retract the message. Your Gopika ma'am not human, it seems. Who is this nitrosonium? We will put some chemical and blast this fellow. हाँ, very good. A, minus 196 degree Celsius. Okay. Human reproduction class, Gokul, after this, after 10 minutes break. Double fertilization is what? Tell fast this also. I didn't eat anything, Selvi. Yeah. Double fertilization is fusion of two male gametes with one egg, fusion of one male gamete with two polar nuclei, fusion of two male gametes of a pollen tube with two different eggs, syngamy and triple fusion. Just now we did this question. Repetition it is 2018. Okay. This is going to be plus 32, no, here. Oh, sorry. Yes, plus 36. Plus 36. 40. We are stopping at 40. 40. In the train they tell, no, T, like that it will plus 40. Yes, D, very good. So 40 marks from my class, okay? 40 marks. Now 40 plus the quiz marks should be posted on the comments. Now let me tell you what you should comment. That is very, very important, okay? I made something very fancy for you people. See? So today's Biology 360 Club part 1 is done. No one is going anywhere. Baswa sir is in the charts and he's going to check on the numbers, okay? If you reduce the numbers, okay, so don't do that. All of you take a break and come back to the class. I will give you the break timing as well. Now listen here. What you are going to type in today's comment section is very, very important. Then only you are going to be the member of the club. Okay, if I like your comment, that means you are a member of the club. If I did not like your comment, that means you are not the member of the club. Clear? Okay. Yes, definitely. Droppers, partial droppers, full droppers, sleeping droppers, any droppers can join this batch. Okay. There's nothing called as who are not even in a mindset to study. That's why I told you sleeping droppers also can get up and start watching this class. Now look here. You have to type that day 1. Okay, Biology 360 Club members, you just smashed day 1 with the reproduction unit. Okay, reproduction marathon. You are officially unlocking the elite badge. If you attend 3 sessions, then you are going to get the VIP badge. That means these students are going to get quizzes also from our end. You will get PDF of quizzes, whichever chapter you ask. Okay? Clear? Yes or no? So this should be the comment that day 1 reproduction marathon done and you people are the elite. Okay, I should like your videos. That means you are elite. After that, you should tell your quiz marks. Okay, quiz marks in the comments after doing the quiz, after the class end. And also you should tell my 40 marks, how much you got, and then whatever Baswa sir gets. So total everything and tell the marks. Okay, clear are all of you? So that's it for today. Thank you so much for joining and I hope all of you also will visit our Christmas offers and all of you are going to be amazed with our Christmas offers as well. Sir has done a video and all of you can go check it out. But before that, do not forget to do the quiz. That is the most important task for the day, to do these 2 quizzes today. Sexual reproduction in flowering plants, human reproduction. The team has worked so hard to make sure that we curate the quiz for you today itself. So after the class, you're getting the revision and I told you, Biology 360 Club is not just a class, but it is a full package of concept, diagrams, clearing your doubts at the same time, quiz and in-class quiz and after the class quiz. Okay, so more than this, I don't know what we can do for you students. We are trying our best level to do maximum. So see you all in human reproduction. Your break is going to be, you're going to have a small break because I gave you break. Okay, so you're going to resume back. Okay, resume back for human reproduction. Human reproduction at 8:55. Okay, 8:55 p.m. You're going to resume back and sir is going to start, okay, sharp at 8:55. Clear? So resume back at 8:55. So I hope all of you enjoyed Gopika ma'am's class. Please do let me know in the comments how you found my class, how you found Baswa sir's class, and please don't fail to drop that, message, because we have worked whole night yesterday full day to make sure that we can concise the whole chapter in 3.5 hours, because this chapter is a 5.5 hours class and I have done it at a faster rate also. Okay, so thank you so much for joining. See you all. Don't go anywhere. Come back. Till then, let's make the target likes target likes to be 350. Okay, target likes 350. Yes, I'm going to run and eat something. You people also go eat something, wash your face, and sit for human reproduction. And after that, definitely do both the quizzes and drop the comments. Only if I like your comment, that means you're part of the elite batch. That means you have done something wrong in the comment. That means day one is not completed for you. So till then, see you all. Bye bye. Don't forget to let me know how Biology 360 Club went, and are you excited for the next class?
Just check if it's working properly, that's all, bro.
Yeah, check it.
Hello students, welcome to Vedantu NEET English. This is Baswaraj sir, your biology master teacher. Quickly let me know in the chat, students, how is everyone doing? And quickly rate your preparation in your NEET preparation right now out of 5. Quickly go, quickly students. And just for a preview right now, this is how we're going to study today. Entire human reproduction. Okay, so let me know in the chat, students, quickly right now if you are excited for human reproduction, because it is going to be a long lecture, and I'm going to be honest with you. If you attend the lecture till the very end, I repeat again, if you attend the lecture till the very end, you will obtain those 40 marks. Otherwise, no, it's gone otherwise. So if you are serious, if you attend lecture till the very end, quickly some fire emoji in the chat, because this is going to be the hard work which you put in right now. This is the hard work which is going to count. Okay, so with that being said, students, let's start today's amazing class. Yes, students, quickly help me in the chat, students, if I'm audible and visible to all of you. Am I audible and visible to each and every one of you? Audible and visible. Yes, all of you. Quickly let me know, chat, students. Am I audible and visible? Yes. Amazing. Now, let me ask all of you a very simple question. That is, what do you call the type of reproduction in the case of humans? Is it a type of sexual or asexual? Very simple question. Tell the answer in the chat. Quickly in the chat, students. The type of reproduction do you find in the case of humans? You will tell me name of the chapter, sir. Sexual reproduction. So in the case of humans, we find sexual reproduction. Now, if you ask me what is the weightage, of this chapter, students, generally speaking, usually we end up getting around 4 questions. We end up get usually 4 questions to 5 questions. And you cannot ignore them. Let me tell you again. All the questions will be generally moderate level questions. Okay, moderate level questions. Now, students, a question to all of you again. Tell me, is reproduction a fundamental feature or is it a defining feature? Now, if you want answer to this question, it is not given in this chapter. I'll repeat again. The answer to the question, is reproduction a fundamental or defining? The answer to this question is given in the chapter Living World. Now, if you have finished the first chapter of your class 11th, you will tell me, sir, reproduction is a fundamental feature. Why is it fundamental? Because there are certain exceptions here. Now, quickly, students, quickly in the chat, tell me what are the exceptions to sexual reproduction. You tell me, sir, there is infertile couples, there is a male drone bee, there is hybrid animals like mule which cannot undergo reproduction. So it becomes a fundamental. Small context. Now, students, let me tell you today, right now. Honestly, I'll tell you right now. If you do not know anything about this chapter, zero knowledge. If you have zero knowledge about this chapter, today I will teach you in such a way that 80 to 90% you will understand in my class itself. And again, if you have learned this chapter somewhere from me itself or coaching centers or any other YouTube video on the internet, you will take away the 100%. That is my guarantee to all of you. Deal? Deal, students? I'll make you understand every single concept in a way that you will be able to solve every single PYQ that exists from this chapter. That is my deal from my side to all of you. Okay? Now, students, sexual reproduction. Humans fall under the category of sexual reproduction. Now, again, one more concept coming up. Can any human complete the life cycle? Can a human complete the entire life cycle without reproduction? Simple question to all of you. Can a human, male or female, can they finish their entire life cycle without reproduction? Or reproduction is very important, Without reproduction, the life cycle is incomplete. You will tell me, sir, reproduction is optional. A human life can exist, human life can finish without reproduction also. Infertile couples as an example again. Okay. Now, apart from that, students, apart from that, there is a concept called as viviparous. How many of you heard? Raise your hands. How many of you heard about viviparous? Quickly raise your hands, all of you, raise your hands. Viviparous. I say humans are viviparous. Meaning is that humans give birth to the young ones. A human will give birth to a young one, that is called as viviparous. Contrarily, there is something called as oviparous, that is laying eggs. We are viviparous. Remember that much. Okay. Now, student, the first concept today is called as events. Events in sexual reproduction. What are the different events? Okay. For example, let's take a male here. Male. And let's take a female here. Female. Male and female. Okay. Now, male and female, students, what do you think is the first event which happens here? The first event which takes place in sexual reproduction. Obviously, production of gametes. One sperm will come here. Here there will be one, what, students? Egg. Egg. If you know the answer, tell the answer in the chat. Simple as it is. Okay. The first step is formation of gametes. There is a terminology in biology. The terminology for formation of gametes is called as, gametogenesis. It is called as gametogenesis. It is called as gametogenesis. Okay, gametogenesis. Now, students, now this gamete has to go here. Can it fly and go? No. It has to undergo sexual intercourse, and whenever there is sexual intercourse, the sperm is released in the vaginal tract. The sperm is released in the vaginal tract, and that process of release of sperm into vaginal tract is called as, insemination. It is called as insemination. Insemination. Done. It is called as insemination. Now, once there is insemination, the sperm and the egg will fuse together. The fusion of sperm and the egg is called as, students, what do you call that? Quickly in the chat, students. Hello, hello, hello. Hello, hello. Students, am I audible? Sound check, sound check. Is it proper? Yeah, proper only. Some scam you're doing. Don't do scam. Okay. Now, insemination happens. Now, if the sperm and the egg fuse together, what is that called as? Students, tell me. The fusion of sperm and the egg is called as fertilization. It is called as fertilization. Now, students, tell me, all of you, quickly tell me in the chat, what is the ploidy of the gametes? What is the ploidy of gametes? The sperm is generally haploid in nature. The egg is generally haploid in nature. When they fuse together, we end up getting a diploid structure that is called as what, students? If you know the answer, you tell the answer in the chat. If you know the answer, tell the answer in the chat, all of you. That is called as zygote. That is called as zygote. Then there is zygote formation. Now, this zygote will start to develop. It will divide. This zygote right here is actually unicellular. It is unicellular. This unicellular zygote will undergo mitosis or meiosis. Tell me, students, in the chat quickly. It will undergo mitosis. And it will divide in number. And once it starts dividing, it produces a structure called as blastocyst. If you don't know, I'll tell you today. If you don't know any of it, I will tell you today. Okay. It forms something called as blastocyst. Now, this blastocyst will attach to the endometrium layer. That is called as implantation. Blastocyst will attach to the endometrium layer, that is called as implantation. Now after implantation, what happens? The embryo starts to develop now and there will be development of baby. That is called as what, students? Gestation. 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 And finally, students, after gestation, the baby is completely developed. After 9 months of gestation period, after 9 months of gestation period, the baby is born. The birth of the baby is called as parturition. That is called as parturition. Right here. It is called as parturition. So, it starts with gametogenesis, insemination, fertilization, implantation, gestation and parturition. That is going to be our goal today. It doesn't matter how long it takes. It is my duty right now to today teach you the every single concept in detail, not even missing a single point to you to all today. So if you have the determination, if you have the determination and that skill to sit for the next 3 hours, you will properly gain the 40 marks. So students, with that being said, can we start today's amazing class, that is human reproduction now? Can we start, all of you? Can we start? This is going to be our entire menu today. What we'll be studying today. Every single topic in detail we'll be learning today. In detail. Now first topic today. That is called as the male reproductive system. Now students, what is actually system? What is the meaning of system? The meaning of system is that multiple different units are working together. For example, simple computer system. All of you have computer in the house, no? Computer. One mouse will be there, keyboard, monitor, CPU. Every single unit is working together to run the computer. Similarly, there is a digestive system, esophagus, buccal cavity, stomach, small intestine, large intestine. Every single unit is working together for digestion of food. There is excretory system. Similarly, there is reproductive system. Now, if you ask me the location, what is the location of reproductive system? Students, can anyone tell me what is the location now? What is the location? The location of reproductive system is actually between the 2 coxal bones in the abdomen region. Can you see here, students? This region is called as abdomen region, and that is the location of reproductive system. Male reproductive system, students. Male reproductive system. Now, male reproductive system consists of 2 separate categories. The first one is called as primary sex organs. Primary sex organs. Now, students, listen to me very carefully. Listen to me very carefully. A primary sex organ has 2 main functions. All of you, how many? 2 main functions of a primary sex organ. Tell me in the chat, quickly tell me in the chat. A primary sex organ should be able to, produce the gametes, as well as the hormones. Gametes as well as the hormones. Now students, right now we are talking about the male reproductive system. The gametes involved here are the sperm. And the hormone involved is the testosterone or the androgen. Testosterone or the androgen. Now students, I spoke about gamete. I spoke about hormone. Tell me, which is the primary sex organ actually? Which is the actual organ here? The actual organ right here is nothing but the testis. The primary sex organs are the testis. Is the primary sex organ. Okay. Now, a primary sex organ will directly be involved in the reproduction. Direct involvement. At the same time, there is something called as secondary sex organ. Does anyone know? A secondary sex organ are the ducts. Ducts plus glands. There are different type of ducts and different type of glands. There is seminal vesicle gland, bulbourethral gland. There is something called as prostate gland. 3 different glands. There is ducts such as vasa efferentia, radiate testis, vas deferens, epididymis. All of these are called as the ducts. Okay, there is something called as ducts as well as glands. If you do not know anything, do not worry, I'll teach you today. I'll teach you the entire thing. Ducts as well as the glands. Okay. Now students, listen to me very carefully. This entire testis, right, to a pair of testes. A pair of testes is actually outside the abdominal cavity. See, I told you the entire structure, the entire system is in abdomen. But the testis is actually outside the abdomen. You might ask me why and where. Why, I'll tell you later. First I'll tell you where first. Okay. The entire testis is located in a pouch-like structure called as, anyone? The entire testis is located in a pouch-like structure outside the abdominal cavity called as scrotum. That is the location. That is the location. Now you might ask me why. Why is it outside the abdominal cavity? Reason is very simple. Listen to me, the reason. It is a PYQ. PYQ. The testis is outside the abdominal cavity. The main reason is that the formation of sperm, which happens inside the testis, the formation of sperm requires 2 to 2.5 degrees less than the body temperature. See, body temperature is something, 2 to 2.5 degrees, it should be less. So, it is outside the abdominal cavity in a pouch-like structure called as scrotum. Okay, sac-like structure, 2 to 2.5 degrees Celsius. Now, students, one extra information here. Now, sometimes they might ask you, when does the testis develop? Listen to me, students, listen to me. The testis develop at the 7th month. 7th month of fetal development, 7th month. Now, is there a chance that, students, in some cases, in certain animals, will the testis doesn't come out only, doesn't descend in the scrotum? There are the chances. Okay, listen to me. I'll tell you an extra concept today. Out of NCERT, I'll write it in pink. Okay, there's something called as intra-abdominal condition. Intra-abdominal. Now, in the case of intra-abdominal, it happens in the case of elephant. In the case of elephant, the testis is not coming into scrotum. They're not descending into scrotum. Okay, do not descend into scrotum. Do not descend. Okay, do not descend into scrotum. Okay, that is called as intra-abdominal condition. But students, I'll tell you one more fancy condition. Want to know one interesting fact? I'll tell you today. Fact. In the case of certain mammals which can fly, can anyone tell me? Students, quickly in the chat. Can anyone tell me which is the mammal which can fly? Flying mammal. Flying mammal is called as bat. So in the case of bats, right, only during the breeding season, the testis will descend and again it will go up. Amazing mechanism the bat has. Only during the breeding season, the testis will descend and again it will go up. Okay, so that happens in the case of bats. Now there's a disease in humans called as cryptorchidism. In cryptorchidism, the testes do not descend at all. Okay, in the case of the disease is called as, let me write the extra information here, it is called as cryptorchidism. In this disease, the testes do not descend at all. There is no descending at all here. Okay, so all of this is about testis and scrotum. I believe there should be no doubt here. So, sir, can you say intra-abdominal in simple words? Intra-abdominal means very simple, students. In humans, the testes descend into scrotum, right? In the case of intra-abdominal animals like certain mammals where elephant and some sea mammals, it does not descend at all. That is a permanent condition. That is called as intra-abdominal in certain mammals. In the case of bats, it will come and go. Okay, extra information here and there for all of you today. Okay, this is the disease. Cryptorchidism is a disease in which the testes fail to descend. It does not descend at all. Okay, now students, now we talk about testes again. That is Functions. I told you already. Production of sperm and secretion of androgen. Now students, let's understand the internal. How does the testis look from the inside? Okay. How does it look from the inside? If you take a testis and you cut it open. What exactly happens here? I'll tell you. Okay. Now students, if you see here, can you see all of you? Is it visible to all of you? If you open the testis, there is a outer layer here. Okay. Testis actually has 3 layers. We'll not get into that. 3 different layers and all. It's written here also. But I'll tell you what you need to focus on. Okay. If you look into testis, it is made up of around 250 compartments. It is made up of, students, it is made up of 250 compartments. And these compartments are called as testicular lobules. Can you see here? They are called as testicular lobules. Different compartments. Now, inside each compartment, for example, there is a big house. Imagine this is a big house. In this house, there are 250 rooms. There are how many? 250 rooms. And each room is called as what? Each room is called as testicular lobule. Okay. Now, inside each testicular lobule, there is 1 to 3 highly coiled structure called as seminiferous tubule. Can you see, students? This one. Can you see all of this? It's called as seminiferous tubule. All of you can see this. Now, this is the different ducts I told you. Rete testis, epididymis, vasa efferentia, right? These are different ducts. These are different ducts here. Okay. Now, students. Now, I'll tell you one small extra concept. I can listen to very carefully because all of these small things are very important for a concept building. Ready, all of you? Ready? One extra concept to all of you. See this now. I told you that testis will descend into scrotum. Okay. For example, this is the, there's a tube like this and this is the scrotum. For example, this is the scrotum here and testis are inside here. And here there is a tube-like structure through which testis is descending. This entire tube, students, this entire tube is called as inguinal canal. It is called as inguinal canal. Okay, inguinal canal through which the entire testis is descending downwards. Now, this inguinal canal is very important. Why? Because inside here there is something called as spermatic cord. Spermatic cord is inside this. There is blood vessels. There are certain nerves here. Even the epididymis. All of these structures are present here. Inguinal cord and testis is present here. That is how the testis is coming down. Now I'll show you the diagram. Can all of you see this? Can you see here? This is the inguinal canal here. I hope it is visible to all of you. Can you see here? This is the inguinal canal through which the testis should descend down. Now there is a condition called as inguinal hernia where there is a blockage. This is the inguinal hernia. This is the inguinal canal and there is a testis here into the scrotum. Okay. So inguinal canal and testis. This is the diagram. Inguinal canal and testis. Okay. Clear, all of you understand? I hope all of these small details, I usually skip it, all of this, but today I decided that it's fine. I think my students should understand the concept rather than just mugging up everything. Okay, so small things I'll tell you extra today. So pay attention, extra attention to it. If there is any doubt, you can always ask me. Okay. Now, beautiful concept this is. I hope all of you can see this inguinal canal and this is the scrotum and the testis is here. Okay. The blockage can happen, that is the inguinal canal hernia. Okay. Now, students, this is the entire testis. Inside testis, students, can you see? This is called as, this right here is called as testicular lobule. Testicular lobule. Okay. It is called as testicular lobule. There are around 250 of them. 250 testicular lobules. 250 compartments. Okay. Now, inside each one of them now, inside each testicular lobule, there is something called as seminiferous tubule. There is something called as seminiferous tubule. That is how much? 1, 2, 3 highly coiled structure. Okay. So this is the entire thing is the testis. Testicular lobules, there will be 250. Inside testicular lobule, there will be highly coiled structure called as seminiferous tubule. I will show you one more diagram. See this. I hope this is visible to all of you now. Can you see this? Cross section of a testis, testicular lobules, and then there is seminiferous tubule inside. Highly coiled structure called as seminiferous tubule. Now this is the concept which I want everyone to understand. Because NCERT is telling you these small lines. But when you visualize them, you will understand better, and that is my goal today. It could be external genitalia of female. It could be any diagram. Today I will show it to you. I will make you understand. Okay, I will make you understand. Okay. So this is called as the TS of testis, testicular lobule, seminiferous tubule. Okay. Seminiferous tubule. Now students, understand. So right now the mind is set, right? Inside testis, 250 compartments called as what? Testicular lobule. Inside each testicular lobule, 1 to 3 highly coiled seminiferous tubule. Now understand very carefully. Inside seminiferous tubule now. Understanding? First testicular lobule was there, compartments. First we learned the compartments. Inside each compartment there is seminiferous tubule. Now we will look inside the 1 seminiferous tubule now. Inside seminiferous tubule. Ready all of you? Ready? Is the brain braining still? Is the brain still braining? Yes. Okay. Now understand. Okay. Inside the seminiferous tubule now. Inside the seminiferous tubule, students, there are two types of cells. Inside seminiferous tubule, there are actually two types of cells. Inside seminiferous tubule. Okay. Now students, one cell is called as the male germ cell. Male germ cell. Now understand students, this male germ cell has one more name. Male germ cell has one more name. The male germ cell is also called as, spermatogonia. Spermatogonia. Understand students, why is this important? This spermatogonia right here will undergo meiosis. This spermatogonia undergoes meiosis and will produce the sperm eventually. This is diploid and this is haploid. Okay. This is called as male germ cell or spermatogonia. How many of you know that? All of you know that. Okay. Now, second type of cell is called as Sertoli cell. The Sertoli cell is also called as nurse cell. Now you might ask me, sir, what is the function here? Multiple functions. I'll tell you only two functions here. The main function of nurse cell, what will nurse do, student? Nurse provide nutrition. It provides nutrition. Also students, this nurse cell has something called as, ABP. What is ABP? ABP stands for androgen binding protein. What does it do? This ABP will completely bring all the androgen and it will help in spermiogenesis. Not spermatogenesis. Spermatogenesis will happen here. These are two different terminologies. I will teach you what exactly it means. Do not have to worry at all. Nothing you have to worry about. These two I'll teach you today only. What is the difference between spermiogenesis and spermatogenesis? Spermiogenesis, nurse cell or Sertoli cell. Spermatogenesis, spermatogonia will undergo. Beautiful concept I'll teach you today. Okay. This is the entire Sertoli cell, male germ cell. Okay. Now students, understand, all of you. This outer part here, outer part which is present here, outside the seminiferous tubule. Outside the seminiferous tubule is called as interstitial space. In this interstitial space, there will be something called as interstitial cell. In this interstitial space, there is an interstitial cell. This interstitial cell is also called as Leydig cell. Leydig cell. Leydig cell. Okay, Leydig cell. Now you might ask me, what is the meaning of, what is the function of Leydig cell? The main function of Leydig cell is to produce the androgen. To produce the androgen. To produce the androgen. Now again, I'll give you a small recap. Listen to me very carefully. You will learn very beautifully today. Listen to me very carefully. Okay. So what we just learned now, students? We learned that from here I'll teach you. This is the testis. Testis will descend from the inguinal canal. Inguinal canal has the spermatic cord, blood vessels, nerve cell. This is the inguinal canal, testis. Now, inside each testis, there are 250 compartments called as testicular lobules. 250 compartments. Inside each testis, there is something called a seminiferous tubule. 1 to 3 highly coiled. Here can you see seminiferous tubule? Inside each seminiferous tubule, there are two types of cells: Sertoli cell, nurse cell, male germ cell, spermatogonia. Spermatogonia will develop into sperm, spermatogenesis. Male Sertoli cell or nurse cell provides nutrition, androgen binding protein, and help in spermiogenesis. Entire space is called as interstitial space cell, or it is called as interstitial space. Leydig cell is present here. Production of androgen. Can you see a diagram again? Diagram, all of you. One is the highly coiled structure, seminiferous tubule. Two types of spell. Spermatozoa is the sperm. Spermatogonia is the male germ cell. Sertoli cell is also called as the nurse cell. All of that is present inside itself, inside. Can you see this part here, outer part? This outer part is called as the interstitial space, where we have the Leydig cell, blood vessels, WBC cells. Yes, here. Leydig cells are present here. Leydig cells, immunocells, WBC, blood vessels, everything is present on the outside. If you see the NCI diagram also, each one is the seminiferous tubule. Inside the seminiferous tubule, there is spermatogonia, spermatozoa inside, Sertoli cells on the inside. In the red part here, red color part, that is the interstitial space. There is interstitial cell, also called as the Leydig cell. Also called as the Leydig cells. Beautiful, right? Tell me, students. Very easy. Very easy. Yes. All of you understanding? All of you understanding? Yes. All of you. Interstitial space has Leydig cell, immune cells, blood vessels. Inside what is there? Sperm, male germ cell, spermatogonia, Sertoli cell is present inside. Okay. This is the entire inside part. Okay. This is the diagram given in the NCERT, which does not explain anything much, but it shows the different type of ducts. Okay. So that was all the primary sex organs done and dusted. All the primary organs done now. Now we move on to the secondary organs now. We move on to the secondary sex organs. Okay. Now we learned that primary sex organ is directly involved in reproduction. Secondary sex organ is indirect role. Like you have no, there are two friends. One has direct influence, one has indirect influence on you. They have indirect influence also here. That is, these help in transport, maturation and storage of gametes. No production, nothing. Okay, they help in. Now what do we have here? Here we have different type of ducts, glands. Ducts and glands are part of here. Ducts and glands. Okay. Now students, I will teach you the entire flow chart now. Listen to me very carefully, the entire flow chart, because this has been asked multiple times in examination. Multiple times they have asked you the entire flow chart. So are you ready? Are you ready, students? Yes. Ready, all of you? Ready? Ready? Listen to me very carefully, the entire flow chart now. Entire flow chart is going to be very important. What exactly happens, from where it happens, what is the flow of sperm, that is very important. Okay. Now, understand all of you. Tell me, students, where exactly the sperm being produced? The exact location of sperm is seminiferous tubule. Exact location of sperm production is seminiferous tubule. Okay, inside seminiferous tubule, sperm is being produced. Okay, so the first point here is the testicular lobule. Okay, from testicular lobule, the sperm will now go to the retate testis. From retate testis, it goes to vasa efferentia, third. Fourth is epididymis. Students, listen to me. This epididymis right here is actually the longest here. This epididymis right here is highly coiled. It also has three parts: outer, middle, inner. Three parts are there: proximal, postal and everything, anterior and all. Then from epididymis, it goes to vas deferens. Now students, tell me, if I cut the vas deferens and if I tie the vas deferens, what is that called as? If I cut the vas deferens and if I tie it, what is that phenomena called as? What is that process is called as? Tell me in the chat. Okay, this is the fifth one. Okay, now listen to me very carefully. Here vas deferens is looping over the urinary bladder, and this point here, all of you focus at this point right here. At this point right here, two ducts are meeting here. I repeat again, two ducts are meeting at one point here. The point of meeting, the two ducts, what are the two ducts? One duct is coming from vas deferens from top here. The second duct is coming from seminal vesicle. The seminal vesicle and vas deferens, both of them are meeting at this point, and the point of meeting is called as ejaculatory duct. This point right here. Point of attachment is called as ejaculatory duct. Right here. From here, there is secretions of seminal vesicle, secretion of prostate, secretion of bulbourethral gland. What are secretions? NCERT tells you three main things. That's all. Fructose, enzymes, calcium ions. Three things, that's all. Fructose, repeat after me, students, repeat after me. Fructose, calcium ions, and as well as enzymes. But today I'll teach you the entire detailed one also. So you don't have to worry about anything. Okay. Bulbourethral gland will produce lubricants. It will produce mucus for lubrication. Okay. Here, this is paired. Bulbourethra is also paired. Prostate is single. Okay. Paired, single. Now from here, the entire secretions of all the three glands plus the sperm from here together is traveling to the common point called as urethra. Now what do I call it common point? Why do I call it common point, students? Listen to me very carefully. Urethra is a common passage for both sperm as well as urine, and that is why it is called as urogenital tract. It is called as what? Urogenital tract. It is a common passage. It is a common passage for the release of sperm as well as the urine. There is also certain erectile tissues. Can you see here? I'll tell you that also today. Don't worry, I'll teach you the erectile tissues. Both the erectile tissues I'll teach you today. Erectile tissues. Okay. Now, finally, the sperm is coming to the swollen end of penis. That is called as glans penis. This right here is called as the glans. Okay. Glans penis. Glans penis. Glans penis, it's written here only. Glans penis. Okay. Now the entire penis is covered by a loose sheet, loose skin folding. That is called as foreskin or prepuce. Foreskin or prepuce is the loose folding on the outside. And that is the entire male reproductive system. Entire male reproductive system on the board to you. Now if you have trouble remembering the diagram, I have a flow chart also today. Students, I sat and made this to you for all of you today. Okay. See, sperm is produced in the seminiferous tubule. Now I want everyone to add green hearts in the chat if you understand every single point. Ready, all of you? Ready? Ready? All of you? Okay. Sperm is produced in the seminiferous tubule. Done. All of you know that. Then it passes through rete testis. Second. Reaches vasa efferentia. Done. If it leaves the testis to travel through epididymis. Next. And remember, students, within the epididymis, the sperm is matured also. Extra point. Students, I'll tell you the extra point so many. Listen to my words today. You will understand so many new concepts. So in epididymis, the longest sperm matures there also. Okay. Now, located along the posterior surface of the each testis, which leads to the vas deferens. So, seminiferous tubule, rete testis, vasa efferentia, as well as epididymis and vas deferens. Here also. Can you see, all of you? First is the seminiferous tubule, second rete testis, vasa efferentia, epididymis and vas deferens finally here. Okay. Now, the vas deferens ascends over the abdomen. Yes, done. Loops over the urinary bladder. It receives, I told you, it receives a duct from the seminal vesicle. Two ducts are coming together and that point is called as what? Ejaculatory duct. Ejaculatory duct. When it receives a secretion from the prostate gland also, prostate gland. It reaches the urethra, which is the common duct for urine and semen. It receives a secretion from the bulbourethral. Along the urethra, exits from the external opening called as urethral meatus. Now, what is the urethral meatus here, students? This point here. Can you see here? This entire opening here is called as urethral meatus. This opening is called as urethral meatus. Urethral meatus. Okay, urethral meatus. And that is the end of entire sperm flow. All of you understood? All of you understood? And tell me if you understood, or if you have any doubt, quickly in the chat. Quickly tell me in the chat. If you do not understand anything, tell me in the chat now. Beautiful flowchart you learned right now. Beautiful flowchart you learned. Everything passage you learned properly now. Okay. Now I will tell you some extra information which will give you a better clarity on the glands now. On the glands. See here. The first one I told you, the paired structure called as seminal vesicle. I told you one pair. Okay. Base of the bladder. Okay. Alkaline secretions. Okay, it produces alkaline secretion. Secretion contribute 60% of semen. So 60% of semen will be from seminal vesicle. Okay. It produces fructose and calcium for energy. Prostaglandins to facilitate uterine contraction. Clotting proteins are also produced in the seminal vesicle. Coagulate the semen after ejaculation also. So all of these contained in the seminal vesicle secretion. Three main things. NCERT does not tell you this. Does not tell you NCERT. I'll repeat again. Okay. Fructose and calcium, prostaglandins, clotting proteins produced by the seminal vesicle. Done. Then there is prostate. I told you, prostate is a single gland, not paired. Single like all of you. Okay, single. 20% of semen. So, and 20% of semen is produced because of prostate. Now here, students understand, secretion contains albumin and proteolytic enzyme called as fibrinolysin. Fibrinolysin is present in the secretion of prostate. This is the extra point I've told you today. Extra point. You can note it down. They'll not ask you, but you have to learn it in the MBBS. Because all of you, I know, next year you're sitting in exam, next year you'll be in your college. That time you'll remember all this. That time you'll remember, yes, Basva sir told me fibrinolysin is produced in the prostate. Okay. Last one is called as Cowper gland. Now, what is this Cowper gland? Not given in NCERT. Let me tell you. The Cowper gland is the other name of bulbourethral gland. It is also called as bulbourethral gland. Now, what is the main function? The main function of bulbourethral gland is to produce lubrication during intercourse. Okay. Also produce alkaline secretion. Okay. So with that, all the glands is over. All the glands is over with that. Okay. All the glands are done. Now students, I'll teach, this is the entire part I told you. Glans penis here. Swollen end is called as the glans penis. Opening is called as urethral meatus. Entire thing covering has a foreskin, all called as prepuce. Now here students, I told you, there are certain erectile tissues. The tissues which makes the penis erect. There are actually two types of erectile tissues. Two types. Not given in NCERT, I'm telling you again. The first one is called as corpora cavernosa. Corpora cavernosa, this entire thing, two columns of corpora cavernosa, two columns. Then a single column of corpus spongiosum. Corpus spongiosum, a single column here. Can you see? Single column of corpus spongiosum, single column. Two columns of corpora cavernosa. Two different types of erectile tissues. Again, not given NCERT, extra information for all of you today. Because I believe in, today I have decided, I was always holding it back. Like, it's fine. My students need to learn all this. Okay, because someday you will learn it. Yes or no? Okay. Now here is a small question to all of you. I hope it is visible. I hope is the question visible to all of you. Let's see who can tell the answer first. Let's see who can tell the answer first now. They're asking you the flow chart. Arrange the following male sex axillary ducts in the correct sequence for the transport of sperm from the testis. Okay, what is the correct sequence? Okay, what is the correct sequence? Tell me, students, quickly. What is the correct sequence? Students, quickly smash the like button. What is the answer? Quickly. See here. The first, where it goes, tell me students, the first one, it is coming from where? The first, seminiferous tubule. After seminiferous tubule, it goes to rete testis. Yes or no? It goes to rete testis. After, tell me. After testis, tell me where it goes. Vasa efferentia. Vasa efferentia. Then epididymis. Epididymis. Then vas deferens. Finally, last it goes to ejaculatory duct. Then it goes to ejaculatory duct. Right. First, rete testis. See, rete testis, efferentia, epididymis, vas deferens, ejaculatory duct. Rete testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct. What is the answer? What is the answer, students? What is the answer? Option number 3, right? Yes. Option number 3. Okay. Now, what exactly is semen now? What exactly is semen? Now, semen is actually made up of two main components. It is made up of two main components. The first component is the sperm. Sperm is coming from the seminiferous tubule. The second component is the seminal plasma. Now, you might ask me, sir, what is the seminal plasma? What is the seminal plasma? Just now you learned, right, 3 different glands. The three different glands such as Cowper gland, prostate gland, seminal vesicle gland. All the three glands will produce something. The production, the secretion of three glands is part of seminal plasma. It is part of seminal plasma. The secretion of these three glands plus the sperm is called as the semen. Plus the semen is called as the semen. Clear all of you? Clear? Alkaline nature, ejected from the penis during ejaculation. Till here it is very simple and good. Okay. Now students, till here, anyone, any doubt, shoot in the chat. I will take up any doubts. Anyone? Anyone, any doubt? Anyone, any doubt till now? Whatever we learned till now, students, we have learned about the entire male reproductive system. All the ducts, all the glands, okay, all the ducts and glands we have learned right now. Okay. Yes? Anyone, any doubt? Quickly tell me, ask in the chat. No doubt? Yes? No, Janani, you don't have to understand. See, I'll tell you what exactly is given in NCERT. I'll show you. Wait. I'll show you exactly what is given in NCERT. You will only laugh. Okay. 1 second. Is the board visible, students? Is the board visible to all of you? Is the board visible to all of you, students? See here. See now. What is present in NCERT? I'll tell you. Male axillary glands includes a pair of seminal vesicle, prostate, bulbourethral. Secretion of these glands constitute the seminal plasma. Okay. I told you three things: fructose, calcium, enzymes. So NCERT is telling you only three things. NCERT is telling you only three things. Do not, they will not ask you the entire thing which I told you. What I told you was entire detailed version which might be useful to you somewhere. But NCERT is telling you only three things. You can remember three things, right? First one is your fructose, calcium, enzymes. Three things you have to remember. That's all here. That's all. Secretion of bulbourethral gland also helps in lubrication. That is all, students. We learned about foreskin. We learned out this entire thing was done. Entire thing is done. Okay. Now, Avengers, it is always variable. See, if you refer to any standard book, some of the books says 20%, some books says 75 or 25%. So that margin, they will never ask you the difference in margin. Do not worry. Okay. So I refer to the book, standard books, which told 20%. 25% can also vary. Okay. Now, next concept is called as the sperm. Now, all of you know the entire sperm can be divided into three main parts. The first one is called as the head region. Then there is middle piece and tail. So, head, middle piece and tail. Head, middle piece and tail. Now, what is the head region? First of all, students, there is a very famous question here. The famous question is that what type of nucleus do you find in the case of sperm? Always remember, the nucleus is haploid in nature. So if it is haploid in nature, students, if it is haploid, can anyone tell me how many number of chromosomes are present here? Haploid means 23 chromosomes. Very simple, yet many students go wrong here. Many students go wrong here. Okay, 23 chromosomes. Now here, can you see here? The entire thing is covered by a outer layer. It is completely covered by outer layer, is called as plasma membrane, is the outer layer. Apart from that, there is a cap-like structure here. Cap-like structure here called as acrosome. Now you might ask me, so what is this cap? What is there in this cap? This acrosome right here has a lot of enzymes in it, which we will learn later on when I'll teach you about fertilization. Okay, acrosome is here. Okay, acrosome. Okay, then there is middle piece. Middle piece. Middle piece contains a lot of mitochondria. Why? Because mitochondria is powerhouse of the cell, provides energy. Then there is a large tail here, whip tail. Tail helps in the sperm to move. Helps him move. Okay. Now students, a big question will be in your mind right now. Are these notes good? The best notes you can find. The most detailed notes you can find today anywhere. Okay. So these detailed notes, if you want, everything will be available on the Telegram channel. All of you know my Telegram, right? This is my Telegram. But for Sir Biology, you'll get my Telegram, you'll get the notes. Okay. Now, students, that was one type of question which you can ask. Now, one more type of question which you can ask is the hormonal control. And here students get difficult, or students don't find it easy to remember the hormones. I will give you an easier method. Listen to me now. Okay, what is the entire hormonal control? How is the hormonal control even coming here? Okay, understand, all of you, understand. Okay, the hormonal control is very simple. First of all, there is hypothalamus, which will stimulate the anterior pituitary. Okay, it will stimulate the anterior pituitary. Listen to me very now, carefully, all of you. Anterior pituitary is there, right? Anterior pituitary will act on two gonads. That is, it will act on FSH. It will act on LH. It will act on FSH and LH. Now the confusion here is very simple. Which one will act on Leydig? Which will act on Sertoli cell? How do I remember it? Very simple trick, listen to me. L for luteinizing hormone, follicular stimulating hormone. The luteinizing hormone, the L, luteinizing hormone, L, and also Leydig cell. It will act on Leydig cell, LL, you can remember. Luteinizing hormone will affect the Leydig cell, act on Leydig cell. FSH, follicular stimulating hormone, S, Sertoli cell, act on Sertoli cell. Now students, remember, Leydig cell produce what? Leydig cell will produce the androgen later on. And this, yes, Sertoli cell will take part in spermiogenesis. Now, will they ask a question here? Yes, they can. Listen to me. See this question. This is a NEET PYQ. I don't know which year, but it has been asked before. This right here is a NEET PYQ. Okay, see this question, all of you. I think it was the last year, right? I think this one is a NEET 2024 PYQ, I believe. 2024 PYQ. See here. GnRH, LH and FSH. LH and FSH. Okay, L and FSH. L will act on what, students, tell me? L for Leydig cell. S for Sertoli cell. This will help in spermiogenesis. Tell the answer. Students. Yes. This is NEET 2024, right? NEET 2024 question. Tell me the answer. Now. Tell me the answer. All of you drink some water. This is my favorite water bottle. All of you know, my pink water bottle, Tupperware one. All of you drink some water quickly. Now, such question can be asked in examination. Such questions can be asked in examination 2024. This is NEET 2024. If you know the small trick, this would have become very easy to you, right? Very easy to you. Reproductive health will happen later on, not today. Okay. Now students, with that, the entire male reproductive organ is over. Now I have gone as detailed as possible to you. If I go any more detailed, it will go like this. If I teach you the different structures of epididymis, different layers of testes, all that is not required. How much ever is required for your preparation and also little bit extra for understanding the concept, that much I have taught you today. Okay. Now with that being said students, can we start right now the female reproductive organ? Can we start? Chedam is asking me disorders, they don't ask you. Disorders, they don't ask you disorders. Avengers, they never ask you disorders, never they have asked you disorders. Cryptokyrridism, erectile dysfunction, never, they don't ask you. Okay. Disorders, they don't ask you. So please schedule class at 6 p.m. It will be at 5 to late night. Okay. Disorders, don't worry. See, this is called as FOMO. Just because someone told disorders now, or all of you are like, what disorders? Do not worry, they will not ask you disorders. Okay. I told you one disorder, right? Cryptorchidism. That is all maximum they can ask you. Okay. Now students, male reproductive organ, male reproductive system was located in the pelvic region. Do you think the female will be the same place or somewhere else? The female reproductive system is also located in the pelvic region, in the entire pelvic region. Now if you look here, the female reproductive system also has something called as primary sex organ and secondary sex organ. Now students, tell me, all of you, quickly tell me in the chat. A primary sex organ is an organ which has to perform two main functions. What are these two main functions? The first function is what, students? The first function was it should be able to produce gametes, and at the same time it should be able to produce the hormones. All of you agree with me? Yes, the hormone should be produced, gamete should be produced. And the example here is the ovaries. Ovaries can produce something called as egg as gamete and hormone such as estrogen and progesterone. Okay. So here we have something called as, students, here we, again in the female reproductive system, primary sex organ should be able to produce the gametes plus the hormones. Gametes plus the hormones. The gametes, the primary sex organ is the ovaries, gametes is the egg, hormone is the estrogen as well as progesterone. Okay, as progesterone. Now then we have the secondary sex organs. Secondary sex organs play a indirect role. Secondary sex organs play a indirect role here. Can anyone tell me what is the examples here? One simple example is called as fallopian tube. What is the other name of fallopian tube? The fallopian tube is also called as oviduct. There is oviduct, then there is cervix. Cervix is there, uterus, vagina and external genitalia, right? As well as mammary glands. All of these make up the secondary sex organs. And together, primary and secondary sex organs, both are extremely important. For the development of the baby. Okay, now listen to me very carefully, all of you. Now, ova, hormones, ovarian hormones, all of you learned. Uterus is supported by ligaments attached to the pelvic wall. This line is very important. Understand this line. Uterus is supported. Can all of you see this? If this is the entire uterus here, the entire uterus is supported with the help of ligaments here. Can you see this entire thing is the ligaments called as broad ligaments. Because these broad ligaments, the ovaries and is attached to the uterus. Entire part is called as the broad ligaments. Okay, broad ligaments. Okay. Now students, we learned the TS of sperm, like TS of the testis. Similarly, if you cut the ovary what do you see on the inside? Can anyone tell me? If you cut the ovary, the entire ovary is covered by an outer layer. Okay, entire ovary is covered by outer layer called as epithelium. Germinal epithelium. Entire ovary is covered by a germinal epithelium. Now, inside the ovary, the inside part is called as stroma. It is called as ovarian stroma. Inner part is called as what? Ovarian stroma. Outer epithelium, inside part is called as stroma. Okay. Now listen to me very carefully. The inner stroma is actually divided into two parts. The inner stroma is divided into outer part called as the cortex, middle part called as medulla. So outer cortex and inner medulla, these are the two parts of the stroma. Two parts of the stroma. Now sometimes they can ask you, what is actually present in the outer cortex? In the outer cortex, there is development of follicles. Can you see here? The follicles are developing in the outer layer. Now, what is present in the inner part? That is inner medulla. In the inner medulla, there is blood vessels, nerves, as well as the lymphatic vessels. So, outer part is called as epithelium, outer layer. Inner part is called as stroma. Stroma is divided into two parts. Outer cortex, inner medulla. Outer cortex. That is the entire structure of the ovary. All of you understood? All of you understood, students? Any doubt there? Should I repeat again? Ask me in the chat quickly. It doesn't get more simple than this. It's so simple to understand. The structure of ovary or the layers of ovary. Epithelium, stroma, divided into two parts, outer cortex, inner medulla. Okay. Now we come to the axillary ducts. The axillary ducts or axillary organs, the first one is called as oviduct. Can you see here? This entire thing is called as a oviduct. The other name of oviduct, is also called as fallopian tube. Do not get confused. Same meaning. Okay. Now students, if I cut the fallopian tube and if I tie it, what do I call that? If I cut the fallopian tube and I tie it off? The procedure is called as what? Can anyone tell me? Okay. Now the entire fallopian tube, students, listen to me very carefully. The entire fallopian tube is divided into three parts. Infundibulum, ampulla, isthmus. See here, diagram is very important. Let's decode a diagram now. This entire thing is called as the fallopian tube. Okay. Now here, the closest part to the ovary is called as infundibulum. The closest part is called as infundibulum, which is cone shape. Cone shape infundibulum. Can you see cone shape infundibulum? At the end of infundibulum, there is a finger-like structure called as fimbriae. Okay. Now this fimbriae right here, helps in picking the egg. From the ovary. If the ovary is releasing the egg every month, one egg from each side. Like if this month this one produces egg, next month this will produce the egg. Okay. The egg is picked up with the help of fimbriae, the finger-like structure. Then it goes to a cone-shaped structure called as infundibulum. From here, the egg will travel to a much more wider part called as ampulla. Ampulla is much more wider in shape. And the interesting part here is, students, let me tell you. This ampulla right here is the region of fertilization. The fertilization takes place in the ampulla region. Okay. Now, from the ampulla, it goes to a much more narrow area called as isthmus. Isthmus is a narrow area called as isthmus. And that entire thing is called as the fallopian tube. First one is infundibulum, ampulla, isthmus. Cone shape, wider area, isthmus is a narrow. Finger-like structure called as fimbriae helps in picking up the egg. Okay, that entire part is called as the fallopian tube. Now, from fallopian tube, students, here the egg travels to a inverted pear shape called as uterus. This entire thing is called as the uterus. Okay, let's get to that. This is the fimbriae, infundibulum, ampulla, isthmus part. Isthmus. Okay. Now, what is uterus? Uterus is also called as womb. Womb, we said womb, mother womb. Womb is the other name is called as hysteria. It is called as hysteria, which also means womb. If you remove the uterus by surgical methods, it's called as hysterectomy. Hysterectomy is removal of uterus. Okay. So it is hollow, muscular structure, size, shape of inverted pear, lies in the pelvic cavity. Simple lines of NCERT, all of these are. Okay. Now this is not given in NCERT. This is not given in NCERT. That is the entire classification of uterus. See here. The above part is called as fundus. This is mentioned in the NCERT. It's called uterine fundus, dome shape. Then there is a much more narrow part, that is the cornus, corva. That is the upper corners here. This one, upper corners. Then there is the main part, which is divided into perimetrium, endometrium, myometrium, main part. Then the lower part is called as the cervix. Cervix goes down. This is the entire uterus. Entire uterus is right here. Okay. Now, that told you the main body, right? The main body. The main body of uterus has three main layers. That is outer perimetrium, middle myometrium, inner endometrium. For example, if you see on the diagram, here also, the outer part, can you see the outer part? The outer one is called as perimetrium, middle myometrium, and the inner part, this one, can you see the inner part is called as the endometrium. Now, every single layer has specific properties, right? That you should understand now. Very simple it is. First is the perimetrium. It is the outermost layer. Simple squamous epithelium. That's all. Nothing you should remember. This is also not given in NCERT. Do not have to remember these 2 points also. Just remember, perimetrium is the outermost layer, thin layer. Now, second one is called as myometrium. Myometrium is the middle layer. It is the middle layer. Listen to me very carefully, the famous point here. Myometrium is the layer which undergoes changes during childbirth. Very important point. During labor and childbirth, coordinated contraction of myometrium responds to the oxytocin. Parturition, right? It is the myometrium layer which undergoes changes. Myometrium. Now the inner layer is called as endometrium. The famous endometrium which undergoes cyclic changes during menstrual cycle. It is highly vascularized innermost layer, first point, rich in glands and undergoes periodic cyclic changes. So three main layers, perimetrium, outer, middle, myo, inner, endo. Three main layers of the uterus. Okay. Understood? Very easy, it's very simple and very easy to understand. Okay. Undergo cyclic changes. You can see here also. Now, uterus was here. Uterus fundus, top part, main uterus, this one. The inner part here is called as the cervix. Can you see? This part here is called as cervix. And this is called as cervical canal. This part is called as cervical canal, and then there is vagina here. Okay. Vagina. So, if I ask you, there is one famous line in NCERT. Okay. The famous line here is, uterus opens into vagina. Uterus opens into vagina through cervix or cervical canal. Through cervical canal. Uterus opens into vagina through cervical canal here. Cervical canal. Okay, that is very important. Okay. Now students, I will teach you what is vagina right now. Okay. Okay. Now, very important point here. Sometimes they can ask you what is birth canal also. Is it different? It is different a little bit. Okay. Now what is birth canal? Understand. This cervix right here. Can you see? Cervix plus vagina. Cervical canal plus vagina together they make up the birth canal. Birth canal. Cervical canal plus vagina make up the birth canal. Okay. Birth canal. Vaginal opening has vaginal orifice, that is called as vaginal opening. Now entire vagina is covered by a membrane layer, it's called as hymen. Now hymen also they ask many questions here. Now what is hymen? Protective covering. Can the hymen, breaking of hymen indicate sexual experience? No. They say that right during the first intercourse the hymen layer breaks, but it is always not the case, because the hymen layer can even exist after even first intercourse. Or sometimes what happens is because of the horseback riding, or if the female suddenly falls, that time also hymen layer can break also. Okay, so hymen layer does not mean, if the hymen layer is broken, it does not mean the sexual experience. Okay. Now there is a famous confusion. Is there. Famous confusion. Okay. Sir, what is external genitalia and what is this entire structure? How is this entire structure different from external genitalia? What is vulva? Like, can anyone tell me what is vulva or where is the vulva situated? Like in the case of male, we can easily see the penis, as well as we can easily identify the glands as well as the ducts. Now here we understood the primary sex organ, secondary sex organ, but where is the external genitalia? Like where is it located? And are there any points here? Yes, there is. The main reason why many students don't understand what is pons pubis, what is labia majora, what is labia minora, what is clitoris, is because this diagram you cannot understand. You cannot understand at all here. It is very difficult to understand the entire female external genitalia with the help of this diagram, because it does not show anything or does not explain anything. All of you agree? All of you agree with me? Yes or no? Yes? Drink some water, student. Drink some water. All of you agree? You cannot tell which is labia majora here. You cannot tell which is labia minora. What is urethra here and where is vaginal orifice? Nothing can be seen here. It is very difficult to see here. Okay, very difficult to see, and you cannot really understand here. So this is the diagram which you need to focus on and understand today itself. Okay. Now if all of you can notice here, students, all of you need to focus here. This entire structure is called as the female external genitalia. How there is male penis, there is the female external genitalia is called as vagina. It's called as the vulva. Vagina was the inner part. Vagina is the inner part. External genitalia is called as the vulva. This entire thing is called as vulva. Now here all of you listen to me very carefully. Entire vulva has the outer coating. Can you see the outer part? This outer part is called as mons pubis. Mons pubis is the layer with thick cushion layer, thick fatty layer with hair on the hair here. Thick fatty layer with a lot of hair. Now here if you notice here, there is something called as labia majora, which is a large flap here. That is labia majora. Labia majora is the around big flap here. Now inside labia majora is the labia minora. Majora on the outside, minora on the inside. Understand now, understand all of you. Labia minora, it surrounds on top, it goes on the top, and the top portion of labia minora is the clitoris. All of you see this? Labia minora is going on top, minora is top part is called as the clitoris. And listen to me very carefully. Clitoris also has erectile tissues. Like how we learned before, remember? In the penis there was erectile tissues. Similar to that, erectile tissues are also present in the case of the clitoris. Okay, clitoris. Okay. Now here students, many students who do not know, there are actually two openings here. See, in the case of male, there was something called as urethra. Urethra was a common opening for release of semen and urine. Urogenital tract, I told you there. But in the case of female, there are two separate openings here. Two separate openings. One is called as vaginal opening right here, where the childbirth happens, the bleeding happens, everything. But there is urethral opening. Can you see? There is a separate opening called as urethral opening through which the urine is released. So in the case of female, two separate openings. In the case of males, single opening for both. This is a small thing which usually is skipped by many teachers. Okay. So this is the information for all of you. Mons pubis is the anterior most part, fatty tissue layer covered by skin and pubic hair. Mons pubis on the outside. Mons pubis. Okay. Labia majora, I told you. Labia majora. Two large fleshy skin folds, boundary of the vulva, covered by pubic hair partly. It contains numerous sebaceous glands. Here. Yeah. Sebaceous glands. This is labia majora on the outside. Inner to labia majora is labia minora. Two small fleshy skin foldings lie under the labia majora. So under labia majora is labia minora on the inside. Okay. Now clitoris, finger-like structure present at the upper junction of two labia minora. You can see here. The two labia minora is going on top. Upper junction of two labia minora is the clitoris. Okay. And clitoris also contains the erectile tissues. That is the entire female external genitalia called as vulva. Not this diagram. Okay. Here you cannot understand anything. This diagram very difficult to understand. Okay. So I hope all of you understood. Yes or no? All of you understood? Target 400. Yes, target is 400 now. 400 likes. If you are able to understand the detailed version today, quickly smash that like button right now, students. All of you. Gopika ma'am is also here. I feel Gopika ma'am ate the dinner also now. Yes, all of you done? Yes, clear all of you? Clear? All of you understood? That is the entire female external genitalia. Okay. All of you clear? Yes. Now, here I can teach you an extra concept also. Okay. This middle part between the two labia minora, this entire part is called as vestibule. It is called as vestibule. Okay. Okay. It's called as vestibule. Generally, around the external genitalia, around the entire under the female reproductive system, did you notice there was no glands till now? See, in the case of male reproductive system, we spoke about seminal gland, we spoke about Cowper gland, bulbourethral gland, we spoke about prostate gland. But here we did not speak about any glands here, because the glands are present actually in the vestibule part. There is two different glands here. Okay, the two different glands here is Bartholin gland and as well as Bartholin gland is present near the vaginal opening. Near the vaginal opening is the Bartholin gland. Okay, Bartholin gland, and there is also paraurethral gland. Paraurethral gland is actually present near the urethral opening. So there are actually glands present here also. Okay, it's just that usually teachers don't tell. Okay. Now students, with that we come towards the end of the reproductive system, the male and female both. The next concept is called as the mammary glands. Now you might ask me, sir, is mammary gland present in every mammal? Female? Yes. Every female mammal has mammary glands without any exception. Similarly, that's why we say, right, platypus. Platypus doesn't look like anything, doesn't look like a mammal in many cases. But it does have the functional mammary glands in the female. And that is why even the platypus is placed under mammals because of the functional mammary glands. Okay, functional mammary glands. Okay. Now you might ask me, sir, what exactly is present in the mammary glands? Right? What exactly is present in the mammary gland? Listen to me very carefully, all of you. Okay. This entire mammary glands has two main things: glandular tissues and variable amount of fat. There are two main things here. The first one is called as the glandular tissues is present here. This entire thing is called as glandular tissues, and there is fat. Two main things are present here. Okay. Now if you look at the glandular tissues, there is something called as mammary lobes. It is made up of 20 to 15 mammary lobes. Entire thing is made up of mammary lobes. Okay. The glandular tissue of each breast is divided into 15 to 20 mammary lobes. And inside the mammary lobe, there is something called as alveoli. And inside alveoli, there is milk production and also storage of milk. Two main points. Okay, so production of milk and storage of milk, both is happening in the alveoli. Okay, both is happening in the alveoli. The cells of alveoli secrete milk, which is stored in the cavities of alveoli. Okay. Now, what type of question you can expect here? The question is very simple here. The question which you can expect here is the flow of milk. Okay, the flow of how is the milk being transferred. I told you inside alveoli the milk is produced. Now from here the milk is transferred to the mammary tubule. From mammary tubule it goes to mammary duct. From mammary duct it goes to mammary ampulla. Okay. From mammary ampulla it goes to the lactiferous duct. Finally from lactiferous duct to the nipples, where the milk is ejected out. This is the entire flow chart. This is what they'll ask in examination. Nothing more than this they'll ask you, students. Nothing more than this they will ever ask you. This entire flow chart. So all you need to remember here is mammary glands is present in every single female primate. That is in the case of mammals. Okay. It is made up of two things: fat, variable amount of fat, and glandular tissues. Glandular tissues are present in the form of mammary lobes, 15-20 mammary lobes. Inside mammary lobes there is alveoli where milk is produced. From alveoli the milk is transferred to mammary tubule, mammary duct, mammary ampulla, lactiferous duct, nipples. That is all you need to know about this entire flow chart. That is all. Nothing more than this. Okay. This is a diagram given your NCERT also. Okay, diagram given your NCERT. Now students, one question is here on the board. Let's see who can solve. Vas deferens receives a duct from seminal vesicle and opens into urethra as ejaculatory duct. Okay, makes sense. The cavity of cervix is called as cervical canal. Okay, cervical canal, which along with vagina forms the birth canal. Okay, makes sense. Both make sense. Yes or no? Yes or no, all of you? Both are true? Both are true. Yes, both are true. 1 and 2 are true. Okay. Next question. Which of the following is not a component of fallopian tube? Isthmus, it is. Infundibulum, it is. Ampulla, it is. Uterine fundus is not. Uterine fundus is part of the uterus. Okay. Next question. Okay, I'll give you time. Read the question and tell the answer now. FSH acts on ovarian follicles. True. Follicular stimulating hormone. Follicles, yes, in female, and Leydig cells in male. Students, S for Leydig cells or S for Sertoli cell? S for Sertoli cell. So this becomes wrong, yes. Growing ovarian follicles secrete estrogen in female, while interstitial cell, this I have not thought, this is correct, while female while interstitial cell secrete androgen in human male. Correct. This is correct. This is wrong. So A is false, R is true. A is false, R is true. Yes. Okay. So with that, students, we finished the major chunk of today's lecture, that is the entire male and female reproductive system. Done. Done. Full done. Okay. Now we'll start with spermatogenesis. Before that, all of you drink some water and we'll start. Okay. All of you drink some water, water break. Yes. Now, there's a concept called as spermatogenesis and oogenesis. Spermatogenesis and oogenesis falls under a category called as gametogenesis. Gametogenesis. Both are separate events. Spermatogenesis takes place in the case of male, oogenesis in the case of female. Okay, female. Now, students, I want everyone to open a book and write down the flow chart which I am about to give you. Can we start, all of you? Yes. Can we start, students? Students, I should not get tired. You should not get tired. Class will go on until the chapter is over. Whatever happens, we have made a commitment here that entire reproduction unit in one shot. Right, entire reproduction unit in one shot, we have made a commitment. And the deal was, if you attend the lecture till the very end, you will benefit. And if you keep up your deal, our deal, it will happen. Okay. So all of you open a book, smash the like button, and let's start right now. Let's start. Okay. The process of production of sperm in seminiferous tubule, inside seminiferous tubule of testis during puberty is called as spermatogenesis. This much all of you know. This much all of you know. Okay. Now, this is extra. Listen to me. Spermatogenesis is divided into two parts. Spermatocytogenesis, spermiogenesis. This is where spermatids are produced. Spermatids gets converted to sperm. That is called as spermiogenesis. You don't have to worry about it. I will teach you right now. I will teach you the entire thing right now. I will teach you the entire thing in 5 minutes. So I want everyone's ultimate focus now. Can we start? Can we start now? Ultimate focus, high energy. Let's go. 5 minutes, ultimate focus. You will learn the spermatogenesis and never forget in your life. Never forget in your life. Okay. Now, let's start. The first thing you need to understand here is spermatogenesis. It takes place during puberty in male. Puberty in the case of male. Now, if all of you remember, inside seminiferous tubule, there were two types of cells. What were the two, students? Inside seminiferous tubule, there were two types of cells. One was called as the Sertoli cell. The other one was called as male germ cell. Other one was called as male germ cell. The male germ cell was also called as spermatogonia. Spermatogonia. Spermatogonia was diploid in nature. It was diploid in nature. Spermatogonia. Students, let me write towards this a little. Let me erase, write this side little, so I can cover the entire thing. Perfect. Okay, spermatogonia. This was diploid in nature. Now And this spermata undergoes initially, this spermatogonia initially undergoes mitosis. Mitosis. Now students, tell me, when it undergoes mitosis, will there be a change in any ploidy? Diploid to haploid? No, no ploidy change. So this spermatogonia will increase in number. If this was diploid, it will just increase in number 2n and 2n. Now students, understand, one such oogonia starts to show growth. It starts to show growth and increases in size. Increases in size, and this structure right here, is called as, it is called as primary spermatocyte. It is called as what? Primary spermatocyte. Primary spermatocyte. Now this primary spermatocyte, students, this primary spermatocyte right here undergoes meiosis. It undergoes meiosis. This was diploid here. Diploid. It undergoes meiosis. When it undergoes meiosis, the primary spermatocyte diploid, when it undergoes meiosis, it produces two haploid structure. And why two, students? Because I am saying it is meiosis 1. Because all of you know there is meiosis 1, meiosis 2. Meiosis 1, 2 haploid gametes. N and N. This is 22 plus X, 22 plus Y. Chromosomes. Haploid, right? Haploid. Okay. And these structures right here, they are called as secondary spermatocyte. They are called as secondary spermatocyte. Secondary spermatocyte. Okay. Now, these secondary spermatocyte, students, listen to me very carefully, all of you now. Now, this secondary spermatocyte undergoes meiosis 2, obviously. Meiosis 2. When it undergoes meiosis 2, it produces 4 haploid structures. 4 haploid structures, and these structures are called as spermatids. Each one is, these two are 22 plus X, these two are 22 plus Y. Spermatids. Now tell me, students, do they spermatids, do they look like sperm in any angle? Look, when nicely you look at it, when look you give, tell me, do they look like anywhere close to even sperms? They do not. They do not at all. Okay. Now this spermatid, students, this spermatids undergo one more process called as spermiogenesis. Spermiogenesis. When spermatids undergo spermiogenesis, it produces 4 haploid gametes. They are called as spermatozoa or sperm. That is the entire gametogenesis, that is spermatogenesis. I'll give you a small recap one more time. Listen to me very carefully, all of you. Very simple, it is very simple. In one sheet I made you understand. First is called as the male germ cell, spermatogonia diploid. Undergoes mitosis, increase in number. One such spermatogonia undergoes growth and becomes the primary spermatocyte, diploid. It undergoes meiosis 1, produces secondary spermatocyte which is diploid, which is haploid. Secondary spermatocyte undergoes meiosis 2, produces 4 spermatids. Undergoes a process called as spermiogenesis to produce the sperm. That is all. That is all the spermatogenesis is. Now here understand, students, once the sperms are produced, they are not released as such. All of this sperm, the head is embedded in the Sertoli cell. Head is embedded in the Sertoli cell. What happens is, head is embedded in the Sertoli cell. Head in Sertoli cell. The head is embedded in the Sertoli cell. Now if the sperm has to be released from there, if the sperm has to be released from the seminiferous tubule, sperm released from the Sertoli cell, actually Sertoli cell itself, released from Sertoli cell or seminiferous tubule, release from seminiferous tubule. The release of sperm from seminiferous tubule is called as spermiation. It's called as spermiation. So what happens is, the head of the sperm is embedded inside the Sertoli cell. Now Sertoli cell and spermatogonia both are present where? Seminiferous tubule. Now if the sperm are released outside seminiferous tubule, spermiation. That is called as spermiation. Okay. Now I'll move aside. You can take a screenshot, write it down. That is better. Okay. Spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid. Spermiation, mature sperm cell. See, spermiogenesis. Differentiation of spermatids into spermatozoa, tid to zoa. That is called as spermiogenesis. Sperm head becomes embedded in the Sertoli cell and finally released from the seminiferous tubule. The process called as spermiation. Spermiation. Can you see here? This is how the sperm head is embedded in the Sertoli cells. From here, they are released outside. The process of release is called as spermiation. Spermiation. See, the question comes like this for all of you. Question comes like this. The release of sperm from seminiferous tubule is called as spermiation. True. Okay. True. Spermiogenesis is the process of formation of sperm from spermatogonia, students. Is it true now? Is it spermatogonia or is it, students? Or is it spermatid? Tell me. Tell me now. Is it spermatogonia or is spermatid? Spermatid, right? So statement number 2 is wrong. It is wrong. Statement number 2 is wrong. Okay. So that was entire thing was spermatogenesis. Now students, tell me honestly, how many of you find difficulty in understanding oogenesis? Raise your hands. Honestly, raise your hands. Do not lie to me, okay? Do not lie. Tell me how many of you face difficulty in understanding oogenesis? Raise your hands. I'll give you 10 seconds to be honest. And I'll drink some water in that gap. Raise your hands. All of you, don't be honest. Because oogenesis is a concept which, if it is not thought properly, it can become very confusing. It can become very confusing if it is not thought properly. And we have to go in a very stepwise manner. Let's see, here we went stepwise, right? Here we went stepwise. Properly went stepwise here, right? Every single step by step we learned. Similarly, I can teach you oogenesis in a stepwise manner. Ready, all of you? Ready? Yes? Ready, all of you? Can we start oogenesis now? All of you have difficulty. I started hating my life after studying oogenesis. Oogenesis is so beautiful concept. Trust me, Avengers, sir. I hope not the game, the movies. Okay. Once you learn from me oogenesis today, and if you write down, you will never forget the concept it holds. It is an amazing concept, Very easy concept. Okay, it is only difficult because oogenesis is, if you read NCERT, right, you will not understand. Oogenesis, half is present in oogenesis, half of oogenesis is present where? In fertilization. That is why it is difficult to understand, because half information is present in oogenesis, half of the information is present in fertilization. That is why it is difficult to understand. But if you focus from the next 10 minutes, if you focus right now for the next 10 minutes, I will teach oogenesis. Ready, all of you? Ready? Ready? All of you. Now, what is oogenesis first of all? The process of formation of mature female gamete is called as oogenesis. Okay. Step by step I'll teach you right now. Just focus with me. Okay. 3, 2, 1, let's go. How spermatogenesis starts at puberty. Spermatogenesis starts at puberty. Oogenesis starts at embryonic stage. First difference. Starts at embryonic stage. It starts at embryonic stage. Okay, starts at embryonic stage. Okay, first important point, listen to me, all of you now. Ultimate focus, maximum effort now. Okay, maximum effort. First we have the primary sex organ here. The first germ cell. The first germ cell, how we had spermatogonia, here we have the oogonia. Let's take oogonia here. Different color. This is the oogonia. Okay, this right here is the oogonia, which is diploid in nature. Should I write this one second? Student, there will be no space. This is very large. It'll make a lot of difference right here. Oogonia, which is diploid in nature. Diploid. This is the 2-1 oogonia. Okay. Now again, initially this oogonia undergoes. Initially, the oogonia undergoes mitosis. Undergoes mitosis to produce more in number. Undergoes mitosis. There is no change in ploidy, no change in ploidy. Okay, no change in ploidy. Now students, understand here. One such oogonia starts to grow and differentiate and become large here. One such oogonia differentiates and become larger, and such oogonia which differentiated and became larger is called as, it is called as the primary oocyte. It is called as primary oocyte. Now if I ask you, what is the ploidy here? The ploidy is going to be diploid, 2-1. Ploidy is going to be 2-1. Ploidy is going to be 2-1. Okay. Focus very well right now. Okay. Now this primary oocyte undergoes, a process called as meiosis. Meiosis 1. It undergoes meiosis 1. Primary oocyte is undergoes meiosis 1. Okay. The problem here is this primary oocyte which starts to undergo meiosis 1, it gets suspended. It gets suspended. Suspended at prophase 1. Prophase 1, diplotin stage. Diplotin stage. Till here I hope it is clear to all of you. What we just now did was, I'll give a small recap. First, starts with oogonia, 2-1. Undergoes mitosis, 2 of them. No change in ploidy. One such oogonia differentiates, becomes larger, grows in size, called as primary oocyte. Called as primary oocyte. This primary oocyte undergoes, right? Primary oocyte undergoes, right? Meiosis 1. Meiosis 1, it gets suspended at prophase 1 diploidine stage. This entire event is happening before the birth of the child. Very important. Okay. All of this is happening. All of this happening at embryonic stage. All of this is happening at the embryonic stage. Embryonic stage. All of this is happening at the embryonic stage. Oogonia, primary oocyte, suspended at prophase 1. Now what happens? Childbirth. After this is the childbirth. Child will grow into this, right? Birth of the child. Birth of the child. It undergoes childhood. And finally, when the female child reaches puberty, this meiosis 1, which was arrested at embryonic stage, finally completes. The first menstrual cycle, menarche. The first menstrual cycle in the females, the first periods in females is the when the primary oocyte, meiosis 1, is completed at puberty. So what exactly happens at puberty? At puberty, students, at puberty, meiosis 1 is over. At puberty, students, understand. At puberty, this meiosis 1 is completed. At puberty, this meiosis 1 is completed, and it results in secondary oocyte as well as the first polar body. So this primary oocyte is giving rise to secondary oocyte and first polar body after meiosis 1. Okay. Now you tell me, students. Any doubt till now? Students? Any doubt till now? Yes? Now, listen to me very carefully. This secondary oocyte is released during the ovulation. That is the most important line which you need to understand. This secondary oocyte right here is released during the ovulation, not the ovum. Ovum is never released. Listen to me carefully, all of you now. This secondary oocyte is released during the ovulation. We learn, right? The LH surge, the ovulation, everything. Listen, I'll tell you one more time. Don't worry, I'll tell you one more time. Listen to me. All of you. Focus. Okay. 1 second. Focus, all of you, focus. First, oogonia, done. Oogonia undergoes mitosis to produce the increase in number. One such oogonia enlarges primary oocyte. This primary oocyte undergoes meiosis 1. But it gets suspended at prophase 1 diploidine stage. Now it is suspended until when? It is suspended until the female child reaches puberty. Until puberty, suspended. Can you see here? Entire event until suspend, embryonic stage. Childbirth, childhood, puberty. Once the female child, listen to me carefully, once the female child reaches puberty, once the female child reaches puberty, this primary oocyte which had started meiosis 1 finally gets over. Where? At puberty. At puberty, this meiosis 1 is completed. As a result of meiosis 1, primary oocyte divides into two structures. Primary oocyte divides into two structures. One is called as secondary oocyte. Second is called as first polar body. Understood till now? All of you understood till now? Tell me yes or no in the chat, because from here I will teach you the second part which is connected to fertilization. All of you understood right now till now? All of you? Yes. Meiosis 1 is started at embryonic stage, suspended at prophase 1 diploidine stage, suspended. This meiosis 1 is only completed when? Students? Meiosis 1 is only completed when the female child reaches the puberty. At puberty, what happens? Meiosis 1 is completed, primary oocyte will give rise to secondary oocyte and first polar body. Now if I ask you, this secondary oocyte right here, which is produced at the puberty, is because of first menarche. So during menarche, usually also, or during menstrual cycle, every single month, during ovulation, what is being produced? During ovulation, secondary oocyte is being produced, not the ovum or the egg, technically speaking. So every single month in the female, secondary oocyte is being produced. That's all. Now students, this secondary oocyte here starts to undergo meiosis 2. It starts to undergo meiosis 2, but again arrested. Again it is suspended at metaphase 2. So this suspended stage is technically being released. This suspended stage is technically being released during ovulation. Listen to me very carefully. All of you. Listen to me very, very carefully. This secondary oocyte okay, this secondary oocyte will finish meiosis 2. It will finish meiosis 2 when secondary oocyte comes in contact with the sperm. When secondary oocyte comes in contact with the sperm, that is when the meiosis 2 is technically over. So if I tell meiosis 2 generally gets over only in the females where fertilization is happening. Otherwise, it is not getting over at all. Otherwise, it never gets over. Okay. So meiosis 2 is completed when the secondary oocyte is meeting the sperm, and it will result in two separate structures. One is called as ovum or ooted, and the second structure is called as second polar body. Beautiful, right? Beautiful process. Let me recap one more time. Let me recap the entire thing one more time. Understand it today itself. Do not take this backlog anywhere else. Understand it today itself. Write and learn this. Okay. See this. First is the oogonia undergoes mitosis. Two different structures. One is called as primary oocyte. Primary oocyte starts to undergo meiosis 1, but it gets suspended at prophase 1 diploidine stage. Suspended. Now this structure right here, suspended until when? Until the puberty. At puberty, what happens? At puberty, the first meiosis is over, which results in the secondary oocyte, first polar body. Now this secondary oocyte is released during ovulation, which starts to undergo meiosis 2, but again is suspended at metaphase 2. metaphase 2. Now this secondary oocyte finishes the meiosis 2. It finishes the meiosis 2 when the secondary oocyte comes in contact with the sperm. That is when it is over. When it gets over, when meiosis 2 gets over, it releases in the otid or ovum or the second polar body. Now here understand students, understand here. The second polar body is technically produced where? What is the location of its production? Second polar body is produced in the ampulla. Why? Because that is where fertilization is happening, in the ampulla region. While the first polar body is produced inside the ovary. Beautiful concept, right? It is produced inside the ovary because all this is happening inside ovary. Fertilization is happening where? Fertilization is happening in the fallopian tube, ampulla region, second polar body. So such questions can asked. Such question can be asked if they become very technical. If they become very technical. Okay. The function of polar body is not known. Function of polar body is not known. Okay. Now students, if you think this much is enough to understand oogenesis, you are wrong again. Oogenesis is very difficult to understand because oogenesis has two things happening simultaneously. Listen to me very carefully. Oogenesis has two things happening simultaneously. When you connect the two things, then only the oogenesis finishes. Listen to me now. Along with oogenesis, there is also something called as development of follicle. Development of follicle. There is something called as primary follicle, secondary follicle, tertiary follicle, as well as Graafian follicle. Graafian follicle. What is present in primary follicle? Inside the primary follicle, there is primary oocyte plus one layer of granulosa cells. One granulosa layer. That is primary follicle. Secondary follicle will have again primary oocyte plus more layer of granulosa cells plus new theca formation. Now what tertiary follicle? This is the most important part. In tertiary follicle, the primary oocyte is getting converted to secondary oocyte. This one here. This part here, right? Primary oocyte is getting converted to secondary oocyte. This thing is happening in the tertiary follicle. It is happening in the tertiary follicle. Primary oocyte is getting converted to secondary oocyte. And in tertiary follicle, there is a formation of fluid-filled cavity. Fluid-filled cavity called as antrum. Fluid-filled cavity called as antrum formation happens here. Okay, fluid called antrum formation. And also, the theca becomes theca externa and theca interna. Okay. Finally, in the Graafian follicle. Listen to me. Okay. Finally, in the Graafian follicle. Okay. Tertiary follicle. See, in tertiary follicle, what is happening is the primary oocyte here. This happens during puberty. Okay. During puberty, what is happening here? During puberty here, primary oocyte was getting converted to secondary oocyte. During puberty, I told you. Secondary oocyte. This thing happens inside tertiary follicle. Primary oocyte is getting converted to secondary oocyte inside tertiary follicle. Last one is Graafian follicle. Inside Graafian follicle, what happens? The Graafian follicle ruptures. The Graafian follicle completely ruptures and it releases the secondary oocyte. Secondary oocyte is released from the Graafian follicle during ovulation. And this Graafian follicle, students, during this time, the secondary oocyte has a separate layer also. Separate layer around the secondary oocyte called as zona pellucida. Now you might be wondering, sir, how is this two connected? Students, all of this happens simultaneously. It happens simultaneously. Here there is oocyte formation. Around the oocyte, follicle starts to form. So follicle is the entire thing. Inside the follicle, there is oocyte. Okay. If this is the entire thing is the follicle, inside the follicle, there will be oocyte. Okay. So from here, you can get many questions from here also. So if I ask you students, in which stage primary follicle becomes secondary follicle? What is your answer? Sorry, primary oocyte becomes secondary oocyte. Tertiary follicle is the answer. Okay. Where do we have the fluid filled cavity? The fluid filled cavity is present in the tertiary follicle. That can also be answered. Tertiary follicle. I can ask you question as which follicle gets ruptured? Graafian follicle gets ruptured. We can ask you a question like that. Okay. So many questions can come from here. See here. Follicular degenerate is genetically programmed. That is, students, once this oogone is fixed, once a female oogone is fixed, no more oogone can be added externally. The female is always born with a fixed amount of oogonia, and as the years passes by, the oogonia will keep on decreasing. Okay. And the final, and before birth, lakhs of follicles are present in both ovaries, but at puberty only 60,000 to 80,000 follicles are left in each ovary. That is, inside ovary only 60 to 80 primary follicles are left. Okay, primary follicles are left. Okay, so I want everyone to watch this lecture one more time. Okay, the external thickener is very simple. That is, there is a layer formation called as theca. Okay, the theca layer formation will happen in the secondary follicle. Okay, so in the secondary follicle, in tertiary follicle it will open up externa interna. Okay, see here, this is how the follicles will start to develop. Can you see? The follicles will start to develop like this: primary follicle, secondary follicle, Graafian follicle. So primary follicle, primary follicle will be produced at the embryonic stage only. Embryonic stage, primary follicle, right, until it's prepared. Then secondary tertiary formation happens. Can you see here? This is called as secondary oocyte is present here inside tertiary follicle. Secondary oocyte inside tertiary follicle. How can I tell tertiary follicle, students? Because of the antrum. This is tertiary follicle. Tertiary follicle has secondary oocyte. Tertiary follicle has secondary oocyte. That is very, very important point. Tertiary follicle has secondary oocyte. Okay. See here, secondary oocyte is released during the ovulation. Okay. You can get questions like this. You can get some questions like this. See this question. Okay. Primary follicle gets surrounded by more layer of granular cells and a theca and shows fluid filled cavity called as antrum. It is called as secondary follicle, students. Is it called a secondary follicle? Fluid filled cavity. Graafian follicle ruptures to release the secondary oocyte from the ovary by the process called as ovulation. This is correct. Secondary oocyte is released, ovulation. This is correct. But this one doesn't make sense. Fluid filled cavity is present where? The fluid filled cavity called as antrum is only present in tertiary follicle. Not in secondary. Okay. So first statement is incorrect. second one is correct. Okay. So I believe all of you are able to understand the beautiful concept of oogenesis. Yes, if you watch the lecture for the very first time, there's a very high chance that you're like, sir, oogenesis, I'm not able to understand little. Samajh nahi aa raha, you can tell. But watch the lecture one more time, write down the entire flow chart, write down, and then you'll understand. Okay. This one, see, at which stage oogenesis procedure initiated? You can tell me embryonic stage. Embryonic stage. How many Y chromosomes are present in second polar body in human beings? Y chromosome in the female, 0. 0. Okay. So that was the entire gametogenesis over. You go and try to solve beautiful questions. You will find so many questions on gametogenesis. You will find many questions on oogenesis. Try to solve them. Try to get this flow chart, draw the flow chart, and then you see this. You will be able to solve every single question of PYQ, any question you can take and start solving. Okay. If you learn the flow chart, if you learn the entire thing, any single question you can solve, and that is my guarantee. Okay. Now students, let's start with menstrual cycle. I think the class will easily go up till 12 o'clock. Only if you are up for it, you will watch it today. Sir, ploidy, they can ask you. Janani is asking me, what is the ploidy of secondary oocyte? You tell me, all of you, tell me. What will be the ploidy of secondary oocyte? Tell me, all of you. What is the ploidy of secondary oocyte? See, primary oocyte is diploid. It is undergoing meiosis. Primary oocyte is diploid. It is undergoing meiosis. This is going to be what now? Tell me. Tell me, students. This is going to be what now? Diploid to haploid. Haploid. All of you understood? See. Secondary, primary oocyte is diploid. Meiosis. Haploid. Done. Haploid. This is also going to be haploid. Because meiosis 1 is reduction division. Meiosis 2 is equational again. Okay, it is going to be haploid. No, reproductive health, separately we'll do it. It is a small chapter. There are hardly 1 question, 2 question you get. Okay, let's finish this entire chapter today. Okay, 1 chapter today. Because students, let me tell you, today I'll be teaching you fertilization in such a way. Today I'll be teaching you fertilization in such a way that you have never learned it. Never learned that detailed fertilization, because we always learn that fertilization is just male and female gamete coming together and over. No, it does not happen like that. There is actually a simple process going on called as capacitation, acrosomal reactions. If you have heard about these, you know how amazing these concepts are. If you have heard about what is capacitation, what is acrosomal reaction, what is this, how does the coronal radiator gets completely degraded, how do we prevent polyspermy, what is this fast block, slow block, this I'll teach you today. Okay, if you know this, if you learn this, you will say, thank you sir, I truly understand the concept right now, not just mugging up the lines of NCERT. You will understand the true meaning of the human reproduction chapter. Okay. Now students, I will teach you the entire menstrual cycle in the next 10 minutes. Okay. Yeah. Now students, understand. Menstrual cycle I will teach you with the help of entire diagram, and you will love it. Menstrual cycle. And trust me, the way I will teach you menstrual cycle, you have never learned it that way. With the help of a diagram. Everything with the help of diagram, menstrual cycle. Okay. Now, what is menstrual cycle? An average duration of menstrual cycle is about 28 days. Easy. Cyclic discharge of blood and mucosal tissue from the uterus through vagina, that is menstrual cycle. There are 2 events happening here. Uterine cycle and ovarian cycle. What are the 2 events? Understand here. Okay. The starting of menstrual cycle is called as menarche, starts around 10 years to 13 years now, it is increased, or it's a decrease now, 13 years in female. And menopause generally happens around 50 years. 50 years. Menopause means stopping of menstrual cycle. Stopping of menstrual cycle. Okay. Now students, can we start? I want ultimate focus right now. Ultimate focus. Ultimate focus. Can we start, students? Can we start? Can we start? Students, can we start now? All of you. Listen to me very carefully, okay? Listen to me very carefully. The entire menstrual cycle, there are two main events happening simultaneously. One event is influencing the other event. Okay, listen to me. There are two events happening simultaneously, right? Two events happening simultaneously. One event is influencing the other event. What do I mean by that? One event is happening in the ovary. Can you see here? Ovarian event. Here, ovarian event, first event. Second event is happening in the uterine event. So one event is happening in the ovary, second event is happening in the uterus. One event is influencing the other event. Very important line. Okay. Now, understand all of you. Two events. One is called as ovarian events. One is called as the ovarian event. Other one is called as uterine event. Okay, listen to me. Listen to me carefully, all of you. In ovaries, what is happening? In the case of ovary, there is something called as follicular phase. Follicular phase. At the same time, in the case of uterus, there is proliferatory phase. There is something called as proliferatory phase. Okay. Two events are happening simultaneously here. Proliferatory phase in the case of ovaries, proliferatory phase in the case of uterus. Follicular here, proliferatory here, uterus events. Then there is a common event called as ovulation. Common event called as ovulation. After ovulation, in the case of ovarian, there is something called as luteal phase. And in the case of uterus, there is something called as secretory phase. There is something called as secretory phase. Secretory phase. Okay. Secretory phase. Here, before the two phase, there is something called as menstruation. Menstruation. Now, if this is clear to all of you, what is happening in ovaries, what is happening in uterus, with this information, you need to understand this particular diagram. Okay. To understand this diagram, you need to know something called as the hormones. There are two types of hormones again here. The first type is called as pituitary hormones. The second type is called as the ovarian hormones. The two hormones also play a major role here. Listen to me carefully. What happens the first thing? Now tell me, students, all of you in the chat, tell me which hormone do you think is higher here? First of all, students, tell me which hormone is higher here? Do you think FSH is higher or LH is higher? All of you tell me. Which hormone is higher? First of all, tell me. Red line or blue line? You will tell me, sir, the red line is higher first of all. And I can see, first of all, FSH is higher here. Higher. Now, what is FSH? FSH stands for follicular stimulating hormone. Follicular stimulating hormone. FSH will stimulate the hormone production. Red line is on top. So, this will develop the developing follicle. secondary follicle, tertiary follicle, Graafian follicle. Okay, so FSH, follicular stimulating hormone, will stimulate the developing follicle and mature follicle we get. Now, understand. Every month there is only a single secondary oocyte produced. Every single month a single secondary oocyte is produced every single month. Very easy to understand. Every single month. Now, once the dominant follicle is obtained, there is no more the need of one more follicle. Yes. So, because of the granulosa cells here on the outside, it will send a feedback mechanism, and can you see there's a decrease in red line here now? All of you can see decrease in FSH now. Red line is decreasing, it is decreasing. Why? Because the dominant follicle is obtained, so decrease feedback mechanism, negative feedback mechanism, and the FSH decreases, decreasing, done. Now understand students, this particular developing follicle will increase the estrogen. Can you see? Estrogen is increasing. Estrogen is increasing in first. Now this estrogen will influence the development of uterus. So it goes on like this. First FSH, increase in FSH, follicle will increase estrogen. Estrogen will increase what? Development of uterus, also called as proliferation of uterus or piling up of uterus. So uterus starts to pile up right now. Can you see? Uterus is piling up. Why? Because of the estrogen. So estrogen helps in formation of endometrium layer or thickening of endometrium layer. So can you see here? The first phase is called as follicular phase. Why? Development of follicle in the ovary. It is also called as proliferatory phase. Why? Because there is proliferation of endometrium layer. That is why it is called as proliferatory in the uterus. So this is the first half. I'll repeat again, all of you. Increase in FSH, follicle development. Follicle development, estrogen production. Estrogen production, development of follicle. Development of endometrium layer. Done. Now at the 14th day, what happens? Exactly at the 14th day, there is an increase in LH, there is increase in FSH. Both are increasing. And LH is in much more higher concentration. This is called as LH surge. Because of LH surge, what happens? Because of LH surge, the Graafian follicle ruptures open and the secondary oocyte is released outside. And that is called as ovulation. Can you see? This is the secondary oocyte. Secondary oocyte is released. That is the 14th day. Common point. Then what happens after that? After that, what happens? Now students, all of you tell me. Here, is the blue line on top or red line on the top? You will tell me, sir, blue line is on the top. So can I tell at this point right here, LH is very high. Okay. LH is very high. Now this LH will do what? Once the secondary oocyte is released, the remaining part is getting converted to corpus luteum. Remaining part. Okay, corpus luteum. This LH will maintain the corpus luteum. This LH right here, LH, blue line on the top, will maintain the corpus luteum. Maintaining a corpus luteum is done. Now understand. This corpus luteum will increase the level of progesterone. With that, can all of you see the level of progesterone is higher right now? The red line is increasing. And this progesterone will do what? It will maintain the endometrium layer. Beautifully this is happening. So what is happening here is LH is high. LH is maintaining the corpus luteum. Done. Corpus luteum is producing progesterone. Done. This progesterone is maintaining the endometrium layer. Here estrogen was making the endometrium layer. Progesterone is maintaining the endometrial layer thick. Maintaining is done by progesterone. Beautiful. That is why it is called as luteal phase. Why it is called luteal here on top? Over here it is called as luteal because luteinizing hormone is high. It is called as secretory phase here because the endometrium becomes secretory here, secretory. And that is the second part of the menstrual cycle. Now students, what will happen if there is fertilization? First of all, if fertilization occurs, there will be production of ovum, first of all, and then there will be implantation, zygote formation, implantation, things go on. Endometrium layer is maintained. It is maintained. Okay. Endometrium layer is maintained because of corpus luteum. Now, what if there is no fertilization? If there is no fertilization, what happens? Can you see? If there is no fertilization, LH is dropping now. Can all of you see? The LH is dropping, the blue line is dropping now. If blue line is dropping, if LH is dropping down, can I tell the corpus luteum is also degenerating now? Because LH was maintaining the corpus luteum. LH was maintaining the corpus luteum. So corpus luteum is also degenerating. If corpus luteum is degenerating, can you see the blue line is also coming down now? Blue line is coming down. It means progesterone is also coming down now. It is coming down. If progesterone is coming down, who will maintain the endometrium? No one. And endometrium is not maintained. Endometrium collapses. Endometrium collapses with all the blood vessels, secondary oocyte and mucus and every single thing, and everything shed down through vagina, and that is again beginning of the cycle, bleeding stage. And that is the beautiful menstrual cycle, how it happens. Two separate events, ovarian event, uterine event. Entire thing is maintained by 4 hormones, LH, FSH in the pituitary. Same time, ovarian hormones, progesterone, estrogen. Fluctuation happens. Because of these fluctuations, there is changes in the endometrium. Formation, maintaining. And imagine students, every single month it happens. All of this happens in the females. So that is the beautiful thing. Okay. So I hope all of you understood the entire menstrual cycle. Follicular phase, proliferatory. Luteal, secretory. Done? All of you done? Done, students? This is the entire menstrual cycle. Okay. The question comes like this. Okay. Endometrium is necessary for implantation of. Okay, this should not come here. I didn't teach you blastocyst still. We'll skip this one. Answer this question now. Answer this question. Tell me, students. Tell me. Proliferatory phase, starting part, is similar to proliferatory phase where the proliferation of endometrium happens, follicle development happens. Yes. Secretory phase, luteal phase. Menstruation, breakdown. Easy, right? Easy. Okay. Sir, will the secondary oocyte come out? Yes. Secondary oocyte is released during menstrual cycle. Secondary oocyte is released in ovulation. During ovulation. Okay. See this question. Main function of mammalian corpus luteum is to produce what? Progesterone. Only progesterone. Okay. Only progesterone. So with that, students, we come to the end of the chapter. Almost towards the end. The last segment, we learnt about pre-fertilization, gametogenesis, male and female reproductive organ, oogenesis, as well as spermatogenesis. Then we learnt about menstrual cycle. Now we come to fertilization. Now we come to the entire fertilization. Now here, students, here. It may seem very easy, fertilization, that sperm is coming, egg is coming, fusion, zygote. But it is not really that easy, because right now I will teach you how exactly it happens. How exactly happens. Are you ready for it? Are you ready, students? Are you ready? All of you. Let me check the likes now. Students, quickly smash the like button, all of you. All of you smash the like button. All of you quickly smash the like button, because right now we'll start. Right now we will start the fertilization. Start. Can you start? Now it is the fertilization. Actually how it happens, and you will, once you understand this, right, once you understand the fertilization, you will see the importance of it. Okay. Now, all of you are aware the female gamete, male gamete fuse together, fertilization, offspring. But is it easy to understand this? Yes, it is. First, there is journey of ovum. Ovum is produced in the ovary. From here it goes to the ampulla region, into the ampulla. The sperm also is released into vagina by insemination, and the sperm is going. The sperm travels, 50% goes this side, wrong turn they take, no one is waiting there, scam happens. 50% of them turn towards the region where the egg is there, and in the ampulla region they fuse. In the ampulla region they meet. Okay, and once they meet, there is fusion. Okay, now I want you to understand the difference between what is syngamy and what is fertilization. Fertilization means fusion of gametes. Syngamy means fusion of nuclei. Simple as that. Okay. Now students, understand, all of you, very simple. This is gonna be the most important part today. Okay. If you understand this next 10 minutes, what I'm going to teach you, next 10 minutes, you will say thank you. Okay. First, understand what happens. Okay. Now the sperm is entering. All of it, imagine, it's a small thing. The sperm is entering now. Now the sperm finds the egg. Now it has to attach there. How will it attach? How is the attachment even happening? Okay, that is the first point. The sperm will bind. The sperm will bind to sperm receptor. The sperm will bind to sperm receptor. But where is the sperm receptor even found? Where is it present? The sperm receptor, students, called as the Z3 molecule, called as ZP3 molecule, sperm receptor, it is present on zona pellucida. Present on zona pellucida, the sperm will bind there. Now once the sperm is binding, as soon as the binding happens, two main events are happening. As soon as the binding happens, two main events are happening. That is, the first one, understand. As soon as the binding happens, the sperm undergoes something called as capacitation and acrosomal reaction. Acrosomal reaction. Now what is capacitation, first of all? Capacitation, when is it happening? Capacitation is like evolution of sperm. The sperm becomes more motile, it becomes more vigorous, it changes its shape also a little bit. It is basically evolution. All of you know Pikachu or Charmander becomes Charizard, Pikachu becomes Raichu. Like Pokemon evolve, right? Here also sperm is evolving. The sperm is evolving and becoming much more vigorous and everything. That is called as capacitation. The second thing is called as acrosomal reaction. Now if all of you remember, students, the sperm had a cap-like structure, if you remember, right? Cap-like structure. What is that called as? The cap-like structure called as in the sperm acrosome. Acrosome has certain enzymes. Those enzymes will function right now. Those enzymes will play a role right now. Okay. Now inside acrosome, there is something called as sperm lysine. Sperm lysine. Okay. In the acrosome, there is something called as sperm lysine. Now this sperm lysine has something called as hyaluronidase enzyme. It is an enzyme. Uronidase is an enzyme. It will work on breaking the granulosa cells. Granulosa cells, there is a matrix of granulosa cells. It will completely break it down. Okay. Then there is something called as corona penetrating enzyme. That is called as CPE. This CPE will completely break down the corona radiata. Then there is something called as zona lysine, or it is called as acrosin. Acrosin will completely break down the zona pellucida layer. So do you understand? When these are called as acrosomal reaction. These are called as acrosomal reactions. The outer layers is broken down one by one. The sperm can't directly attach there, not possible. The head will open up, all the enzymes are released, all of these enzymes will start breaking down the extra layer. All will break down one layer of the egg, of the ovum. Start breaking down. If you see here, all of this layer, the outer layer, that is corona radiata is broken down, done. Zona pellucida layer is broken down completely. Plasma membrane is exposed. Once the plasma membrane is exposed, one more event happens. Understand? There is something called as, in the ovum and in the sperm. In the ovum and in the sperm, there is a specific type of monosaccharides and protein. Understand? Very, very crucial, this is point. In ovum, there is something called as fertilizin. And here we have anti-fertilizin. Fertilizin and anti-fertilizin. Okay. Now, you can tell me, right, why can't zebra and giraffe mate? Like what? Or zebra hippopotamus? They cannot mate because this fertilizin and anti-fertilizin should be from same species. Then only the identification will happen. What we get, the mule, liger and all, that is made in the lab. Okay. In nature, generally the same species will have fertilizin and anti-fertilizin. That is why only male and female can work. Okay, male and female from the same species can function. Okay, do mate. Okay. Now, this anti-fertilizin and fertilizin is present, and they will be identified on the egg, on the sperm. Will be very specific. Now, once the identification happens, the sperm head will completely bind here. The sperm head will completely bind. So this anti-fertilizin on the sperm, fertilizin on the ovum, both are identified and they will join together. Okay. Now before I go further, let me give a small recap what we did right now. First step was what? Sperm will bind to the sperm receptor. Sperm receptor is generally present in zona pellucida. It will identify and bind there. Once it starts to bind there, what happens? The sperm undergoes capacitation and acrosomal reaction. Capacitation, entire sperm becomes more energetic, evolution type. Acrosomal reaction, three main things. Okay. Hyaluronidase enzyme will break down the granulosa cells on the outside. Corona penetrating enzyme will break down the corona radiata on the outside. Zona lysine, also called as acrosin, will break down the zona pellucida. One by one, every single layer of the entire ovum is broken down. If you see your NCERT diagram, you can see here. Corona radiata, zona pellucida, all of this is broken down one by one because of the enzymes in the acrosome. Okay. And once what happens? Finally, everything is broken down. Ovum has fertilizin, anti-fertilizin in the sperm. Both are identified and they are attaching. They are attaching now. Now students, once the sperm attaches here, there is a formation of a cone here. The plasma membrane will bend inwards and form a cone here, and this is called as, here there will be cone formation, that is called as fertilization cone. Point of attachment is fertilization cone. Now once the fertilization cone is formed, what happens? There will be opening of sodium channels. What will happen? Sodium channels will open up here. Sodium channels will open. Sodium channels will open. Now once sodium channels open, the entire plasma membrane gets depolarized. Once the sodium channels open, the entire plasma membrane gets depolarized. This is called as the fast block. As soon as one sperm even comes here, as soon as one sperm also comes here, what happens? As soon as one sperm is entering, fertilization cone formation happens, sodium channels open up, plasma membrane depolarization. Now once plasma membrane is completely depolarized, no more sperm can enter inside, or no more second sperm can even come and join there. Not possible at all. Not possible. This is called as fast block method. There is something called a slow block also. I'll give you the name. Explanation I will not do because it takes a long time. Okay. What is slow block? Okay. What is slow block here? Understand. There is something called as cortical reactions and zonal reaction. Zonal reaction happens. There is opening of calcium actually. There is here there will be cortical formation will happen. All this granules will be formed because of calcium ions formation. That you don't worry. That is extra information, too much extra information. Okay. This is called as slow block. This much you remember, that's all. Fast block means plasma membrane depolarization. Slow block is cortical reaction and zonal reaction. That happens. Okay. This is what happens. And once this happens, no more sperm can enter at all. No more sperm can enter at all, and it's finally done. Now once this happens, no more sperm can enter inside. Only one sperm is entering, one nuclei is entering. From here, what happens? See here. Only the nuclei fuses. You can see here, all of you. See, cone formations, fertilization cone formation, and the head part is released inside. The head part is released inside, and once it is released inside, students, once it is released inside, the male nuclei and the female nuclei will fuse. And the fusion of nuclei is called as amphimixis. It's called as amphimixis. And finally there is zygote formation. Then there is zygote formation. Okay. Then there is zygote formation. This amphimixis is also called as syngamy. Not fertilization, it is also called as syngamy. Syngamy. Okay. Syngamy, amphimixis. That is fusion of zygote formation. Okay. Significance of fertilization. The process of fertilization has following significances. Fertilization restores the diploid number of chromosomes, 2 in zygote, 2 variations, prevent polyspermy, determines the sex of the child. All these are significances. Okay. Look at this question. Capacitation occurs in. Tell me students, where does the capacitation occurs? Epididymis, no. Female reproductive tract. 2017 question. Look at this question. What happens when during fertilization in humans, After many sperms reach close to ovum, what happens? Two sperm enter, no. Secretion of acrosome helps one sperm enter cytoplasm of ovum through zona pellucida. Yes, answer is 2. Okay, and all of you are aware with the sex determination. Males have heterogamity, that is X and Y, the female as XX, right? This is very easy to teach. All of you know this very much. X in the case of female, in the case of male it is X and Y. If X gamete from here and X gamete from here fuse, will develop into a female. X gamete from here, Y gamete from here will develop into a male. This is very easy. Okay. Now students, again, post fertilization. Now once fertilization is over, what exactly happens afterwards, after fertilization? That is called as post fertilization events. Now what is happening here? Understand, all of you. Okay. Now can you see here? Here there is zygote formation. This right here is the ampulla region. Here fertilization takes place and there is zygote formation. Now this zygote right here, students, this zygote right here starts to undergo division. Now you might ask me, sir, which type of division? Is it mitosis or is it meiosis? Can anyone tell me in the chat? Anyone? Quickly tell me in the chat. Is it mitosis or meiosis? What exactly is happening to the zygote? The zygote is actually undergoing mitosis. But the mitosis here is a little different. Generally, mitosis has what? Mitosis has G1S, G2. Mitosis before M phase, there is G1S, G2. But in the case of this division here, G1 and G2 are not present. It does not take place. Only S phase, only the DNA replication goes on. G1 and G2 does not happen at all here. That is why the nucleocytoplasmic ratio is not balanced here. Okay, it is not balanced here. Now students, understand. Zygote, 2 cell, 4 cell increases. Each cell here is called as blastomere. It is called as what? Blastomere. So each cell is called as what? Blastomere. Don't get confused. Do not get confused. Each cell here is called as a blastomere. Okay. Can you see here? Egg, zygote, 2 cell, 4 cell. What is morula? Morula is actually when the cells are around 8 to 16. When there are 8 to 16 cells. That is called as morula stage. It is called as morula. When there are 8 to 16 blastomeres. That is called as morula stage. Morula. The morula gets converted to blastocyst. How? How does it get converted? There is a process called as hatching. Okay. Remember, there was a layer called as zona pellucida. Zona pellucida layer was not completely destroyed during the fertilization. Only small amount of it is destroyed. Okay. Now what happens here is morula cannot get attached to the uterus. Implantation cannot happen because of morula. Not possible. Why? Because morula still has the ZP layer around it. Morula still has the ZP layer, zona pellucida layer. That is why it cannot get attached to the endometrium, cannot undergo implantation. This particular morula has to completely lose the ZP, zona pellucida. That is called as hatching. It hatches and we get blastocyst. Cyst after hatching. Okay, so blastocyst is a layer which does not have zona pellucida. Okay, you can see here 2-celled, 4-celled, 8-celled, 16-cell is called as morula. Early blastocyst late blastocyst. Okay, late blastocyst. Now, if you look at a blastocyst, entire thing is called as a blastocyst. Inside the blastocyst, all the blastomeres, all of these are blastomeres, right? Blastomeres are arranged in two ways. How are they arranged? Inside blastocyst, all the blastomeres are arranged in the outer part, that is called as trophoblast. Outer part is called as trophoblast. Okay, can you see a round everywhere? Trophoblast. Or they are concentrated towards one corner, This concentration towards one corner is called as inner cell mass. It is called as what? Inner cell mass. Understanding, all of you? So blastocyst, inside blastocyst, all the blastomeres cells, they are either arranged towards one corner, inner cell mass, or they are towards the periphery, trophoblast. Okay. Now students, this inner cell mass eventually becomes embryo. Trophoblast becomes placenta. This eventually converts to placenta. Inner cell mass becomes embryo eventually. Okay. Now next step is called as implantation. Implantation, what happens? Remember, implantation can only happen because of blastocyst, not morula. Can you see here, diagram? Trophoblast is the outer part, inner cell mass on the inside. Okay. After attachment of blastocyst, the uterine cells divide rapidly. Here, this is the endometrium layer. The uterine cells start to divide very rapidly. They'll divide rapidly and they'll completely take the blastocyst inside. As a result, blastocyst becomes embedded in the endometrium of the uterus. This completes the implantation. So what is happening in implantation is the blastocyst, all the trophoblast layer is getting completely engulfed. All this uterine tissue, all the endometrium starts to divide very fast division and it gets completely on the inside. That is called as implantation. That is called as what? Implantation. Okay. Now they can ask you a question like this. Okay. This stage right here, student. I hope it is visible to all of you. This stage right here. Right. Identify the human developmental stage shown below as well as related right place of its occurrence, a normal pregnant woman. This right here has the outer trophoblast, inner cell mass. So this is a blastocyst. It is a blastocyst. It is found in the uterine wall. It undergoes a uterine wall. Okay. Okay. Uterine wall. Now, what happens after fusion? Very simple thing. Understand. Okay. After implantation, once the blastocyst gets implanted, after implantation, a finger-like projection appears on the trophoblast. All of you remember, right? This was a trophoblast. On this trophoblast, there is a finger-like structure called as chorionic villi. Okay. All of you can see this. Okay. So on the trophoblast, there is a finger-like structure called as chorionic villi. Here, chorionic villi on the trophoblast. Trophoblast, chorionic villi, which surrounded by the uterine tissue. Okay. So chorionic villi, finger-like structure called as chorionic villi. Chorionic villi plus uterine tissue of the endometrium, uterine tissue plus maternal blood. All of these three components are very important for the formation of placenta. Now what exactly happens here? Understand exactly. Chorionic villi and uterine tissue, both of them become what? interdigitated. They become completely interdigitated. See here, it's shown here. Can you see? This is the chorionic villi, and here is the blood vessels. They both of them, this is the actually placenta, entire placenta. They become completely interdigitated, which produces the placenta. Okay, see here. Chorionic villi plus uterine tissue become completely interdigitated along with maternal blood. All of these three together forms the placenta. It forms the placenta. Okay. So chorionic villi and uterine tissue becomes interdigitated with each other, jointly form a structural and functional unit between the developing embryo and maternal body called as placenta. That is all. What is it? Very simple to understand. Maternal blood as well as uterine tissue as well as the chorionic villi, interdigitation, placenta. Now students, tell me, if the baby is growing inside, will the baby require oxygen? Yes sir. Will the baby require food? Yes sir. Will the baby release CO2? Yes. Will the baby release waste? Yes. So all this waste release, sending of oxygen, all of this is mediated with the help of placenta. So mother, nutrition, placenta is the middle barrier, like broker. Okay, nutrients to the fetus, metabolic waste through the placenta, outside the mother. So all of this mediation is done with the help of placenta. But students, placenta has one extra function. Can anyone tell me what is that? What is that, students? What is that extra function of placenta which makes it very unique? The extra function of placenta is actually temporarily an endocrine tissue. It is an endocrine gland. Okay. It is temporarily an endocrine gland because it can also produce hormones. It can produce hormones. Okay, produce hormones. See here, all of you. Placenta will produce something called as HCG. Now, what is this HCG, students? HCG stands for human chorionic gonadotropin. Human gonadotropin, HCG. HPL, human placental lactogen. Human placental lactogen. Then there is relaxin, which helps in relaxing the body. Yes, relaxing the ligaments during the childbirth also. Then there is estrogen, progesterone. All of this is produced by the placenta. But there is a question here. What is the question? Which hormones are released only during pregnancy? The answer is 3 of them. HCG, HPL, relaxin. These 3 hormones are released only during pregnancy, not any time at all. They are not produced at all. Only during the pregnancy, HCG, HPL and relaxin. They produce only during pregnancy. Okay. Relaxin is also produced by the ovary. Remember that. Okay. So the question is here. See. Tell me the answer. Only during pregnancy. Relaxin, HCG, HPL. ACD. Right? ACD. Option number 4. Or they can ask a question like this. Pregnancy is characterized by metabolic changes in the mother. Yes. Metabolic changes happens in the mother because of the hormones. During pregnancy, the level of thyroxine is increased in the maternal blood. Yes. Thyroxine also increases in the maternal blood. Okay. Now, if that is placenta formation, now placenta is formed, everything is going well. Now, that is with respect to the trophoblast. Now, what is exactly with respect to the inner cell mass? What exactly happens to the inner cell mass now? Now, inner cell mass undergoes a process called as gastrulation. That is a process by which inner cell mass gives rise to the structure called as gastrula. Gastrula. Okay. Now, what exactly is this gastrula, students? This gastrula is a structure. This structure here, inner cell mass, gets converted to a gastrula structure called as three main layers. It has something called as outer ectoderm, middle mesoderm, and inner endoderm. Three different layers. The best part is all of this is made up of stem cells, and these stem cells can give rise to any type of organ present. Now you will learn more about this in the animal tissues, that is structural organization in animals. But right now I've made a small flow chart for you to remember what is present of endoderm, mesoderm, ectoderm. What are the organs produced by each one of them? You can look at it, download the notes. Notes are available in the Telegram channel. All of you know that. Okay. Now students, the female is the embryo is developing now. Ectoderm, mesoderm, endoderm. All the different layers is developing. It is forming different layers now. Forming different layers. Now, as the embryo is developing and the fetus is developing, there are changes happening every single month or with respect to trimester. Now here I have made a table for you which will help you to remember this entire thing in a very smooth fashion. Understand? See here. Change the embryo. There is first trimester. Now if I ask you what is first trimester? First trimester means 12 weeks. 12 weeks. Second trimester starts with 16 weeks, ending is 24 weeks. Third trimester, 36 weeks, 9 months. So I have made a table for you here which will help you to remember the entire thing very smoothly. Okay, very smoothly. What happens in the first trimester or the first month? In the first month, baby grows rapidly, heart is formed, and you can hear the heartbeat. The heartbeat can be heard first month. Second month, limbs and digits are formed. Third month, that is end of first trimester. End of first trimester, limbs and digits, genital organs are formed. So all the major organs are formed by the end of third month or end of first trimester, 12 weeks. What happens in the 4 months? This is not given in your NCERT. I have given this extra. Ability to taste, hear. Baby can hear now. Okay. Now, 5th month, very important. 5th month, movement can be seen. That is, people say, right, the baby is kicking. They say, right, baby is kicking, football player. The kicking is happening because of the 5th month. First movement is seen. Okay. Hair on the head appears. By the end of second trimester, 6th month, what happens? Eyelid separates out. Body is covered with fine hair. Everything is done by the end of second trimester. Then what happens? This is not given NCERT, don't worry. This is also not given NCERT, don't worry. The extra information I have given you. Okay. Frequency type movement, brain occurs, growth of brain occurs, not given NCERT. Directly NCERT is going to 9 months now. What happens 9 months? 9 months fetal turns upside down with head near the cervix and the baby is ready to come out. 9 months. You can see here. This is the first month. Can you see? The heartbeat can be seen. Okay. Angiogram. Second month of pregnancy. Limbs and digits. Second month. First trimester. Major organs are found. First trimester. Movement of the 5th month. 5th month, movement of the kick of the baby. Okay. End of second trimester. Fine hair on the head. Eyelid separation. Okay. Eyelashes are formed. End of second week. Finally, during the 9th month, the baby is ready to come out. 9th month, the baby is finally ready to come out. Okay. Good morning to all of you. Okay. And finally, the last topic for today is called as parturition. That is, by the end of 9 months, the baby is like, enough bro, enough. I want to come out to the world. Okay. So process of delivery of the fetus is called as parturition. Now you might ask me, who will send the signal? Who will send the signal that baby is ready to come out? The signal is actually sent by two people. The signal is sent by the baby itself. The baby only is telling you, I want to come out. Or it is signal is sent by the placenta. Okay. And the first signal is called as fetal ejection reflex. The fetus is ready to eject out. Okay. Delivery of the fetus is followed by expulsion of placenta. Okay. Fetal ejection reflex. This is how the fetus comes out. Okay. Now students, this entire process of baby coming out is a complex neuroendocrine mechanism. Fancy, no? Fancy? Very easy to understand. Okay. The baby is ready now. Baby is like, I want to come out. I'm like, not possible, bro. I want to come out of the world. The baby and placenta will send a signal. The baby and placenta will send a signal. The signal will go to the mother's brain. The signal will go to the mother's brain, and from the mother's brain, what releases? From the anterior pituitary, from the paternal pituitary, oxytocin is released. Not anterior, pituitary, oxytocin released. Once oxytocin is released, oxytocin causes uterine muscle contraction. Signal is sent by the baby, mother is releasing oxytocin. Oxytocin is causing the muscle contraction. The muscle is contracting. Contractions are coming. Now muscles contract, more oxytocin. More oxytocin, more contraction. More contraction, more oxytocin. So it happens in a cycle. More contraction, more oxytocin. More oxytocin, more contraction. So it will keep on happening until the cervix is wide enough for the baby to come out. For the birth canal to be open enough, baby can come out. And that is called as parturition. Now after parturition, the baby needs food. Now it is recommended by the doctor that the baby gets only the breast milk of the mother starting. Why? Because it is rich in antibiotics. Okay. The mammary glands produce milk towards the end of the pregnancy. Process of breast milk to the baby with the production of mammary glands. The early milk of mother is called as colostrum. It is called as colostrum. Milk produced in the initial days of lactation. Colostrum is rich in antibiotics, especially IgG, and it provides protection to the baby, telling that everything is alright. Baby can develop very well. Okay, develop very well, and that is the entire mechanism. Now here there is one small point which I want every single student of mine to understand. Prolactin stimulates milk production. First point. Prolactin will stimulate the milk production. Oxytocin stimulates the milk release. So oxytocin will release the milk. Prolactin will do stimulate of milk production. So this is very important. Okay. Oxytocin, milk let down. Prolactin, milk production. Okay, milk production is by prolactin. Oxytocin is releasing the milk. All of you should remember this. Two main points. Okay. Last question on the board for all of you. Breastfeeding during initial period of infant growth is recommended by doctors for bringing a healthy baby. True. Colostrum contains several antibiotics, absolutely essential for developing resistance to antibiotics of new baby. Also true. So the answer is option number 4. 4 is the answer. And that is the end of the long marathon of Bio Club. Done. Students, if you go through the lecture one more time, if you miss out any point, let us know in the comment section, because this is what we call as Bio Marathon. This is what we call as Bio Club. You learned so many concepts today, so many concepts, all the way from the plant reproduction to the human reproduction, in detail, from basics to the most advanced concept. Okay, most advanced concept you learned today. We did not put any shortcut methods anywhere. No shortcut. This was pure concept. Every single topic was covered, from the most basic one, Okay, next one is gonna be next one is gonna be human health. After that we'll do genetics. After we'll do genetics. Okay. Now students, before we go, I want everyone to comment. I want everyone to comment in today's lecture. I want everyone to comment in today's lecture. Right? Day 1 completed. Day 1 completed. If you really watch the entire thing. If you really watch the entire thing, then you should comment day 1 completed in the comments today's video. Day 1 completed with the bio club. And if you attend the entire video, if you watch the entire video, you have got some key pass, you have got 40 marks, yours. No one can take it away from you. Okay, no one can take it from you for you. And if you go to the description, students, human reproduction, plant reproduction, quiz is given to you. Attempt the quiz, check your knowledge by attending the quiz. If you click here, you have to fill the Gmail ID. If you fill it up, attend the quiz right now. Okay, so you can start attending the quiz. And if you want all of these notes are available in the Telegram channel, Basvaraj Sir Biology or Gopika Ma'am Biology. Okay, so with that, students, thank you so much for today's class. Have a great morning. Have a great morning. So bye bye. Take care, all of you. Until then, I'll see you in the next class. Bye bye. Have a nice day, all of you. Bye bye.